No, you're not shouting. Good morning, everyone. I know we're not all here, but if I can ask us to please start taking your seats, take your coffee with you. I know it's early. Thank you for being here on time. We're a little bit behind, but I think we'll catch up. I hope you're awake and rested and fed. All right, we're in for a really nice, exciting morning. I know lots of people mentioned that they would like to refresh their immunology and vaccinology basics, and that's what we're doing for this morning session. All right, so in the interest of time, I'm handing over to Prof. Elisa Nemes, who's going to take us through the basic principles of immunology and applications in vaccinology. So over to you, Prof. Thank you. Sorry, it's early.
Thank you so much. Thank you. Good morning, everyone. How are you doing? Are my slides up there? Oh, yes, they are. So it's a huge pleasure to be here another time. And I always start with this picture. And every time I think, oh, maybe I should change it. Then, on Sunday, I was sitting with some of you. And when I say I do the immunology lecture, that's kind of the face I got. No, it wasn't that terrified, but there was a little bit of terror. So I see some of them here. Hello. So I think my job is to try and take that expression away, not just because it's so early in the morning. Yeah, I think everybody who works in vaccinology or has an interest in vaccinology, even if you're not an immunologist, immunology really underpins how vaccines work. So what I will try to do is to give you an accessible overview, and then in the Q&A session, we can go a bit deeper if you want to, or go back to some more basic concepts. So when we talk about the immune system, it's a system because it's not just one organ like your liver in one place. And the reason why we need a system to protect us against pathogens is because pathogens arrive from in different ways. And so when you think about how we can be infected, it could be through direct contact, it could be exchange of bodily fluids, it could be contamination of food or water, it could be airborne, it could be a vector. And so what our body needs to stay healthy is physical protections. And so these will be the skin and your mucosal surfaces. That's basically what separates inside us from outside us. And then a complex system, which is composed of different organs, cells, and molecules. And I will try not to name any molecule, or at least I will not talk about 50 different cytokines. It's a promise. Just a few cells. some of them. Okay, so this is a big picture of how an immune response to infection works. And what I really would like you to take away from this is that there are two key concepts in immunology. One is the innate response, and the other is the adaptive response. So the key features of the innate response is that it's quick. So the cells of the innate immunity responds very quickly to pathogens. It's not specific and does not remember. This means that an innate immune cells, broadly speaking, there are always exceptions in immunology, always. But broadly speaking, will respond in a very similar way every time it sees a pathogen, which is different from what the adaptive immunity does. So the adaptive immunity is slow. It takes days to respond, and we'll see why that is. The key feature is that it's specific. So a certain T cell or B cell will only recognize a certain portion of a specific pathogen, and it remembers. And we'll talk more about this, but the fact that adaptive immunity remembers is really the underpinning concept of vaccines. I try not to use too many technical words, but there is one that I really need to use, which is antigens, because I'm going to say this a lot. I'm going to explain what we mean for antigen. So an antigen is any structural substance which is recognized by the adaptive immune response, so B or T cells. And then we have two big distinctions. We have self-antigens. It means it's any molecule, really anything within the body. Normally, in an healthy immune system, there is not an immune response against self-antigen. If this happens, then you have autoimmunity, which is a pathology. What we're mostly interested in vaccinology are known self-antigen, which means that are substances that don't come from within. They are foreign to the host. And these are typically proteins or large polysaccharides, and they are the substances that do induce an immune response. That wasn't that bad, was it? Okay. Again, breaking down the basics and the different types of immune responses. So we mentioned innate immunity earlier. Two key cells are macrophages and dendritic cells. There are others, but these are the ones I want to focus on. And their job is to recognize molecular patterns on microbes. Once they do that, they communicate danger signals to other cells. That's inflammation. And the other key job that they have is that they present microbial antigens to the adaptive immune cells. And we'll go a bit more into details about that. And then we have two types of adaptive immunity. We have the cellular and the humoral immunity. The cellular adaptive immunity is composed by T cells. They recognize specific antigens, but they can only see proteins. They don't see polysaccharides. This is a very important point. And they only see proteins when they are presented to them by the antigen-presenting cells. That's why we also need to understand a little bit of innate immunity when we talk about vaccines. They do different things. They either kill infected cells, and these will be your cytotoxic T lymphocytes or CD8, or they orchestrate a broader immune response, and those will be your helper T lymphocytes or CD4s. The humoral immunity is what many people who are even not in the field are used to think about immunology. It's composed by B lymphocytes. They also recognize specific antigens, but contrary to T cells, they can recognize both proteins and polysaccharides. So that's important for all those bacteria that cover themselves in polysaccharides. And their main job is to produce antibodies. So digging a little bit deeper into the adaptive immunity, we say there are two different branches, if you want, humoral and cell-mediated. Other key differences are that B cells can recognize intact antigens. It means they see a protein, they see the protein. While the T cells, they only recognize bits and pieces of that protein, and that's what the antigen-presenting cells do. They chop up proteins and present to the T cells. And then once they recognize the antigen, which is very specific, that's why I have that key and the lock to remind me that. So every T cell or every B cell only recognizes a very specific portion of a protein or a polysaccharide in the case of the B cells. So once they recognize their antigen the first time, what they do is that they multiply. Because you can imagine if every T cell or every B cell can only recognize one thing, we don't have very many of them. Otherwise, our body will just be a bag of B and T cells. And so these initial naive cells that carry receptors that are specific for these substances are very, very rare. So what they do is that when they see the antigen, they need to make more of themselves because otherwise, like, 2 cells are not going to stop an infection. And this is one of the key reasons why the adaptive immunity is slow. That time is what it takes for the cells to make more of themselves. While the innate cells, they just recognize all of the pathogens, and so they are already there in sufficient numbers. So when T and B cells recognize the antigens, they make more of themselves, but they also change, they grow, they differentiate, and they become more specialized to accomplish different tasks. So for example, B cells differentiate into plasma cells that secrete antibodies and defend against extracellular pathogens. This is the other key difference between the two. And then you have memory B cells that go in the bone marrow and sit there for a very, very long time. T cells also proliferate and differentiate into activated or effector cells and memory cells. And the key difference here is that T cells generally defend against infected cells, cancer, and transplant tissues. In a way, B cells see things that are outside other cells, and T cells see things that are inside other cells. And so what that means is that antibodies or the humoral immunity is generally more important to prevent infection, that initial infection, because they can see a pathogen or a toxin before that pathogen gets into other cells, while T cells prevent severe disease. So T cells generally do not prevent infection, but they contain it. They can contain it very well, so it doesn't spread or it doesn't cause more severe disease. So how do we use or how do we apply this principle of immunity in vaccinology? Again, we can distinguish active and passive immunity. Active immunity can be natural, so it's when we are infected by something and we develop our own natural immunity to it, and can be artificial, and that's what vaccines do. So they mimic this exposure to a real infection so that you can induce this immune response before you actually see the pathogen. And then passive immunity can also be natural or artificial. Natural example is maternal antibodies that can be transferred to the fetus or the breastfeeding baby. And then there is also artificial passive immunity. For example, some biologics and therapeutics, we can even infuse monoclonal antibodies that are synthetic into people. Okay, so the key concept is that vaccines prevent disease by teaching our immune system how to fight off infections before they see the real thing. So, but what's in a vaccine? So mostly water, actually, and then there are some preservatives and stabilizers so we can store them for some time. There could be residual traces of the way the vaccines are produced. But the two key ingredients that we're interested in here are adjuvants, and we'll talk more about adjuvants. There are substances that we add to vaccines to induce a better immune response. And then the active ingredient. The active ingredient is what makes the vaccine specific to a certain molecule in a certain pathogen. So those are really the two key components in a vaccine. One thing to note is that generally, some people can have allergic reactions to vaccines, and those are generally against the preservatives and stabilizers or the residual traces more than the specific components of the pathogen that is in the vaccine. So again, very basic concepts, but the way the basic components of a vaccine are put together will shape which type of immune response we're going to get. So the antigen, so the exact piece of the pathogen that we put in a vaccine will determine the specificity of the immune response and the breadth. The breadth, it means how many molecules the immune system is going to recognize. If you put one antigen in your vaccine, you will have a response to one antigen. If you use a live attenuated vaccine, you will have a response to most components of the pathogen. Formulation is very important, and this we can play with the type of vaccine. Again, I gave a couple of examples. You can have a subunit and adjuvant, you can have a live attenuated, you can have RNA, etc. And then the way the vaccine is diluted, the dose, so how much of it we give, will determine the quality and the magnitude of the adaptive response. The liver is also very important. So how do we give it? That is really essential. So the immunization schedule. At what age do we start? How many doses do we give? How much do we give in every dose? This and the route of administration, we'll talk a bit more about that, as well as age and special groups. All of these are decisions that many people in this room are going to make, and they determine the longevity and the effectiveness of the vaccine. This is a busy slide. I'm not gonna expect you to read it. You will have all the slides, but it's just a really nice summary of different types of vaccines and how they developed over time from the 1700 up to a couple of years ago. And it just shows how the evolution or the increase of our understanding of host pathogen interactions and the development of biotechnology allowed us to design more rational vaccines, more complex vaccines that are more efficacious and safer. So now we try to put it all together, all these basic principles, and we see what happens inside the body when you vaccinate somebody or in case of natural infection. But it's a vaccinology course, so we'll talk about vaccines. So in this example, we inject a vaccine in the muscle. And so the first thing that happens is that this engages your innate immunity. So your innate immune cells will pick up the antigens in the vaccine, they will process it, and they will release these danger signals. After they do that, they move. So they move from the site of vaccination or the site of infection, and they go to the lymph nodes. And while they go to the lymph nodes, they mature. They also change what they do. Now, this very initial innate immune response is what determines the inflammation and the local site reactogenicity. So if you think about when you get the vaccine, in a few hours, you can have some redness, some pain, swelling, etc. That is your innate immunity that's working. So it can be unpleasant, but often it's a good sign. It means something is happening. Then what happens? Then these dendritic cells, they get into the lymph nodes, and the lymph nodes are like a hub where then the B and T cells can circulate, and they stop there to see if their antigen has been presented by somebody. And so, for example, the dendritic cells will present the antigen to T cells, both CD4 and CD8. And as we saw before, these cells then proliferate and differentiate. And this is because, as I mentioned, we have these very rare naive adaptive cells that need to undergo what's called clonal expansion. It just means they make more of themselves and differentiate. Also in the lymph nodes, there are B cells, many of them. So B cells can also see the antigens directly. They also need to undergo proliferation, and then they differentiate into memory B cells and plasma cells that make antibodies. a key concept here in vaccinology is that if B cells do not receive help from T cells, they are not going to make memory B cells. They can make antibodies for a while, but they will not sit around and give you long-term memory. So even if a vaccine is inducing antibodies to protect, you still need the T cell help for those B cells, not only to make antibody, make more and better of them, but critically to make memory so that those B cells can give you antibodies for a very long time. So all of this process, I hope it wasn't too complicated. This is like really the basic steps of how it all happens. But there are age differences. So like everything, when we are born, our immune system is immature. So it means it doesn't do things as well as after a while it's been exposed to pathogens. So I know we vaccinate babies because we want to protect them as soon as possible, but there are some features of their immune cells that are not as good as older children, for example. And then you can also have instances where these maternal antibodies that can protect the child, they can also interfere with some of the vaccines because, for example, the measles vaccine is a live attenuated vaccine. And so if we give it too early and there are maternal antibodies around, the maternal antibodies will neutralize the vaccine before it can actually give you a good immune response. That's one of the reasons why the measles vaccine is given a bit later in life. It's not in the first weeks or the first months of life. Then the immune system matures. Then during pregnancy, wonderful things happen. So if you remember, we talk about self and non-self antigens. So a baby is essentially a non-self antigen, right? That grows inside the body. And so the immune system needs to change to make sure that it doesn't attack this foreign object that's growing in the body. And so there is all sorts of modulations and induction of tolerance during pregnancy to make sure that the pregnancy can go on. But that, if you think about the mother, it's a moment of vulnerability where the immune response doesn't function in the same way as when a woman is not pregnant. And so that is a very important group we need to think about when we develop vaccines. And then there is immunosenescence. So like all other things, as we grow older, things get slower and harder and start to fall apart. And our immune system does the same thing. So vaccinating elderly people, you will not get the same immune response as younger adults. And that is why sometimes you need to use, for example, a higher dose or more injections. And this is because their immune system is not as good as a younger person immune system. Okay, location is another very important thing. So we say the immune system is a system. It's everywhere. There are certain organs like secondary lymphoid organs, lymph nodes all around the body, but the cells also sit like just under these mucosal surfaces, etc. So it's quite important not only that we can induce an immune response, but it needs to go where we need it. Because if I have all my T cells in the knees and I'm exposed to tuberculosis in my lungs, they're not going to be very useful. Ideally, I want pre-existing immunity in my lungs. And so one of the ways that, and there is some form of imprinting, depending where the cells are primed, they will tend to go there. This is mostly T cells, antibodies, they can float around. But essentially, the route of vaccination, which is something we can control, right, can also affect where that immune response is more likely going to be. So, for example, the most common routes of vaccination, intramuscular or intradermal, will give you a very good systemic immune response, but maybe not as good on the mucosal surfaces when you compare, for example, to oral vaccination for polio. You want the response in the gut, because that's how you're exposed. Or there aren't many examples of aerosolized vaccines, but obviously that's very fashionable with COVID and influenza, etc., because you can imagine things that we breathe in. If you already had an immune response that stops it there, then logically it will probably be better. Okay, so the key concept, how we apply immunology to vaccines, is this concept of memory. And I introduced that already. The first time our immune system sees a pathogen or a vaccine, it takes a long time for adaptive responses to develop. You're looking into weeks. So once that memory is induced, the second and third and the fourth time we see the same antigen, our cells are already there. They multiply, they differentiate, they are where we want them to be, hopefully. And so the response is much faster. Now the adaptive response also takes hours, but we need to induce it first. The other feature is that it's stronger because these cells already proliferated and differentiated, then they're already there and geared up to do what they need to do. And better. This is a very scientific term. In particular for antibodies, you have class switching, they change, they increase their affinity, etc. So the more these cells are exposed to pathogens, the more they respond appropriately when it works. So for how long does this memory last? It really depends on a lot of things. Some vaccines, generally live attenuated vaccines, are very good at inducing lifelong memory. Think about the yellow fever vaccine, one shot, you're good for life. In many cases, the memory wanes, and this depends on the type of immune response that is induced, natural exposure to the pathogen, population, environment, etc. So there are different determinants of duration of immunity. So what we've been talking about until now is this priming event. So what happens the first time our immune system sees a pathogen or a vaccine. But so now if the memory starts waning, what we can do is to boost it. That's why, for example, for your DTP vaccine, we should all get it every 10 years. Because to keep that memory up. A very interesting concept that I'm surprised has not been explored as much before COVID is this difference between homologous and heterologous prime boost. So most of the vaccines that are now given in multiple doses like DTP, etc., it's the same vaccine, like it's the same formulation, you just give it again. While with COVID, what we saw, because we had so many, we were spoiled, and also not all vaccines were available at the same time in different places, etc. Then people got what they could get, right? So I got J&J and then Pfizer. And then there have been all these combinations. And actually, there are quite a few studies that show that it might be better. And if you think about it, you can have different vaccines that shape or direct different aspects of your immune response. And if you combine them or you give them sequentially, you can develop a much broader type of immunity, because maybe you give a vaccine that's really good at inducing antibodies, and then you give one that's very good at inducing T cells, and then you have both. So the other concept that, for example, I work on tuberculosis, so it's something we're very used to, but it sounded like a new thing. There is a new term, very sexy, hybrid immunity. We used to call it pre-exposure, pre-existing immunity. Now it's hybrid, but it's cool. I love it. I now use it in all my TB talks. And so this is when you combine natural immunity with artificial immunity. So you can have all sorts of combination. You can have previous exposure. So you have some of your natural immunity, you vaccinate, or you first vaccinate, and then you can still be infected, but not as sick. And apparently this combination of natural and artificial immunity is better. Again, it's a very specific term. There are different features of the immune response that change when you combine both natural and artificial immunity. So the other key concept why immunology is so key in vaccinology and how do we use it like practically is when we think about correlates, immune correlates of protection. So what are immune correlates of protection? They're measurable immune responses that predict vaccine efficacy. It's something that you can measure, for example, in the blood and will tell you if that person is protected or not. It's very practical having something like that, right? And for the vaccines for which we know what immune correlates of protection are, they're normally defined in a very specific way. It's going to be, I don't know, a neutralizing antibody against this antigen at this concentration. So there is also a threshold. So you can go measure the correlate of protection, and depending on how much it is, you can predict if that vaccine will give you lifelong protection, if the immunity is waning, and so it might need boosting, or unfortunately, in some cases, there is no protection at all. And so this is really one of the applications of immunology that can really guide what then is done on the ground. And this is how, for example, the yellow fever vaccine, it used to be every 10 years and then started measuring immune antibodies that are one of the correlates of protection. And then they realize, well, but they're still there. And so based on that, then WHO changed the policy. A key difference is that correlates of protection are not necessarily correlates of risk. So when we talk about correlates of risk, are immune responses that are prospectively associated with disease risk, regardless of vaccination or vaccine efficacy. So these, for example, are things that we study to better understand immunopathogenesis of a disease. For example, in a cohort study, you measure immune responses, you follow people up, you see who gets sick and who doesn't, to try and understand the mechanisms a bit better. While correlates of protection are mostly, not only, but mostly defined in the context of a placebo-controlled clinical trial with the clinical endpoint. It's a mouthful. But the key concept here is that just like when we measure vaccine efficacy or protection, we need to define protection against what. And again, there are some important examples from what happened during COVID where there was a bit of confusion between, prevention of infection, prevention of disease, prevention of severe disease and death. And so there were different studies measuring different endpoints. And then, for example, some vaccines were kind of disregarded because they weren't so good maybe at protecting against initial infection. But it turns out they were very effective in protecting against severe disease and death. And so it's very important to define what your endpoints are when you design a clinical trial, but also this trickles down to the immunology that you do with it. You need to define if you want a correlate of protection, is a correlate of protection against what? So why am I talking about this? Why are correlates of protection useful? For example, if you have a vaccine with an unknown correlate of protection, so TB will be an example of that, you have no way, like you have a new TB vaccine candidate, you need to go into efficacy trials, you need to vaccinate. Like now there is a phase 3 trial with 20,000 people, it costs $500 million. And you vaccinate 20,000 people and you wait several years to see who gets sick. There are other vaccines that have an established human challenge model. For example, the malaria, there are many others, but malaria, salmonella, etc., they all have human challenge model. These have really transformed how we do research for vaccines in these diseases, because we can take a very small group of people, infect them on purpose safely. But these studies have been really key to define what immune responses are correlated with that protection. And then you can use that correlate in a big study. So when you have vaccines with known correlate to protection, you don't need that are accepted by regulators, etc., but you don't need to do big efficacy trials anymore. You could vaccinate a smaller number of people, measure the correlate of protection, and infer whether the vaccine is going to work or not. So this is key. For example, every year we have a new influenza vaccine. Imagine you couldn't do an efficacy, like the time you've done an efficacy trial, you have a new strain. So it's licensed based on correlates of protection. Or for example, you do your efficacy trials in certain populations, and then you want to bridge to a different population. So you could do a smaller study where you measure your correlate instead of doing efficacy in all of them. So for most commonly used vaccines that work, antibodies have been defined as the immune correlate of protection. And these are mostly acute infections where where natural immunity is protected. So if you got measles once, normally you don't get it again. While for other pathogens that are more chronic, persisting infections where natural immunity is only partly protective, so examples TB, malaria, HIV, etc. We don't know if antibodies are a correlate of protection, but the thinking is that the immune response that you need is way more complicated and probably also involves T cells, which are much harder to study. And that's also why there are less studies on T cells than there are on antibodies. So the other reason why understanding correlates of protection is so important is because you can use them to measure herd immunity. And so this diagram just shows that if you have a completely naive population, imagine back in 2020, and there is a very contagious infection that comes, it just spreads in the population very quickly. But if you have pre-existing immunity, either given by natural exposure or by vaccination, and if this pre-existing immunity stops infection, then it means that even the people who are contagious, they cannot transmit so easily to the population. And so there is this indirect protection for people who maybe cannot get vaccinated for various reasons or have some immune conditions that they get vaccinated and their immune response doesn't develop properly. They can be protected by the people that surround them if they themselves are protected. So when we think about vaccine efficacy, I didn't go into a lot of details of this, but I briefly touched on all these points. What determines whether a vaccine is going to work or not? It's many different aspects. So you start from which type of vaccine you're going to use, which dose, how you're going to formulate it, how you're going to administer, so the route of vaccination. And then there are big differences, different ages, different ethnicities, different, so nutrition, microbiome, that's a whole thing, etc. And then it also depends which type of immunity, of protective immunity you need. Generally, I'm a T cell person. Making antibodies is easier than try to develop a T cell vaccine. So it kind of really depends which immune response you need. So there are many barriers to vaccine development and success, which are summarized in this nice review. What I tried to do today is to touch a bit on how understanding how immunity develops and specifically how it develops for different types of pathogens is important to inform vaccine design, to inform how we give it, how we monitor it, etc. But there are many, many other aspects that touch on implementation, host variability, the pathogen variability, of course, vaccine safety, and then environmental and socio-demographic factors. And you're going to hear about many of these during this week. And so hopefully at the end of the week, you will have this big puzzle that shows you what opportunities and challenges there are. And with that, I hope it wasn't too bad and that you feel a little better. And now it's my turn to be scared because normally you ask me a lot of questions I don't have answers to. So I will do my best. I thank you for your attention and we can have a bit of a discussion now.
Thank you so much, Prof. Neves, for a very accessible, I will just say that, and thorough presentation. Thank you so much. We already have questions, so here you go.
Thank you so much. That was very interesting. I was just wondering if you could maybe describe a bit around how you get reservoirs and carriers if they are not unwell, but how the immune system allows them to still transmit the disease.
That is a very interesting concept. So, um, when we talk about carriage or colonization, am I understanding where
Yes.
you're going?
Yeah,
Okay.
yeah.
So some pathogens like strep or pneumococcus, etc., they can basically live on the surface of the mucosa and transmit to other people without giving disease in the person who hosts them. And the way that that's maintained, I think, is very similar to how we interact with our microbiome. As you know, like two to three of the kilograms that we weight are germs, not pathogens, but our microbiome. And so we need them, and our body and immune system has evolved to cohabitate with the microbiome, and similar mechanisms apply to colonization. So the immune system will sense that there are these pathogens on the surface, and has a different way, a more tolerogenic way to interact with it, where there aren't these danger signals that will trigger a full-on immune response. So what happens with this is also, it depends on the location. For example, strep pneumonia, if it just sits like in the upper respiratory tract, that's how, you sneeze, you breathe, and then you can aerosolize it and give it to other people. But it's only when it moves to the lower respiratory tract that can invade tissues and then go places and create damage. So that's when then there is pathology. So there is this very, very fine balance of the immune system, because you can imagine if we react against everything, like you couldn't eat, and so it's really all about this balance and understanding what's dangerous and what is not. And partly that's why this innate sensing is so important, because basically the innate cells are the ones who decide, am I going to ring an alarm and call a whole lot of other cells and initiate an immune response, or is this not harmful, so I'm just going to leave it there?
Just to, follow on from that, is that, would you say then more for bacterial infection rather than a viral or the infection?
I think so, because viruses normally they need
Inside.
to infect a cell to replicate. So I would say some bacteria, can very happily just survive like on a surface of your mucosa, but viruses, I don't think they can.
Okay, thank you.
So I wanted to know how many times can I get a booster dose if my titer always remains negative? Is it safe?
That's more of a question for a medical doctor, but I don't think there are safety problems. There can be many reasons why you get the vaccine and you don't develop a particular immune response. There could be some polymorphisms that, I don't know, interfere with that specifically. But I don't have a number. I don't know how many times you can get it and be safe. I think generally vaccines are safe. I wouldn't just get them too close because it doesn't help. Like you already have a dose, things are happening, or maybe things are not happening, because if then you don't develop antibodies, maybe your T cells are not that strong. But so given too many doses too close doesn't really help. Like you need to let things take the time they need. Sorry, I don't have a better answer.
No, you're fine.
I just have a question with regards to using correlates of protection. So if you use it to assess efficacy and it reduces the need for large phase 3 clinical trials over long periods of time, how do you how do you balance that with ensuring long-term safety?
So that, I guess that comes also from your pharmacovigilance. So the thing is that to identify the correlate of protection, at some point, you will have to do those trials, at least a phase 2b. And so you would have already gathered a lot of safety data. And then when a product is licensed, then there are phase 4 studies where you keep collecting safety data. Actually, the talk just after me, it's about vaccine safety. The correlates of protection are more useful if you want to bridge to other populations, etc. It's because they can be specific for a type of vaccine. And in any case, like once the vaccines are licensed, then the numbers of safety observations really increase a lot. And so then it's not anymore a comparison to a placebo, for example, but it will be kind of population studies where you constantly monitor if there are specific safety signals associated with vaccination. For very, very rare safety signals, you can't even pick them up in a phase 3. Like it's only when you go to a whole population that you may pick it up.
Question in the back left corner. Thanks.
All right, I wanted to ask, how can validated data on good data on COP, COR, and ISC can accelerate approvals or emergency use in a country like South Africa?
Well, they can, again, because it depends on the scenario, right? It depends on the pathogen and the vaccine, etc. But it can, because if you don't need to do big trials and you can license based on a correlate, depending on the disease, doing big trials takes a long time. I think about the TB example, right? Like any efficacy trial, you have at least 5 years follow-up. It's not for COVID, it wasn't the same. Like in a pandemic situation, you have 6 months and you have all the endpoints that you need, or when there were the Ebola outbreaks, etc. But the key thing is that it depends. If it's an established correlate of protection for a vaccine that has already been licensed somewhere else, it could be used, like you could do a small study, but you need the local regulators to accept that correlate. So you need to talk to them before even trying to do something like this. And that's useful, like in many countries where rightly so the local regulators want local data, like to say, okay, is this vaccine going to work in my population? Then you could do a much smaller study. If you had a validated correlate, you could do a much smaller study, just demonstrate that the vaccine given in that population, in that context, can induce the correlate of protection to the level that's protective. So I wouldn't say South Africa is any different from any other scenario where you could apply this if you have when you have a validated correlate.
Okay. We have, good morning, Elisa.
Hi.
A question from Alan Baguma. He's saying, thank you for the great presentation. And I just want to note that there's quite a few comments online that are saying you explained the concepts in a simple language that everyone could understand.
Thank you.
So they're grateful for that. So Alan's question is, why do populations in Africa demonstrate less vaccine efficacy and effectiveness compared to populations in high-income countries?
I guess it depends on the vaccine and the pathogen. I'll give you an example that I know very well because it's BCG, the vaccine for TB. For some reasons, it looks like it's less effective in tropical area around the equator compared to more temperate areas. One of the reasons could be that there are many more environmental mycobacteria in tropical areas, and these are very similar to Mycobacterium tuberculosis. And the fact that people have immunity against these other mycobacteria can either mask the BCG efficacy because you're already protected by your cross-reactive immunity, or could block BCG, similar to the example I gave for measles, so that BCG then doesn't work very well. I would say, although it is really specific, depending on the vaccine and the disease, in general, good nutrition is important for everything, and socio-demographic aspects are important for a good immune response. So depending on the areas, in communities that are more deprived, there is more exposure to other pathogens, and there can be socio-economic factors that make it that our immune system actually works a bit less well. And so the immune response to vaccines might be a bit lower. It could be some genetics, but genetics is not that as common to say, this vaccine doesn't work in that country. There could be certain polymorphisms, as I was mentioning earlier, that make certain immune responses a bit different, but I wouldn't say that really accounts on population level observations. So there are many reasons. I don't know, I see Greg, maybe you want to add something or no? Okay. Yes. Yes.
And because of enteric viruses and breast milk feeding, etc., etc. But the antibody responses to rota and polio tend to be less in Africa compared to the more developed countries.
Okay, thank you. And it's also like probably this continuous more or less exposure could be good or bad. Like if you are constantly exposed to something, it keeps your immune memory up, but it could also maybe interfere with the immune response that follows.
And then can I ask you to grab her?
Good morning, Prof.
Morning.
Thank you very much for your presentation. I have one question. Normally we always say we give children second chance for vaccination in case they did not seroconvert. So I want to understand what could be the reasons behind children not seroconverting. Thank you.
I think it's similar to what we just discussed. We're all different, and so everything like even when you do a trial, it's like a probability of things, right? So you can do a study and you say, okay, I give one dose at this concentration, most people will develop a response, but there could be many factors that some people may have a response, but is not maybe quite high enough. And so you want to give another dose to just stimulate the immune system a bit more. I think except if there are diseases, so there are like hereditary immune deficiencies, etc. That's obviously a different story, but we're all a bit different. And so when we select a dose, a regimen, etc., it's based on the majority of people developing the response that's desired. It doesn't mean everybody, but that's why then you do these studies and you say, okay, if I give one dose, maybe 80% of the people, of the babies seroconvert. If I give two doses, maybe I'm 95%. And so that justifies maybe the second dose. These are all considerations that when a vaccination schedule is designed, goes into that.
Thank you so much, Prof. Nemes. I think a resounding acknowledgement for your accessible, easy to understand presentation. Thank you so, so much. Our next session, before we dive straight in, we're handing over to Professor Michael Gold, where, there he is, sorry. For an introduction to adverse effects following immunization. But Prof, before we do that, we would just like to acknowledge you. I know you were in flight on Sunday evening as we were celebrating our long-standing faculty contributors to the annual African Vaccinology course. So in recognition and appreciation of your dedication and exceptional contribution to the AAVC course, but also to vaccinology education in Africa, we'd like to give you this long-standing faculty award, and I'll hand over to Greg to just.
So Mike, this is from VACFA to you in recognition of the amazing work you've done with us over many, many years. And Mike is currently based in Australia at the University of Adelaide. He's a global expert on vaccine safety. And in fact, the two of us go back together probably about 50 decades. We both worked together at Red Cross Hospital.
Said you were 100.
So Mike, this is just a small gift of appreciation. You've been around for almost 20 years. But thanks very much. Okay.
Look, the privilege is mine, really. And if it wasn't for Greg's invitation all those years ago, I wouldn't be here. So it really feels like coming back to the family. And thank you very much for this acknowledgement. It's much appreciated. Well, look, we're going to start off with a woo clap. I asked Salim at 10 minutes ago to put this together. So he amazingly he did it. So let's give it a go. Okay, let me know when you've got your code happening. Now, we don't have any prizes. That's the only downside to this. But it's going to give us a good indication of where everyone's knowledge is up to. Okay. Is it working? Can we? Okay, great. I can see we got 48 logged on. Okay, Salim, do I need to do anything to get it happening? Okay, great. Okay, which was the first immunizing agent that was developed because of concerns about vaccine safety? So you've got 4 choices there. And I'm going to lead into a bit of a historical clarification about this. And just really illustrate that the history of vaccine safety really goes back to the history of vaccination. Okay, I'm going to, Salim, can you click on correct? Okay, excellent. So the majority got the correct answer, which was cowpox, and I'll explain that to you shortly. Most common adverse vaccine reactions are detected by pre-licensure clinical trials. True or false? We're getting a 2/3 split. I'm putting the emphasis on common. Okay, let's go for the answer. The answer is true, and the emphasis there is on common and reactogenicity, and I'll be explaining that shortly. At the time of licensure, a new vaccine, at the time of licensure of a new vaccine by the national regulator, the vaccine is known to be completely safe. This is a really important concept that I'll be explaining to you. We're getting almost a 50-50 split. It's very good for me to see that there's a lot of incorrect answers in the room, because obviously that would validate the talk. Okay, let's go for the answer. Okay. All right. Vaccine safety is not signed off at the time of licensure, and I'll be explaining why shortly. Post-licensure surveillance only detects adverse vaccine reactions, true or false? So a lot of understanding the basic concepts about vaccine safety is in the terminology and the language. And I realize obviously that for many of you, English is may not be the first language, so it makes it even more difficult to really understand the terms and how to use them. Okay, let's go for the answer. So that's correct. The majority got that. Which of the following definitions of an adverse event following immunization is correct? So this is going to be a key principle to explain to you. So just to get a good sense of how we define this very important term. Okay, so again, I've got a 2/3 split. Let's go for the correct one. Okay, so I'm going to explain the difference between an adverse event and an adverse reaction. So in an adverse event, the definition of that is there's not directly a causal relationship between the event and the outcome, and I'll explain that shortly. Passive surveillance always establishes a causal association between a vaccine and an event. True or false? Okay, that's a lot more clear cut. Let's click on the correct. Excellent. So majority got that as being false. A vaccine reaction is the same as an immunization reaction. Okay, let's go for the correct answer, which is false, which is the what the majority of you responded to. Okay, in the interest of time, I'll come back to this question at the end of the talk. Okay, in brief, if the quiz has confused you, that's excellent, because there's a lot of clarification that we can now get onto. So let's get on with the talk, which is largely going to be explaining the basic concepts. Excellent. So here okay, ready to go, Salim? Okay, great. All right. I just wanted to cover this briefly. So the history of vaccination really is one of improving vaccine safety. Let me explain to you why. Greg did present a very nice historical overview yesterday, but what's very important is to differentiate variolation from vaccination. So variolation is really taking smallpox lesions and really scarifying them into someone in order to protect you against smallpox. This is what originated really centuries ago. And that when you describe that importation to America, it was really the importation of variolation. But the issue about this, because you were actually scarifying someone with the actual smallpox itself, they had a very high mortality, 1 in 30. It was a process that was observed by the wife of the Turkish ambassador to the UK, and it was imported into the UK in about 1720 by Lady Mary Wortley. So vaccination really is using cowpox to protect you against smallpox. So you can see that really this is using a weakened form or attenuated form of the pathogen in order to induce immunity to cowpox, to smallpox, and that had a much lesser mortality, less than 1 in 100. It was first observed empirically, as Greg said, by an actual farmer, but the actual GP, as it were, that did the experiment was Edward Jenner. And in 1840, variolation was outlawed because that was the more dangerous procedure. And then cowpox was used to protect against smallpox. So I give this historical example to you as a way of really showing that really the origins of vaccine safety really go back to the origins of vaccination. I'll come back to this about COVID afterwards. All right. I think 3 of you in the room mentioned that you were involved in pharmacovigilance. Some of you in the room may have some involvement with adverse events and vaccine safety, but I think regardless of what you do in vaccinology, you're going to have a view of vaccine safety, but that view is going to be different. And that's why I like this parable about the blind and the elephant, is that really you may look at vaccine safety from completely different aspects. So my job really is to bring you all into the same page and really give you the absolute basics, the 101 of safety. And interesting, this parable has been around for over 2,000 years, and yet we still use it and it's still relevant. So I'm going to cover 3 things. The first is why concerns about vaccine safety are important. The second is a very simple thing, is how do you know a vaccine is safe? Someone may ask you this. It's very important that you have a concept of how we actually come to this information. And then lastly, how do you know that a vaccine caused a reaction? Vaccine safety really is important because of vaccine uptake. And we discussed yesterday a lot about vaccine uptake, about DTP1, 3, MCV1, 2, uptake in Africa, various countries. So it all comes down to vaccine uptake and coverage. This is a framework that you will probably see later in the week. And the point is that I want to really drive home to you that it's really perceptions, and they've got thinking and feeling there, rather than the actual evidence. And I'm sure this point will be discussed with you on Friday, but it's really perception of disease and perception of safety that's most important. And of course, thinking and feeling and perception of safety is only one factor that results in, final uptake of a vaccine. There's motivation, there are a whole lot of practical issues, but nevertheless, it's an important component. The point I want to make here really is that when we ask parents or the community really whether they are more concerned about vaccine efficacy, as you can see here, only a small proportion in the red, probably less than 5%, are concerned about whether vaccines work or not. but the majority, over 50%, are more concerned about, in fact, whether vaccines are safe. And if you bring this into the context of Africa, and this was a study done here in South Africa, that shows similar results. So I don't think that the vaccine is effective, 7.8%, but concerns about side effects, 26%. And if you break that down more, that only a small number of these individuals are really concerned about vaccine safety in general, and really concerned about conspiracies. So concerns about vaccine safety rather than efficacy affect uptake. And this is a cartoon that you can see on the left here, a very famous one that depicts these cowpox lesions in individuals who were vaccinated with the cowpox vaccine, and dates from soon after Jenner did that experiment in 1796. So again, coming to the point that really that concerns about vaccine safety go back to the origins of vaccination. Why is this all important? Because it leads to reduced uptake, reduced coverage, and many examples of breakthrough in terms of vaccine preventable diseases. So best documented in terms of pertussis and concerns about the wholesale pertussis vaccine, going all the way through to COVID. And this is an important graphic that really illustrates a few points. And the message that I want to get across here is that when you've got high prevalence of disease, as you increase your vaccine coverage, you'll get to a point where there's low prevalence of disease, and yet the rates of an adverse event, as you can see in the orange line here, may be at least equivalent to the disease, or you may get more adverse events reported than actual disease. So to an extent, concerns about vaccine safety are really due to the success of vaccination in itself. And Greg showed you that slide yesterday from the CDC that showed the percentage drop in terms of vaccine preventable disease. So you can see that if you follow the cycle, it's all very predictable in terms of then loss of confidence, an outbreak of disease, and we're seeing this with measles at the moment, and then hopefully a confidence again in vaccination and reduced rates of disease. And obviously eradication is a point where you can stop vaccination and you don't have disease. So safety concerns can be about newly introduced vaccines. And we've spoken a lot about the HPV vaccine. But just reflecting on this yesterday, and I come back to Jason's talk really, he differentiated between a novel vaccine and an underutilized vaccine. And when I think about it, it's probably best to take an HPV vaccine and not refer to it as a novel vaccine anymore, because it's been around for a decade at least, and we should really look at this as an underutilized vaccine. In other words, we have adequate safety data, and we should differentiate that from a truly novel vaccine. You know, our best example, obviously, were COVID vaccines, new platform, etc. But you can see that these were the headlines in Australia when the HPV vaccine was used. And I think that's quite relevant, that, you know, our girls are not guinea pigs. And I need to explain to you in the context of vaccine safety why that's such almost an accurate headline, and we need to understand that. But many countries, this is a graph from Denmark showing the drop in HPV coverage due to vaccine concerns. And obviously, you know, to me, COVID was a truly novel vaccine platform, and we could speak endlessly about introduction of that. So how is this relevant to low and middle income countries? This becomes very relevant because I think we need to focus on novel vaccines that are used in low and middle income countries with little or no post licensure experience surveillance data from high income countries. And I've given you a list of those, and many of those we have discussed yesterday. So this is particularly relevant. And at the moment, for example, the dengue vaccine, the Takeda vaccine, is being rolled out in Brazil and Argentina and South America. And there's a safety signal that's been picked up there, anaphylaxis has been picked up. So a classic example of a novel vaccine introduced into a low and middle income setting with a safety signal generated. Importantly, we can get safety concerns about established vaccines. So I don't think any vaccine on the schedule that you currently use is exempt from these safety concerns. And obviously the classic historical example is measles and autism. And this relates to Andrew Wakefield, and that's a very good book if anyone wants to read the history of that whole event. Polio we've had mentioned a few times, and essentially now, I think sadly people are beginning to question whether the end game in terms of polio is ever going to be feasible. Safety concerns in low and middle income countries I think are also important where they're real or perceived safety concerns that have been identified in a high income setting. And we need to ask the question of how this affects low and middle income countries. I mean, the most obvious example and the most relevant example was the COVID vaccine platform, the non-replicating viral vector platform, and the mRNA. And remember that the thrombotic lesions with the viral vector and the myocarditis were picked up very much within high-income countries. And many of those high-income countries, Australia being one of them, started to restrict the use of those vaccines. And yet, for the majority of low-income countries, low and middle-income countries, they were using viral vector vaccines, you know, coming out of the Serum Institute. So how does that, you know, play out when you've got restrictions placed on a vaccine because of safety in a high-income country, and yet still being used in low and middle-income countries? So that's the first issue in terms of this. The second is the change in the landscape that's really changing month by month. And of course, all of you will recognize RFK on the graphic here. And I just bring this up because I wonder how much that is impacting in low and middle income countries, because it is his intent, I'm sure, to review all of the vaccines on the current US schedule. And how's this been playing out so far? So let's take as an example, as a case example, their review of the neonatal hepatitis B vaccine, for example. And their really proposed recommendations about not having this as a universal vaccine, as ensuring, as basically recommending that there should be hepatitis B surface antigen screening in pregnancy. So the way this is going in America, as I read it, and obviously we can discuss it at length, is it's really very much about the individual and not about the population. And how is that going to translate into the low and middle income settings? Influenza vaccine. They've adopted a recommendation not to use the multi-dose influenza vials because of thimerosal, the preservative in it. Now, we even had yesterday a mention about multi-dose vials and the effect of this on cold chain and storage. How is this impacting in a low and middle income setting when thimerosal is regarded as being a dangerous preservative in the US setting, and yet vital to using multi-dose vials in low and middle income settings? And so we can go, you know, on and on about these issues. But also relevant to low and middle income countries are the fact that there may be specific vaccine risks in specific populations. And that is a genetic risk. And I just mentioned this in passing. We don't know much about the genetics of adverse vaccine reactions, but there was an immune defect described within the Pacific Islander population that really resulted in these rare individuals or these rare children developing disseminated vaccine measles illness. So we've got to think of our own populations and what genetic risks there may be for them. So how do we know anything is safe? So I've given you an overview of why we should be concerned about vaccine safety, while the concerns are more than for efficacy usually. But if I asked you, how do you know anything is safe? How would you answer me? Is there a microphone? Let's just take a couple of comments. Okay, I'll take two or three comments.
It's got their hand up. Thanks. By observation over time.
Yes. Right,
Observation.
right, observation over time. I've got that from that. We've got a comment here on the front.
If it's have a minimal side effect or lack of harmful effect, if I get more benefit from the vaccine.
Okay, I'm going to
I
take
didn't-
from yours lack of side effects. All right, data evidence of what? Okay, what if I told you safety cannot be proven? It's really inferred. It's inferred by what? Only by? Okay, not by using it. So
Yeah, Latif.
You said, how do you determine if anything is safe, right? Yeah, anything like a car or anything like that. Yeah, so only by testing, using it.
okay, by using it. All right. Okay, look, I'm going to give you a personal example. I left to fly to South Africa at 6 o'clock on Sunday morning. Okay. I went for a bicycle ride at 10 o'clock on Saturday, and I tested out whether bicycles were safe or not. Okay. I can tell you going into the ground at about 40 kilometers an hour is not a very pleasant experience. Okay. The point is safety is inferred, and what we measure are the adverse reactions to whatever, to bicycle travel, car travel, airplane travel. It's really the adverse events that we're measuring. So safety is an inference by looking for adverse events. Okay. Well, I'm going to be specific here. I'm going to talk about reactions. So safety is inferred because of an absence of adverse reactions. Now, if we put this in the vaccine context, our reactions are adverse effects that are caused or precipitated by the vaccine, which is what is in the vaccine, or the process of immunization. So that's an important concept. So you've got two concepts here. The one is that safety is inferred by the absence of adverse reactions. Okay. And the second is when you put that into a vaccine context, we've got to differentiate between the actual vaccine, what's actually in that vial, and then secondly, the process of immunization. Okay. Now, the problem is this, that many of the symptoms and signs of adverse vaccine or immunization reactions also occur in the absence of vaccination. So these events unrelated to vaccination are called background events. So if we take febrile seizures, for example, febrile seizures are occurring in the community as a background rate. If we then vaccinate the whole population, of course, there will be children that will have febrile seizures. Some will be vaccine proximate and some won't. So surveillance is a detection of adverse events, but we need to differentiate an adverse reaction from a coincidental event. Okay, there are three unique adverse vaccine reactions that only occur with vaccination and no background rate. So I've given you an exception. So I've said this is generally what happens. You have a vaccine and something happens, but generally all of those things that happen, signs and symptoms occur as background events and can be coincidental, except for three very specific events. All right, we won't have time to ask you, but anyway, this is what they are. So if you think of all the vaccines we use and all the adverse effects that can happen, there are only three categories are completely unique to a vaccine. And these are obviously rare. So in terms of yellow fever vaccine, you can get this viscotropic or neurotropic disease. You can get attenuated live or bacterial disseminated disease. with BCG or measles vaccine in someone who's immunocompromised, or vaccine-associated paralytic polio. And then COVID really delivered one, and this was these thrombotic, thrombocytopenic episodes associated with the viral vector vaccine. All right, so how do we know a vaccine is safe? It's inferred because the absence of adverse reactions. Reactions can be due to the vaccine or the process of immunization. And safety surveillance needs to detect and differentiate reactions from coincidental events. So how do we detect these reactions, adverse reactions? The first thing to say is that dating back to the 1950s, there was no regulatory system for vaccine or drug, okay? So in order for us to detect all these reactions, we need a system, a regulatory system. The Qatar incident occurred when the polio vaccine, which was originally used, wasn't attenuated properly. And this resulted in 200,000 children getting vaccine-derived polio, 200 paralyzed, 10 deaths. In terms of drugs, thalidomide, again in the 1950s. So this is when any drug or vaccine could be used without any regulatory system whatsoever. So what I'm leading into, obviously, is the national regulatory system that each and every country has in order to regulate drugs and vaccines. And it's a complex system, but essentially what we're going to focus on is the pharmacovigilance aspect of that. So let's talk about how that's regulated. So the first thing is the pre-marketing phase and vaccine safety. Now, the important thing here is that we all know about phase 1, 2, and 3 clinical trials, but because it's a numbers game, in other words, if you have a very rare adverse reaction, you're not going to vaccinate enough people in a phase 3 trial to detect reactions which are rare or delayed or unexpected, or occur in subpopulations or with vaccine combinations. So this is where it leads to the very clear explanation that I gave, is that at the time of vaccine licensure, we do not know that a vaccine is 100% safe. And if you just think back to COVID and the vaccines used then, that was a perfect example of that. And for example, we should have been communicating to people that we knew the vaccine was 100% safe because there was a good prediction that it would not be. All right. So at the time of vaccine licensure, vaccine safety information is incomplete. So we need to talk about vaccine pharmacovigilance. So vaccine pharmacovigilance is the science and activities relating to the detection and communication about an adverse event following immunization. And we need to then now go into the definition of what an adverse event following immunization is. I'll just skip this in the interest of time. All right. So an adverse event following immunization, and this was one of the quiz questions, really is an untoward medical occurrence which follows immunization and does not necessarily have a causal link to the vaccine. And the reason for this is very, very important, probably the most important thing that I'm going to say in the entire talk, that you understand this concept. Because if I said to you that at the time of licensure, the safety information is incomplete, we don't want people reporting what is known. We really want to know what is the unknown. So with the COVID, for example, with a viral vector vaccine, at the time of licensure, no one would have known that that could cause thrombocytopenia and thrombosis and cause these terrible clots. The same for myocarditis and the mRNA. So if you have a vaccine and something untoward happens, that's what needs to be reported. It's not a causal association that needs to be reported. And essentially anything can be reported, from a symptom, sign, land finding, or disease. How's post-marketing surveillance undertaken? Largely by passive surveillance, which is the reporting, the spontaneous reporting of adverse events. But there are other ways to do surveillance which we can discuss, such as active surveillance or looking at individual case reports. So what happens to an adverse event report once it's submitted? It needs to go through the cycle of being analyzed. Why do we do post-marketing surveillance? the first important reason to do post-marketing surveillance is really to monitor the rates of the known adverse vaccine reactions and compare these to the observed with the expected rates. So in other words, if we have a background rate and we introduce a vaccine and we measure the same outcome, the difference between the two will be what's attributed to the vaccination. So what are the adverse vaccine reactions? So there can be many sources for this. Greg has already directed you to the WHO site, and this really contains vaccine and vaccine adverse reaction rates. The second reason is to really look for these unknown. So these are unknown at the time of licensure, and a safety signal will be generated, and this will be a cluster of serious events that will require further investigation. And here is a number of these. It goes back to rotavirus. We've already spoken about rotavirus and interception with the original versions of rotavirus. We've already spoken about myocarditis, and we've already spoken about TTS. Program errors or immunizations are critical. And these essentially relate to vaccine vial contamination with infection, as you can see on the right-hand side of your screen, or using the wrong diluent, usually in the measles vaccine, and the very dangerous things that can be mistaken for diluent, and there are many examples of these, would be insulin or a muscle relaxant, and that is a lethal combination. So these are the most important immunization errors to pick up. So how do we differentiate an adverse reaction from a coincidental event? So adverse event, as I said to you, is the surveillance term that we use. There's not a causal association. So we need to differentiate an adverse reaction from a coincidental event. And the process of this is called causality assessment. So causality is a relationship between the two events, the cause and the effect, where the second event is the consequence of the first. And the causality assessment process is determining if such a relationship exists. Now, there's quite a complex process that the WHO has advised that you do for individual case reports. And for those of you that want to come to the session tomorrow, we'll go into that in more detail. So really, I'm going to really just discuss this in broad terms. But if you want more detail about this, come to the session tomorrow, and then we'll explain the process in greater detail. So the last question to answer, how do we know that a vaccine has caused a reaction? So this we can look at two levels. We can look at it at a population level. In other words, does vaccination increase the risk of the occurrence of the event in the community? And then the second is in the individual, did it? So was the vaccine a factor in this individual in developing the event? So in terms of the population, causality assessment or passive surveillance does not establish causality. And that was one of the questions in your quiz. And the reason for this is we're only collecting data on those who had the event and who were vaccinated. So if we look at a 4x4 table, very, very simple statistical concept, you're only really populating one of those cells. Of course, there are those 3 exceptions that are very rare, but those are unique events. So even if they occur, we can say they're due to the vaccine. So how can we establish causality? Well, we need background rates to establish causality, so we can at least populate 2 of the cells. So if we know what a background rate is, for example, of febrile seizures, and we introduce a vaccine and the febrile seizure rate goes up, then the difference is going to be the vaccine attributable rate. So if we know background rates of certain conditions, we can determine causality through that. But the last way is to do really more sophisticated epidemiological studies, such as randomized controlled trials, observational case controls studies. In other words, to try and populate those 4 cells in order to determine causality. Now, in an individual, did it? This is the WHO causality assessment schema. And as I said to you, this is what we'll cover tomorrow. So I'm really going to gloss over this process because we'll cover that tomorrow. What I wanted to really outline for you is that what is the end result of that causality assessment process? So the end result of that causality assessment process is to really take that adverse event or that case report and put it into one of these 5 groups. Okay. And it's just important for you to understand the concept of these groups. I'll give a talk tomorrow that'll go into more detail about each of these groups, but I'll just cover them briefly. So the first one, what we call A1, is a vaccine product related reaction. So this is an adverse event which is due to what's actually in that vaccine vial. So you heard Prof. Nem speak about the antigen in the vaccine, the excipient in the vaccine, the adjuvant in the vaccine. So whatever's in that liquid, and when you inject that, induces an immune response, let's say it causes anaphylaxis, then that's going to be an A1 product related reaction. Number 2 is a quality defect. So this is when the vaccine is not manufactured properly in the factory. This doesn't occur often, but there are definite examples of these occurrences through the history of time. Immunization error-related reaction, that is what I alluded to in terms of vaccine administration, mixing up the diluent, giving the wrong vaccine, giving it by the wrong route. So it's the old-fashioned term was program error. The next one, immunization stress-related responses or anxiety-related responses. These are very important events, most frequently described as mass events, previously known as mass psychogenic events. In the old days, they would have said hysteria, but it requires a lot more explanation. Come tomorrow after 5 and you'll find out more about those. And then lastly, the coincidental. So coincidental clearly, not any of the previous ones, is due to a background condition, is not due to the vaccine. So lastly, I'm just finishing up on this as a bit of a case example if you want. So all of these are injection site reactions, but all of them have different causes. So this, for example, is a 4-month-old baby that had developed this reaction after 6 hours, an adjuvanted vaccine. You've already been explained in the previous talk about that inflammatory response and how if you inject an adjuvanted vaccine very superficially into the skin, you'll get an extreme local reaction. This is an 8-year-old boy, had a BCG. We know if the BCG vaccine is injected subcutaneously and not intradermally, you can get a BCG adenitis, so this would be an immunization error. We do know that this is a 3-month-old, again, had BCG adenitis. Again, there were a cluster of these. We know if the BCG organism is too virulent in that particular batch of vaccine, it can give you BCG adenitis. This is extensive limb swelling due to the DTaP vaccine. Vaccine product, it's due to what's in the actual vaccine. This is a 5-year-old who had a DTP vaccine actually injected into her right knee, believe it or not, and then induced an arthritis. So again, an immunization error, obviously completely wrong spot. This shows a contact dermatitis, coincidental because the vaccines were fine, but this child was allergic to the antiseptic that was put onto the injection site afterwards. And this is a 12-year-old had an HPV vaccine and developed this syndrome called complex regional pain syndrome that gives pain and swelling of the limb if you don't move it properly. And again, that's coincidental. So in summary, concerns about vaccine safety affect vaccine coverage. Safety is inferred because of the absence of a vaccine or immunization reaction. Pre and post-marketing surveillance of adverse events following immunization is the basis of pharmacovigilance. And causality assessment at a population and an individual level is performed to differentiate an adverse vaccine reaction from a coincidental event. And this is the last slide. For those of you who haven't seen it, it's another classic slide, and it's demonstrating probably the immunization stress-related response. And you can see the closer you are to the needle, the more stressful it is. You just follow the expressions going backwards. Thanks.
Thank you so much, Prof. Gold. Fantastic. I'm sure there will be lots of questions. Does anyone have any before we break for tea? Oh, yes, sorry.
Hi, thank you for the presentation. So, in calculating the background rate or knowing the background rate, how do you factor in variation, like seasonal variation or other confounding factors that may affect that?
Yep. Sure. Yeah, look, that's a very good question. And background rates are so easy to say, just measure the background rates. But it's in practice, it's confounded by all of those things. Let's take background rates of febrile seizures in a country that is endemic for malaria. You're going to have to stratify by seasonality. So you're going to have to stratify by age. So all of those things. So that sort of bedevils the background rate calculations. I mean, it's in high income countries, this is done in fairly sophisticated ways by using data, big data. So looking at discharge codes, looking at emergency department presentations, etc. So you can do it with big data if you've got the resources. We tend to focus on serious events. So, you know, we wouldn't necessarily do febrile convulsions, but you may be interested in, for example, Guillain-Barré syndrome and the influenza vaccine. But yeah, you're completely right. It's influenced by many different things.
We have another question here.
Yeah, thank you so much. My question is, since most of AFI surveillance happens passively, and in our context, it may be expensive to conduct RCTs, is there reason to believe that they are largely underreported in the African context?
Yes, no, thanks for the question. So, passive reporting, regardless, is underreported. So, you know, people can guesstimate as to how much underreporting there is. You know, something where some would guess 90%, for example. In one of the in the slide that I didn't have time that I cut out, what are the lessons learned from COVID? One of the lessons that I had was that we can actually enhance passive reporting. So for all countries, when the effort was put in through COVID, whether that was a low-income country or high income, passive adverse event surveillance reporting was enhanced. So if you promote it, people will report. But that leads on to then, well, how do you then augment that? So you may augment it through active surveillance, or you may augment it through passive, you know, stimulated surveillance. Within COVID, they tried to, it was the first kind of instance where there was a global effort to try and set up active forms of surveillance in order to really counteract that underreporting of passive surveillance. So passive surveillance depends on a healthcare provider or a vaccinee or a parent or a family actually reporting. So consumer reporting, I think, is a good thing. And that certainly enhances the rates of reporting. But I know if I went to our institution and went to the emergency department, as we've done, and asked all the clinicians there what an adverse event is and what would you report, you'd get multiple different answers. Everyone would be confused.
Thank you, Prof. The online participants are all saying thank you for a great presentation. We do have one question from Laureen. She's saying she is concerned A child who had a reaction of or a reaction to measles vaccine at 12 months ended up developing rash and fever, but it all disappeared after 3 days and it was not reported. So she's asking whether this will affect her in the future.
Right.
Yeah.
Okay, so look, 10% of individuals who get a measles vaccine will get rash and fever, 6 to 10 days. So live attenuated vaccine, you'll get an attenuated measles vaccine type illness. It's of no consequence really. So so won't affect further vaccines. I don't know whether the question is, should that be reported? Look, we would say just report anything. If you're concerned about anything, whether it be minor or severe, report it. So because of the underreporting that was mentioned, we don't want to put any barriers about what's reported. So I don't know whether the intent of that question was to say that if you have something that is expected, and to me, that would be a side effect. So it occurs commonly and it's not severe or serious, then should that be reported? So I would say that we report everything. In terms of analysis, and if you go back to that slide with the cycle, what you do with those reports, that can, then select all the serious reports, for example. And serious within the adverse event and pharmacovigilance has a very specific meaning. So you remember there's a regulatory meaning to the word serious. So it's got if it's a death, if it's life-threatening, if there's a congenital abnormality, if there's community concern, then it's serious. So this is a well-defined pharmacovigilance term.
Thank you for the presentation. I was wondering, is there a reason why low-income countries can still accept a vaccine, like the example of the viral vector vaccines that high-income countries are concerned about? Is it because maybe the benefits outweighed the risk, or that the incidence was low? I mean, how does that assessment get made? Just because there's also, theories that low-income countries or Africans get the short end of the stick when it comes to inventions like this,
Yes, no, that was my point. So again, if you come back to COVID, I think that raised all of those points. And of course, people would have all those concerns. I mean, why don't you use the viral vector vaccines in a high-income country, and yet you're happy to manufacture and ship it and use it in a low and middle-income country? So that's critical. One of the other things was that we had low rates of reported thrombotic events and myocarditis from low and middle income countries. Now, was that an underreporting, or are there, as I alluded to in the talk, are there specific genetic or environmental factors that may actually promote the development of those conditions? So, these remain unknowns, unfortunately, after the whole experience. But in answer to your question, I think that I'll take as more as a comment, but basically to say that that's the problem, and that's what I was trying to emphasize. This divide between the safety data that's largely collected in high income and trying to translate that into the low and middle income setting.
So there's a comment from Cornelius online. He's just saying many of our vaccinators always give paracetamol, especially when children come for DPT3 in anticipation of fever. So he's just noting that this leads to a huge underreporting of adverse events. And then we have a question from Anthony. He's saying countries report AEFIs to WHO through VGFlow, but the feedback is not always provided. So he's asking, how does this?
Yes. Okay. So all adverse events eventually are meant to land up at the WHO collaborating center in Uppsala. Okay. That's the one sort of theme from that question. The other is that sadly, this reporting scheme in any country, and if your country is different, please let us know, has zero feedback. And that is a complete disincentive for people to report. So if you want to really improve reporting, I think you need to improve feedback, because you report something, it just goes into a big black universe and you never hear what happens to it. All I can say is don't expect feedback from WHO. That'll never come in terms of adverse event reporting. But I think this really needs to be a very local thing. So it needs to be within a country, but even probably more importantly, within a region or district in the country. And especially if that adverse event is serious, and especially if that leads to death, for example, then the process that we'll explain further on Wednesday is clearly there needs to be a case investigation and a causality assessment. And that's the bit that needs to feed back to the family, to the vaccinee, to the vaccine provider.
Fantastic. Thank you, Prof. You've got time for one more, and then we're breaking for tea. So can I ask that in tea time, maybe you go and find Prof to ask questions. Peter?
Thank you, Prof. That's a brilliant presentation. AFI tends to cause some issues when it comes to uptake of the vaccine because of the concerns of the recipients. How do we get to respond to these safety concerns when we want the uptake to go up, especially for HPV, for example?
Right. Sorry, can I just clarify? So you've had an adverse event?
Yes. Whether perceived or real, but they tend to affect the way the vaccines get to be received, or the uptake of the vaccine in general for the rest of the population.
Okay. So what you're saying is that you've had an adverse event. Now remember what I said to you, an adverse event is not the same as a reaction. And if we again come back to COVID, we should have been overwhelmed that people were reporting lots of adverse events, because we want adverse events to be reported, because that's how we picked up the reactions. So it's when you mistake an adverse event as an adverse reaction that leads to problems. So what you're saying is that lots of adverse events, or a particular adverse events, reported, and then that has an effect, obviously, on perceived safety of that vaccine. So is that correct? Yeah. So so look, it's obviously complicated. A lot of it relates to communication. A lot of it will relate to what may be discussed on Friday. Discussing this in 1 or 2 sessions is just scratching the surface. So, there's a lot of good materials that's developed by WHO. What you need to do is look at the context. If you're introducing a new vaccine into your country, we heard about HPV in Tunisia. Even prior to that introduction, there should have been a whole communication plan around reported adverse events and how it was going to be responded to. The biggest problem is that when you introduce an underutilized or new vaccine, you get lots of adverse events reporting. You may or may not know whether they are reactions or not. It's going to take further investigation. So the point I'm trying to make is a lot of the communication is without certainty, without knowing. So it's very difficult communication. But as I say, look, people have devoted a lot of their time to trying to sort the communication issue out, but it's all around communication. But for your own kind of education, a very important point to remember today is an adverse event is not the same as an adverse reaction. And you know the definition of an adverse event as opposed to reaction, because that's commonly misperceived. Okay? Thanks.
Thank you so much, Prof. Both Prof. Gold, Mike Gold, and Elisa Nemes. Thank you very, very much. We're going to break for tea. I think from this session, I encourage you all to think about how you're going to package and communicate complex things that you learned probably for the second time this morning. I encourage you to think about it. These are good discussions to have. So thank you to the online participants as well for sending through questions. Can I ask that you are in your seats by 10:30? after tea. So we'll release you back here at 10:30, please, so we can stick to time. Thank you so much, everyone.
Hello, hello. Ben. Ben. Let's get everyone in. They should know. Welcome back to the second session of today. Iman, we need to sit down and do some work. Hi, thank you for coming back. The reason I'm taking a bit of time is that we're just having a slight change in how we're going to do the session next. And if people are outside, they will have no idea what is going on. And we would like them to have some idea of what's going on. So welcome back again. I hope the tea was good. This is going to be, there's a slight change. You will notice that there's Dudu's name appears on the program, and she was going to address this issue of leveraging the health systems in order to make a meaningful impact on immunization. Unfortunately, she's had a family bereavement, and as a result, she's had to drop out at very short notice. However, we've got a very good plan. You remember, apart from what Edina called herself, I think that was the Minister of Entertainment during the gala. What was the other description? Queen of Health Systems. Okay, so we are in very good hands. So what's going to happen, and Edina is going to come here with Bront, and they'll kind of explain how we're going to engage with the very same issue. The issue is that however you think about immunization and the impact you need to make, it needs to leverage on a functional system as a conduit for being able to do so. And that's what we're going to have an engagement in terms of reflection. And I know that you guys have been raring to talk. So we're going to use this time to engage with that before we continue with the rest of the program. But it's still covering the same issue. Instead of a didactic lecture, it's just going to be an engaged interaction in terms of addressing this issue. Edina, Brown, the space is yours.
Right. So thank you very much for that, Prof. Rudzani. So as Prof. Rudzani mentioned, given the change in the schedule and our deepest condolences to Dr. Duduzile, we thought this might also be a great opportunity, seeing as you had indicated in your feedback yesterday that you'd like to have more discussions. So we don't want to take too much time. I think Bronte is going to set the stage in terms of what we mean by health systems, what we mean by health systems strengthening. And then we'll open up the room, both online and in person, to discussions about what some of these concepts mean for your local contexts. So Bronte, I think you can get us started.
Fantastic. Thanks, Edina. I will just say Edina is still the health systems queen, but she taught me everything I know. So I won't be long. But it's just to give us a bit of background on what we mean when we're talking about health system, when we say the term health systems and health system strengthening. So our morning session was very focused on the immunology, and I would say like the very granular level, what's actually going on in our bodies. And I want you to put your very big thinking caps on it, because we're thinking not just out the box, but thinking really big picture. So we're going from super small to quite big, okay? But bear with us. I just want to cover exactly what it means when we talk about what the function of an actual health system is. And at its core, we need a health function to provide services, generate the human and physical resources that are needed to actually deliver those services. We need a health function to raise and pool those resources so we can actually pay for this healthcare that needs to be provided. And a very key one is stewardship. And I hope we're going to really dig deep into this today. And with that, we're talking about governance, setting direction through policies and regulation, and ensuring accountability for the health system. Okay, and with health system strengthening, which is really the core of what we want you to discuss and share with us today, refers to this process of identifying and implementing the changes in policy and practice within a country's health system so that the country can respond better. So I think the key word there is respond to the health system's challenges. And it really refers to a group of initiatives, strategies, mechanisms, or models that will improve one or more of those core functions that we've outlined here to lead to better health improvements of access, coverage, quality, and efficiency. Okay. And I'm hoping that this is somewhat familiar. It might be the first time some of you are seeing this, but on the left, we have the core building blocks. What we call building blocks of health systems. This is really sort of core health systems theory. And that refers to service delivery, our health workforce, information, medical products, vaccines, and technologies, very relevant to us, financing, and leadership in government. And through access, coverage, quality, and safety, what we're hoping to achieve, but that a health system will achieve, is obviously improved health, responsiveness, what I mentioned earlier, social and financial risk protection, and improved efficiency as a whole. Okay. So some health systems theory, I know we're flying through it, but I just want you to keep that in your mind as you discuss sort of the answers to the questions that we're going to be prompting you with today. So what we know, a bit of background to our questions for today is, and it's all been mentioned, I think most of yesterday, we touched on it today. What are the current challenges, very big picture, broad challenges in Africa that we're facing for immunization programs at the moment? Basic infrastructure deficits, lack of reliable cold chain, storage, distribution, or actual facilities, remote, hard to reach areas, limited access either in rural areas, underserved populations, or very pertinent in conflict-affected regions. And then a big one we've touched on, vaccine hesitancy, and this links more to the softer stuff, so sociocultural beliefs, disinformation, that obviously lead to the community being reluctant to vaccinate, and then any supply chain inefficiencies, okay, which we know quite well. And I'm not going to spend too much time here because I'd like to hear from you more. But when we talk about health system strengthening for immunization policy, which is really the foundation of what we want to be discussing today, what am I kind of talking about? So some things we can discuss is how financing should focus on health system strengthening beyond Gavi support. And I know we're hearing from Gavi in our next session. Countries being encouraged to take over operational costs as they exit from Gavi support. Investments, you can see a big theme on funding and money here, but investments should prioritize populations like our zero-dose children, under-immunized children, missed communities. Interventions should align with national plans and they need to be adaptable to local contexts. I think we all know that very, very well. All right, what works somewhere might not work somewhere else. Interventions need to address specific barriers in those zero-dose or hard-to-reach children and under-immunized communities. And with HSS, I'm just referring to health system strengthening. Those funded activities should prepare programs for future vaccine introductions. And I think Dr. Tony hinted to that yesterday with our introduction and prioritization and how it can integrate with overall primary healthcare strategies. Are you kind of with me? Does it make sense? Okay. I know that was a lot of words. We love frameworks and we love, lots of words in health systems. So now it's over to you. We've got 6 discussion points or questions that we don't have to answer them all, but we're using them as a springboard to get you to please share your specific experiences. And I want you to practice putting your health systems hats on and thinking big picture. Think of your experiences with your immunization programs in whatever context you're in, and share, your best practices, what works, what doesn't, what are your main challenges, all right? So I think we'll maybe come back to question 1, but I'll look at question 2 and 3 for us to start, if that's okay, Dina. The first question being, how can decision-making for immunization be better embedded in health sector planning and budgeting cycles? Okay. And the second one is, what are our key policy and coordination barriers that are currently preventing equitable access to vaccines, with a particular focus in high-risk populations? Okay, so 2 questions there. One about how do we embed planning, health system strengthening within planning and budgeting cycles, and what are key policy and coordination issues? So again, thinking big picture. Is anyone keen to start? Kick us off?
I'm also willing to volunteer people,
Great,
if that's okay,
please.
Bronte.
Yes, yeah.
I'll just step.
As someone who knows virtually nothing about health systems, surely one suggestion could just be, data is always something that's useful. So if you're looking at surveillance systems, especially for, I think, number 3, in terms of the new tracking cycles, it goes back to sort of the AI questions that we had yesterday in terms of, I don't know what the stats are on how many people these days own a phone or a smartphone, but I think it's quite a lot. And if you could really, I think a key thing there would be making the interface and the platforms that you design really user-friendly. I found a lot with like locally designed websites and things that sometimes it's extremely difficult to use, and then people struggle to, myself included, actually make use of these platforms. So if you can really generate platforms that are simple, easy to use for people, that they can make surveillance much easier, I think that would be helpful.
Looking at question 3, I think politicians are not sold on vaccines in most LMICs. because health as a sector is not seen as something that is beneficial to them. So whenever a budget is made in a parliament, you see that the health sector always gets like the smallest size because it does not bring votes. It's at least it's perceived as if it does not bring votes. So that's why you find that, let's say, the judiciary side of government will always get the highest funding and the health sector will get the lowest one because they intervene in their interests. Yeah, but I think the messaging has to be better. We talked about yesterday, $1 that is spent, you get $55 in return because you're saving on, all these consumables, diagnostic tests, medicines, and all that. I think that messaging is not, all these things we're talking about in the end will come back to money and funding. So we're talking about budget cuts and all these. Most of the things that we are worried about are things that most governments can afford, to be honest. But we're used to receiving handouts, and because politicians also are used to other entities and funding bodies funding immunization, because that is health and we leave it to the govies and all of that. So I think part of the barriers is that politicians, if we still maintain no buy-in from politicians and the funding mechanism to immunization, we will always run into these problems. And when someone decides to cut the funding, we'll always be in trouble as if there are things that we cannot afford, but we can. Thank you.
All right. No, thank you so much for that, Latif. So I think one of the important actors you have raised now is, the individuals at the governance level and, their decision making and sometimes what they prioritize and what they under prioritize and how that can influence being able to deliver effective or efficient immunization programs in our health systems. So I just wanted to hand over to our colleague in terms of actors such as those in the policy space. And I'm not sure if there are any links you could make between those in the governance decision-making level and those, say, in the NITAG, for example, from your experience.
Good morning once again. With the experience from Botswana, what is currently happening, I'll say, is a barrier. It's lack of available data to influence decision making. I'm saying this in particular referring to the NITEC. As we facilitate them as the secretariat, we are unable to provide evidence that can influence. For now, we are talking about life course vaccination, not only childhood vaccination. So it's one topic that is on the table. We had a meeting with the NITEC last month. So we are in the process of developing the life course vaccination strategy, and it has a bit of gaps because of those issues. And that will also influence the government because Botswana is a self-financing government when it comes to immunization. So being able to convince the powers that be the importance of embedding the life course vaccination in the routine or in the childhood one is still a mammoth task that we are working on. Thank you.
Thank you so much. And I think it's a very important issue. I think we hear that a lot about lack of local data. Any thoughts on why? Because I wonder if it's lack of local data or that we aren't using the data we have or that we are not generating enough. Which is it?
I think it is a combination of both of them. Most African countries, Sudan is one of them, they have fragmented health system data, which the data will most likely is underreported, is delayed and like this, but also we don't use the whole data we have. because we had this experience when we were introducing the malaria vaccine. We needed to, it was very scarce in the start, and we needed to know where are we going to start the vaccine. We needed to know the data on the prevalence, the incidence of malaria, deaths of under 5, and all this, and we don't have this data regularly and updated. So we had to bring any possible data from everywhere inside the Ministry of Health and outside. And then we did if I know some other colleagues here know it, what is known as the subnational tailoring of subnational estimation of burden for malaria and other diseases. And then in this, you make it is basically just you make the maximum use of the data you have. Yeah, thank you.
Okay, thank you. I wanted to add some points on the third question, which are policy and coordination barriers, especially on high-risk population. So I wanted to mention the issue of cross-border communities or communities living around the border areas. So we were discussing yesterday how it's difficult to define the target population for highly mobile or pastoralist communities, which live usually around the border areas in my country's context. So these communities are really far away from the central area, so there's very weak health system with weak infrastructures. Another thing is it's hard to define these communities belonging to one side of the country. For example, if we take the one with Kenya, the Borana community lives both in Ethiopia and Kenya, and there's no clear line that separates these people. So sometimes they go to the other country, sometimes they come here. And there isn't a really coordinated mechanism between the countries to make sure that these communities, which basically are one and just separated by a line, and how to give access to them freely. So I think it's really important to integrate efforts within other countries as well, and how to reach these communities and how to align the efforts so that these barriers can be solved.
Should I go ahead? Okay. Thank you for that. Yes. Is it also on cross-border? Hello? It's on something else. Okay. That's okay.
Okay, I wanted to talk about how can decision making for immunization be embedded in the health sector planning and budgeting cycle. Generally, the decision makers in our countries, they always want to see what is their what is their gain about if they want to finance something, what is coming up. Sometimes they would think that it's better for them to buy a bed in the hospital, to provide drugs than to give to them. But I think what we need to show to them is when I vaccinate one child, what am I gaining? So going to health economics. So we really need to present them that aspect. What are we gaining when we are vaccinating a child? It is better for us not to have to buy beds where children will come and lie than to, I think this is a way we have to start thinking. Yes, how to present the benefit we have by not having children falling sick than to wait when they are sick and to buy beds. Something else I wanted to add concerning still the same thing. What we did, I want to take the example of Cameroon. What we did now is that we have looked for in the parliament, we have looked for immunization champions. Yes, who can help us to, because our budget, the state budget is voted by the parliament. So we have looked for immunization champions in the parliament so that when the Ministry of Health is coming to defend the budget of health, where the budget of immunization is, we'll be able to make the parliament to vote that budget. This is something we have used to be able to sustain since we're a Gavi country and we'll be graduating soon. So we need to increase state fund to be able to sustain our immunization program.
So thank you so much for that country experience. And it just helps us to think around what's happening in countries like Cameroon and others with similar settings. And I think the points you raised are they're very enlightening and they're very helpful. I'm wondering, I think two things. I'm always wondering about the actors. Whose job is it? So you were saying, we need to communicate to the stakeholders in governance, so they understand what the benefit is. So who does that communication? And then secondly, in terms of those vaccine champions in parliament, what exactly is their role? Are they members of, are they just MPs? Are they mayors? Are they, and how do we get them to become those vaccine champions?
Okay, for the first question, who does the work? We have the support in Cameroon is the support of our NITAC, like getting us the data to know how to present the gain we have vaccination. So in our NITAC, we have a health economist, we have some economists in the NITAC, and then we're working together to be able to present to the politicians what is the benefit in financing immunization or in adding more budget in the immunization program. So that's the way we have a NITAC that is providing on evidence base. Yes, it's NITAC. Yes, our NITAC is really supporting the country in that light. Now for the parliamentarian, we are working with in Cameroon is, I can say something like connection. We have been able to convince some of the parliamentarians. It's not mayor, it's parliamentarians that are represented. We have some, not ministers, yes, people that have been voted by their area that we have been able to which we have presented immunization and have accepted. So people who have been able to convince who are now like vaccine champions that are advocating for the budget of immunization to be increased. So we have been able to have some of them. I think there are 5 of them that are champions. And then every time when there is something concerning immunization, their role also is to convince their peers so that it should be accepted and voted. We are still at the beginning, but yes, we just started this year that we started with that, and then we'll see how it can go. Thank you.
I guess I just had a comment with regards to leadership and governance, and I think that's really important. And in terms of one, really, championing the immunization program, but then also related to the budget that's that's allocated to health. I feel like in South Africa, we probably have the data and we have the budget, but there's so much mismanagement and waste in terms of budget that actually money that could have been siphoned to projects that would really make an impact is being mismanaged, and tender scandals and billions of rands that's wasted. And I think that leads to poor governance and leadership, and really taking accountability, for how money is managed within a country that has so much resources. And I think it also shows our responsibility and accountability to hold our leaders accountable for those misactions, and call them out on it. And I think that's important. And then it will, I don't know exactly, other than, having this one voice and actually saying, but enough is enough. We see it, we know it's wrong, and we're going to hold you accountable. And you have to come out of that position. You can't hold a position if you've known to be a corrupt leader. And I think that's the only way. And I wish there was a magic, like.
No. Okay. Okay, thank you very much. I think mine will be between 2 and 3. I will first begin with the decision making. I think in most of our settings, in local settings in Africa, there is a disconnect between decision makers and the ones implementing immunization programs. In every country, we have API programs, that's my guess, independent. On a particular Ministry of Health, like my country, we have API programs. But who are our decision makers in terms of funding? Now, most often, the people working in API are the experts to come up with empirical evidences, the value for vaccination for vaccinating a child. But overall, this will be submitted to probably a minister or IPS, permanent secretary of a country, who are the ones most often have no much background on immunization. They don't have much evidence to present it to the finance unit. We have a minister of finance who allocate budgets on annual basis to all sectors. You need to convince them we need more money for this sector. And when it also comes to health, some people said we have curative component, preventive component. That's another debate. What goes into preventive? Vaccinating a child, what will be the benefit comparing to when a child has been admitted? You want to hire a surgeon, you want to buy a lot of sophisticated materials. What is the cost comparison? All right. So, but this needs to be done by someone who have an experience, who knows the import and the relevance of vaccinating a child. But most often, people who are defending this for us are people, probably a minister, who just a medical doctor, or could be something else, he knows very well clinical part. So they weren't able to defend well, because most of our countries, like Gambia, we have limited data, local data, to convince our funders. Sorry, the one that's allocated resources. And now we are in the era of evidence. If you ask me, you need $2,000, you need to justify why this is coming from. You can't say, oh, because in Ghana or in Sierra Leone, this is the empirical evidence we have. No, I can't believe in that. Tell us what is the reality in the Gambia so that we can see. They're also highly educated. They just need to see the evidence around them. All right? The other component is about policy and coordination. I think we are very good as Africa. We are very good in formulating policies, strategies, but how often do we evaluate this? We are always forging ahead with implementation. But how often do we have policy analysis, strategic analysis? This is where we know the sense of direction. Are we doing better? In my country, there are a lot of policies that are expired. They are never, because we entirely depend on funding. Either WHO, UNICEF to give a fund to validate our or updated our policies. So the policy will no longer valid. So we mostly operate in silo. We don't even know what we are even doing. All right. So I think these are just few contributions I wanted to make. Thank you.
So I think those were important points, and I think it's the reality in a lot of our countries. I think a lot of our countries have some of the best written policies, don't always translate when it comes to implementation. But you raised the point about the gap between the governance side and the implementers, which is a very real gap. But do we know of any kind of actors that serve as some kind of liaison between the implementers and the people that make the final decisions, those in government? I'm hearing whispers. Sorry? Are there actors that work between the implementers and the people that make the final decision? So the Ministry of Health and the implementers, because our colleague was raising the issue about the gap between the two. And
Yes, I can expand a little bit on that from an industry perspective. So we've been in touch with SAPRA quite a lot for the past year because we've just initiated a clinical study in South Africa. And I think maybe that should be part of the communication mechanism as industry. We sit with all the data. We have the vaccines, we know the players, and how we interact with government. And some of you may know, not BioVac's partly owned by the government. And so we have them on the board of directors, for instance. So how we communicate with them, I think we will see more and more of a role. Unfortunately, the biopharma industry in South Africa is very, very small. So, and I'm talking about South African environment now. So the influence is very small as well. How can we work together with government as a manufacturer, as a local manufacturer, not to only make decisions on immunization, but also where they procure the vaccines from? Why not from the African manufacturer? We see this as an Africa CDC goal. How do we get to that point? And I think industry is going to start playing a bigger role in this communication mechanism.
Thank you so much for that, Nicholas. I see some other hands.
Just on that point of have a good responsibility. That's why in the primary healthcare, we are talking about the area of responsibility. And when we add the private clinic, they don't have area of responsibility. They are in touch with individuals. And there we lose many people, many children. So in that way, it's a bit difficult to have what we are calling the equity interventions. So I think we need to increase or to improve this implementation part of policies. We need to look at the new rules which involve private sectors, even manufacturer, but mainly the health service, the private health services. Thank you.
Thank you very much for that, Tony. So what I'm hearing, Bronte, is the importance of public-private partnerships, which I think you could talk about for a day. All right, over here.
Thank you. I would like to piggyback on the last two comments to say that most of resources dedicated to vaccination or immunization programs in general are looking at the demand side and how can we respond to burning needs of populations in terms of vaccines. And yesterday session, although like it was rated kind of interesting, but not really, we don't know really how we can implement these ideas in terms of self-sufficiency. I would like to say that if we advocate sufficiently enough to Gavi, for example, not to focus only on the demand side and how to respond it in terms of external supply over and over of a global vaccine production and manufacturers putting more and more pressure on national capacity to respond to needs that we created, to focus also on the national regulation capacity, national manufacturing capacity, and African manufacturing capacity. It is sad to say that today in our African continent, like we are 50 plus nations, only two have the capacity, national regulatory authorities that have the sufficient maturity. They are South Africa and Egypt to produce nationally vaccine serums and immunoglobulins and to be able to export them. We have other nations in the pipeline trying to make it on their own. I'm citing Morocco, Tunisia, Ethiopia, and so on. Like they produce nationally products, but they are unable to export them. Although they are quite of good quality, they have like very competitive price, and nobody is there to support the national regulatory authority. And I'm thinking about, okay, we have the niche, which is like the NITAG support hub. What about national regulatory authority support hub? What about local manufacturers to make sure like private public sector, if we invest more in economic prospering of nations, maybe like we can push for African solutions to African continent and not relying over and over on the global market and the patenting issues. And like we have seen what happened with COVID, like we requested to patent waiver and ended up being too small, too narrow, too late on developing nations. And in the same time, we have increased demand because we created the demand to have Pfizer, to have Moderna coming over, to have vaccines that our economies cannot bear. So thank you.
Pertinent points, fantastic. Imen, did you want to add to that? I know you had something.
Yes, just one point on the what you like. Do you when you think immunization is part of the health system? I think as someone who's interested in the vaccine decision making, whether like in terms of policy or people, I think the health system is a key factor. It's like it's very important in the vaccine decision making, and this is because we cannot do what other countries do, because sometimes in a country the health system can handle a disease, can handle the few cases of severe forms that arise. But in our country, and this is why we need to have local data, it's very important, we cannot handle that. So we need to vaccinate. So if the US or the UK go and recommend not to vaccinate because their health system is ready and can handle cases or severe cases and the disease more like burden. In Africa, we cannot. So in terms of recommendation and vaccine decision-making, health system is key. Our health system in our country, in our city, where we live, it's important. And this is why we need local data. And I think we no longer need to rely on what other countries, we can learn from them, but we really needed to make it our own decision. Yeah, that's what I wanted to add.
Thank you. Thank you, Imen. I know we have a question here just to say what we're really hearing is this public-private partnership, but also multi-sectoral. So we know the health system doesn't function in a vacuum. You know, to get finance, governance, private sector is really important to get everyone on board. Where was the other question? Oh, online,
Yeah,
great.
so we have 2 comments online. So Tapioa, can you unmute and Tapioa? Okay. If Tapioa is struggling with her sound, maybe Abdul, you can go ahead.
Okay, thank you. Just a comment on the first point. I mean, I feel immunization works best when it strengthens the whole system that is underneath it. Come to think of it, when it is treated as a vertical program, I mean, we create parallel data systems, parallel supply chains, and that doesn't really work, especially for our clients. It works for quick wins, but it's actually not really sustainable. However, I feel when it becomes part of the health system, it forces us to look at the whole foundation. When you look deeply, the data quality, logistics, workforce, financing, etc., etc. And another issue when it comes to our climb has been mentioned earlier, which is the issue of data. But I just want to reiterate on that because data ownership is a very, very major weak point which affects the ultimate decisions of health systems. And in many of our clients, we end up relying mostly on partners for supported data sets because they have, of course, more updated and more robust data. For example, I can tell you in Nigeria, I am more confident when I get the data from CHAI or probably CIHP rather than our own routine reports or DHS data. And this is a signal that really that our primary health information systems need strengthening, not that we keep relying on partners. It's not really sustainable. Thank you.
Thank you for that comment, Abdul. We'll have one last one from online. Tapioa, are you ready to make a comment? If you're not, Tapioa, that's fine. Colette, can you give your comment? Okay, I'll just read out Colette's comment. She was saying human resources are a major challenge in terms of both quality and quantity in Guinea. She's mentioning that the current workforce consists mainly of volunteers or interns from the central level down to the community level. And that furthermore, given the management of previous epidemics, they were receiving really good incentives and that the life difficulties encountered, community workers are asking to be paid, if not the collaboration, I guess, from, you know, up there to the community health workers has been very difficult.
Thank you. In Somalia, rely on external donors. So we start something called triple P, private-public partnership. So also we're looking for domestic fund to look to fill the gap for the financing.
Thank you. Thanks, everyone. And we're going to stop there. But as you can see, I think we've just scratched the surface. Thank you for sharing many of your country experiences and all the challenges. I think really high level is that, you know, partnership is really important between public, private, cross borders. Not just thinking of your own health system, thinking of it in the context of the region, of the world. How can we strengthen data systems? How do we make sure that our health systems are resilient to shocks like COVID? How can they adapt and absorb those kinds of shocks and still function, particularly within immunization programs? Idina, do we have any final comments before we wrap up?
No final comments from my side. I think just to thank the team in person and online for their participation and the points that they raised. The idea, of course, wasn't to solve all health systems problems, but to really just make us acutely aware of the very real challenges that exist in the health system, and to prime us or to provoke us to start thinking about what is our role, where are we positioned, how can we act. We're very good about flagging challenges, but not so much about saying, this is where I'm standing, this is my position, and this is how I can contribute to enhancing things. So when we say we should, I hope it means that we are in fact going to be acting. So thank you to everyone online and in person, and back to you, Prof. Rizani.
Thank you very much. And I think it's just becoming very clear as you go that the whole immunization space is not just complicated, which is a different thing altogether, but actually complex. And it's this complexity that you'll have to deal with if you're gonna ultimately deliver on the promise that vaccines have for the children of this continent, especially in the context of your own country in which you are responsible. So thank you very much to our two VACFA colleagues for leading us through these discussions. The next speaker is Michelle. I think she's already online. Michelle Jimenez is Senior Manager involved in policy in Gavi. But if you know Michelle, she's got an extensive experience in the policy space and having worked with a number of really important partners in the immunization space, UNICEF, PAHO, WHO, and so forth. And but in the space in which she is at Gavi, she engages with all these stakeholders, including a meaningful link with the WHO Central, with SAGE, with respect to the work that needs to flow ultimately into making immunization possible. So the issue of sustainability has come up during this time, funding and so forth, all these kind of questions. And I'm sure you are anticipating and really expectant of what she has to speak about with respect to making vaccines available to the African continent. Michelle, welcome. And thank you for always being available and for being a really amazing partner and colleague in this work to make vaccines accessible for the African continent. Thank you very much. You can take it from here.
Thank you very much, Rizwani, for that introduction. I will try and live up to that. Good morning, everyone. Thank you very much for giving me the opportunity to be here. As Rizwani said, my name is Michelle Jimenez. I'm a senior manager in the policy team at Gavi, and I have the honor to give you today a bit of a presentation. It's really an introduction to Gavi and the work that we do. Give me a second as I make sure that the slides are online. The work that we do. So over the next 25 minutes or so, I'd like to go through Gavi's mission and its work, some of the impact we've achieved so far, some of the approaches we use to achieving access to vaccines in Africa and in other low and middle income countries, as well as our future agenda and priorities for the next 5 years. I hope that we will have ample time for questions, hearing some of the questions you had in the previous session. They're very difficult, and I'm not sure I will have answers to all of them, but we can certainly have a discussion, which I look forward to. So if we talk about Gavi's mission and work, the first thing to understand is that Gavi is an alliance. It's a multi-stakeholder, a public-private partnership. It was set up in the year 2000 by the founding partners, which are the World Health Organization, UNICEF, the Bill and Melinda Gates Foundation at the time, and the World Bank. But it has always brought together around the table stakeholders such as implementing country governments, donor country governments, civil society organizations, research and technical health institutes, vaccine manufacturers, and other private sector partners. Gavi's mission is centered on equity, and it aims to save lives and protect people's health by increasing access and sustainable use of vaccines. So far, it has supported a routine immunization in 78 countries, and currently in 2021 to 2025, eligibility is open to 54 countries, but there's also some work that is done in middle-income countries. Over the past 2 decades, after these 2 decades of progress, Gavi is now focused on protecting the next generation, above all, zero-dose children, and we'll discuss a bit more about that and how we aim to achieve it. Gavi provides vaccines for over half of the world's children. Since 2000, we have been able to vaccinate 1.2 billion children and provided over 2 billion vaccinations through preventive vaccination campaigns. This has averted over 20 million lives, and through this impact, we have built stronger economies, generating over $280 billion in economic benefits in the countries that have received support from Gavi. The Gavi model, which we will discuss shortly, aims to provide a sustainable future. For immunization programs, and 19 countries have transitioned out of Gavi support and now fully finance their immunization programs. So, focusing on this impact of 1.2 billion immunized children over the past 24 years, this has been incrementing gradually over each strategic period. Gavi works on 5-year strategic periods. We are in 2026, we will be launching what we call Gavi 6.0. And with each of the strategic period, the number of children reached has increased, the number of funds available to reach these children has also increased, as well as the number of vaccines we provide against different infectious diseases. So this impact can be shown in decrease in child mortality. So on the graph on your left-hand side, you see a decrease there's a 70% reduction in vaccine preventable child deaths. This is in the Gavi 57 countries, so the countries that received support from 2021 to 2025. This has led to a 50% reduction in under 5 mortality rate in these countries over a similar period of time. This basically means that children are more likely to reach their 5th birthday as part of this support. One of the other roles that Gavi has is around shaping vaccine markets. So by procuring the vaccines for over half of the world's children, Gavi is able to adjust the cost of the immunization, making vaccines more accessible. And this graph here shows the cost of immunizing a child with pentavalent, pneumococcal, and rotavirus vaccines, and how the cost of that has decreased from 2010 to 2022 by over 50%. As I mentioned before, the Gavi's vaccine portfolio has increased over time. And with each strategic period, we go through a process, which I will describe shortly, where we review the vaccine pipeline and evaluate which vaccines should be added to the portfolio to serve the country's best. The vaccines on this slide include vaccines for routine immunization, as well as stockpiles and vaccines to respond to outbreaks. In the last column on your right-hand side, you will see that Gavi 6.0, we have ambition to increase the vaccines available to up to 25. This is because we have already gone through a process of reviewing some vaccines in advance of an expected product. So how do we achieve access? This slide here summarizes the Gavi model, and I'd like to spend a bit of time going through this. So it's based on long-term funding and the idea of market shaping, a strong donor base, as well as co-financing from countries. So from the very beginning, countries procure a portion of their vaccines, and this increases over time. And I have another slide that describes that. So based on this long-term funding, Gavi is able to pool the demand of the lowest income country, pool the demand of vaccines from the lowest income countries, accelerating access to these vaccines, and also providing signals to manufacturers in terms of which vaccines would be the most useful for the populations that we serve. This ability to shape markets ensures that vaccine products are more affordable. In addition to the procurement of vaccines, Gavi also provides support for health system strengthening. This is in the form of cash support for countries, and the idea is that this support strengthens the vaccine delivery platforms. Finally, there's this sustaining immunization and transition, where little by little the co-financing increases, and the support around health systems also decreases as countries take over more and more of these costs. So Gavi supports countries through three main financing levers. The main one is vaccine support, where vaccines are procured for countries. Then there's this health system strengthening support, which I was mentioning in the previous slide. And then there's also technical assistance support, which is provided through core technical partners as well as civil society organizations. This slide here aims to describe Gavi's co-financing model and how countries transition through it. So for the lowest income countries, there's an initial self-financing phase. They provide a small portion of co-financing for the vaccines. As their GNI per capita increases, this portion of co-financing also increases. Once they go over the low-income country threshold, which is defined by the World Bank, this proportion of co-financing increases gradually. And once they meet the Gavi eligibility threshold, they are moved into accelerated transition, where the co-financing portion increases at a slightly higher rate, with the idea that eventually at the end of these transition phases, countries are able to fully self-finance their immunization programs. Countries use Gavi support to invest in health systems, and these are critical for immunization programs. Most of this goes towards service delivery, demand and community engagement, supply chain, health information systems, and monitoring and evaluation, pooled funding, and human resources for health, amongst other things. So one of the main challenges that we have is closing the gap in immunization, and one of the children born, for example, in 2024, 20 million of these children will be underimmunized. And 1 in 5 of these children are in Gavi-supported countries and will not receive the full course of basic vaccines. 72% of children are in 57 Gavi-supported countries. So in 2024, there were 10.2 million zero-dose children in Gavi-supported countries that did not receive a single dose of basic vaccines. The Democratic Republic of Congo and Nigeria account for 27% of zero-dose children in Gavi-supported countries. And there's a lot of support focused on ensuring that we reach these children. So continuing with the zero-dose agenda, we saw in 2024 a decrease in zero-dose children by 5%. You can see here on the graph from 2023 to 2024. However, there is still a long way to go to achieve the reduction in our 2025 target, and even further to go to achieve the immunization Agenda 2030 goal in 2030. We are working with countries to ensure monitoring and support to reach as many zero-dose children as possible. But as you can see, the challenge is big. Gavi is also focused on revitalizing the HPV vaccine program with the aim of leaving no girl behind. There was an ambitious target of reaching over 86 million girls by the end of 2025 and averting 1.4 million future cervical cancer deaths. In 2024, we reached over 32 million girls, which is more than in the previous 10 years combined. This has been possible because of the change to the one-dose schedule, which has improved supply, as well as Gavi support, both financial, political, social, and technical. Another important vaccine is the malaria vaccine. It is one of the as you all know, malaria is one of the world's leading killers of children aged under 5 years of age. Over 1,000 children die every day of malaria, and it took over 50 years to develop the vaccine. We now have 2 vaccines that are pre-qualified by WHO for use in children, and 23 African countries have introduced the malaria vaccine into their routine immunization programs with Gavi support since 2019, sorry. We expect that 80 to 100 million doses will be required by 2030 to continue reaching as many children as possible. This is something I mentioned in terms of how we decide which vaccines should be added to Gavi's portfolio. So in advance of each Gavi strategic period, we carry out a vaccine investment strategy, which is an evidence-based approach to identify immunization investments for future funding cycles, while sending valuable advance signals to vaccine developers and suppliers. We carry it out every 5 years. It's based on a transparent methodology with an analytical review of evidence and modeling and consultations with independent experts. It leads to strategic decision-making, so avoiding a first come, first served, and provides predictability for long-term planning by industry, governments, and donors. It will inform Gavi's strategy, its resource mobilization, activities, and can lead to different types of investments. So a routine vaccine program, for example, a global stockpile, as well as learning agendas, where we set aside a small amount of funds to carry out some implementation research that will inform a future program. If there is a public health threat or a R&D milestone for a priority pathogen for diseases of epidemic or pandemic potential, Gavi can also go through this process outside of this 5-year cycle to be able to respond in a timely manner. So in terms of our future agenda, as you all know, we live in challenging times, right? There are global trends that are impacting the progress in immunization. We have a deteriorating macroeconomic outlook. We see fragility in many countries as the new normal. Many countries, the world is facing a climate emergency, and we see big shifts in global aid policies. However, there's also some promising trends that can help us drive progress. These are the promise of new vaccines, country ownership, regionalization, and global collaboration. And we're seeing a focus on that, as well as innovation in immunization. So Gavi went through a process of developing its strategy, the Gavi 6.0, which would address our priorities from the year 2026 to 2030. This slide here summarizes that strategy, which maintains the vision of leaving no one behind and the mission of saving lives and protecting people's health by increasing equitable and sustainable use of vaccines. Some of the principles have been adjusted to reflect some of our changing priorities. So I'd like to highlight, for example, country-led and sustainability. Those principles were there before, but they take on a different meaning, in a more urgent meaning in Gavi 6.0. The first priority of zero dose and mixed communities is also maintained. And we've also adjusted our ability to provide differentiated support, particularly responding to fragile settings. We've also added other things like climate sensitive and integrated and adaptive. It is based on 4 strategic goals, including introduction and scale up of vaccines, strengthening health systems to increase equity and immunization, improving programmatic and financial sustainability of immunization programs, and ensuring healthy markets for vaccines and related products. And I can go through briefly through these different strategic goals, as well as some of the changes that we have had to introduce to the ambition to some of these goals, given the constrained financial environment that we are all facing. So in terms of introducing scale-up and vaccines, the programs already introduced in countries will continue. The stockpiles, existing stockpiles, will also continue, including a new stockpile for Mpox vaccines, which was approved by the board this past July. there will be scoping and pacing of new vaccine introductions and preventive campaigns. This is particularly around preventive cholera, typhoid, Japanese encephalitis, measles and measles rubella, as well as yellow fever. And we will also be introducing country vaccine budgets, previously known as vaccine envelopes. And this is a significant change to the model where countries will have indicative budget for their vaccine procurement that will be communicated at the beginning of the strategic period, and they will need to manage within that vaccine budget. In terms of strategic goal number 2, we have an approved health system strategy, which is around maintaining, strengthening immunization programs and reaching zero-dose children. This is the first time that Gavi has had a health system strategy. We also have a fragile and humanitarian approach, which aims to provide Gavi with flexibility to work in fragile settings and with humanitarian actors to respond to the particular challenges in these contexts. There is a new Gavi resilience mechanism, which will provide funding in these settings for unforeseen needs. However, there will be a reduced investment in both the health system strategy and the fragile and humanitarian approach, given some of the constrained funding environment. In terms of improving programmatic and financial sustainability of immunization programs, that co-financing model and transition model that I showed in one of the previous slides, we are maintaining the integrity of that model. There will be increases in country co-financing trajectories, including higher co-financing for IPV and malaria, as well as new co-financing requirements for preventive campaigns. The existing approach for middle-income countries has now been integrated into the Gavi 6.0 model as the catalytic phase and will continue to be part of what we do, but it will have a narrow focus now, focused on vaccine introductions and not including backsliding support. In terms of the fourth strategic goal around ensuring healthy markets for vaccines and related products, healthy vaccine markets are continue to be emphasized as critical to the Gavi 6.0 success, and we will be working to see where further market shaping gains can be achieved, even though there will be some difficult choices required to be able to achieve them. I think this is my last slide. Oh no, there is one more. This is very important as well in terms of Gavi 6.0, and it's around Gavi Leap. So it's really about focusing everything that we do to adjust to the global health ecosystem and provide countries with as much flexibility as possible. So it's based on 4 principles around country centricity, so giving countries more agency, putting country and community needs at the heart. A country sovereignty, so fully transitioning, providing support to fully transition out of donor support. Define mandates, so agencies operating in line with their comparative advantages, and ensuring that we have a finite lifespan, so agencies define a date to put themselves out of business. It has 3 objectives, so ensuring that Gavi's internal reforms place countries at the center, greater alignment with close partners at country level in line with the Lusaka agenda, and catalyzing change around the reform of global health architecture. And I think that is my last slide. So I'm happy to take any questions you might have. and I'll do my best to answer them. Thank you.
Thank you very much for that, Shell. We've got 5 minutes of quick questions before we go to lunch. It's a joke. It's not a very good one, but I'm trying. Okay. I think she's left quite plenty of time for people to ask questions. So we've got a few questions. I just want to see where the hands are. That's one here. Okay. And then we've got Nicholas here, and we've got another question there. Okay. Please go ahead.
Hello, thank you so much for this really instructive presentation. I have 3 questions, but I try to keep it short. My first question is when does Gavi decide to move towards a co-financing scheme? Does it happen by looking at each individual vaccine or moving the whole, based on a whole portfolio? And also, I was surprised to learn that a big part of the HSS grant is used for demand generation. So could you elaborate on how that's used? Is it for funding core communication strategies or just sporadic, risk communication activities? Thank you.
Please, Michelle, you can go ahead.
Oh, okay. Okay. I thought we were taking a few. Yes, of course. So in terms of Gavi's co-financing, it is for the whole portfolio. So in initial self-financing, when countries first receive support, they provide an amount for each vaccine, and this changes over time. Once countries move into the preparatory transition, this increases over time. We have introduced some changes that will make countries more price sensitive to some of the products. So yes, there will be some shifts around that, but it is focused on the whole portfolio. And in terms of demand generation, this really will vary according to country. Countries decide what it is what type of demand generation activities they will need. But there's a lot around when introducing a new vaccine, making sure that there's a communication plan around that, and that the communities are ready to receive that new introduction, as well as maintaining interest in the rest of the program.
Thank you for that. Next question.
Hi, Michelle. Thank you. I'm Nicholas from BioVac here in Cape Town. So with the changing environment and let me say the reduction in funding available from, for instance, the UK government, who's announced that they're reducing the amounts that they're giving to BioVac, to Gavi, as well as the United States drawing all money back, focusing more inwards. How are you planning to bridge that gap where the finance is really limited from this public-private partnership that you've had?
Please go ahead.
Yes, thank you for that question. So we went through a replenishment period, which culminated in June, and there is, as you say, a shortfall. So our board has met in July and did some recalibration and prioritization around that strategy that I presented, and some of the changes through each of those strategic goals already address what we will need to do to be able to address that shortfall. So there's definitely a view to protect as many programs as possible and continue those that are already in the portfolio. But there will be some pacing and changes to some of the ambition to those programs. There will be less cash support as well, for countries, and there will also be an expectation for increases in co-financing requirements. But yes, that is the world that we are facing.
Thank you, Michelle, for good update, and happy with the work your organization is doing to Africa. I have two few questions for you. The first one is I'm just curious, like, since you're an advisory type of one of your function is to advise, why don't we have single dose just for us to focus, or maybe to target some of the population, especially the mobile populations and population which are in smaller sizes, just to avoid waste on that particular point. And the second one is, can you just briefly update us on the you funded some of the African countries to start manufacturing their own vaccine, their own vaccines. So how is it? Can you just give us a nutshell type of update on that venture? Thank you.
Thanks, Michelle. You can respond to those two. I think she just took an advantage of having the mic and asking for two, but please go ahead.
Okay. So in terms of single dose and avoiding wastage, yes, there is, I mean, it is important that countries look at how they optimize their portfolios and decide which presentations make the most sense in their context and in different contexts within countries. There is some work, for example, that has been carried out to switch the measles presentation from 10 dose to a 5 dose, and how that in supporting countries to do that precisely to avoid wastage and encourage health workers to open the vials. So there is some work around that, and as I said, also countries will need to look at how to optimize their portfolios and make sure that cost and wastage is considered. We have a selection of different presentations that are available, so countries do have a level of choice in terms of the presentations that make the most sense for them. And then in terms of the African Vaccine Manufacturers Initiative, there is a commitment to continue with that. There is a set of priority vaccines that have been identified for regional manufacturing, so that that is continuing as planned. It was something that was set up in the as we all know, after the COVID-19 pandemic, and the commitment to that continues without any change.
Thank you very much for that, Michelle. There are two questions online.
So the first question is from Abdul. He's saying there's strong momentum for regional vaccine production at the moment. And he's asking, how is Gavi thinking about integrating African manufactured vaccines into its procurement and pricing frameworks without disrupting the affordability equation that pooled procurement currently provides?
That is a very good question. I think it will be we are revising our procurement policy to cover all these different options. I don't have a specific answer to that question right now, but we will be reviewing all of that over the coming year to ensure that we are able to absorb these new products.
Thank you. The second question is from Cornelius. He's saying Gavi is doing great in Cameroon to strengthen the health systems. So he's asking how is Gavi ensuring that when they exit the vaccination program, or when Gavi exits, the vaccination problem will not fall apart or go back to sleep?
Yes, we have introduced some some contingency for that, thinking around particularly the cash support. In the idea is that as countries transition through the Gavi model, the cash support also decreases. So little by little, governments will need to take on some of those costs. We have also introduced a threshold around operational costs. So some costs will be non-eligible for funding as countries move towards graduation, giving governments time to take on some of these costs in a more sustainable manner, and precisely to avoid an impact from an exit. So making sure that that doesn't happen abruptly and that countries have time to absorb some of these costs, because we all know that making fiscal space takes time. I can't hear the room anymore. Okay, sorry.
Thank you very much. That presentation was very informative. My question is around countries that are transitioning from a Gavi support, especially regarding policy. We have realized, I come from Kenya, and Kenya is one of those countries that is transitioning, that the funding that is supposed to be from the government takes some delay. I don't know what steps you take to encourage these countries to properly allocate the fund, and also probably ring-fence the fund that is allocated for the vaccines. Thank you.
Yes, this does take time. There is some support that is provided, particularly for countries like Kenya, in accelerated transition around health financing and working to ensure that fiscal space is created for vaccines. There's also some work that we support some civil society organizations to advocate parliaments, to advocate to governments to make more of this fiscal space. So there's a few different strategies that we have used for this. But of course, this all is also affected by politics and different things that are not necessarily always in control. But we do provide support on the health financing side and on this advocacy side to ensure that countries make this fiscal space and have time to do that.
I couldn't switch on the mic. And this is just before one of the course conveners is about to ask an amazing question. Hold on for it.
Michelle, don't worry. It's not an amazing question. It's Ben. How are you?
Good. How are you, Ben?
Very good, Michelle. Excellent talk. My question to you, as you can possibly guess, is on NITAGs. How important are they in the next investment strategy for Gavi? And given the lessons learned, what do you think we need to do as partners who support the work of NITAGs to make them more efficient, more effective in the work that they do in supporting the Ministry of Health? Thank you.
Thank you, Ben, for that question. I think, as you know, NITAGs can be supported through the health system strengthening support that Gavi provides. It's up to countries to decide that. And Gavi requires a NITAG recommendation for any new vaccine introduction. So both of those two levers have strengthened, I think, NITAGs within Gavi countries over the past strategic period. In this new strategic period, I think NITAGs will have an even more important role because countries will need to look at their portfolios, prioritize within them, and optimize within them. And for this, they will need evidence and independent inputs, which are what NITAGs bring. So I think the strength of a NITAG, countries will benefit from a strong NITAG precisely because there are some difficult conversations coming up in a fiscally constrained environment around prioritizing and optimizing their immunization portfolios. As partners, I think we have the duty to ensure that within our limitations, there is enough support or as much support as possible to strengthen these discussions and make sure that they're evidence-based and that countries can make these difficult decisions with objectivity, right, and the best available evidence.
Thank you for that, Michelle. Can I, this is just one from me. You know, the Gavi strategy and support has always assumed the dose, it seems, as a major barrier for immunization and vaccination. But as you would have realized with the last discussion, we're talking about health systems and other barriers that are actually the real barriers. You know, in what I'm going to even talk about in the next talk are the other variables that usually are, I mean, we realize that with the COVAX facility, for example, that a number of donated vaccines in a number of countries in Africa expired without being used because there was no infrastructure for delivery. To what extent does Gavi going forward think that may be an important area of investment if we have to realize the level of coverage that we are hoping to achieve?
So I think the health systems funding was set up with that intent to ensure that delivery systems around the vaccines that are procured are as strong as possible. I think in these prioritization discussions, there's definitely been a focus on the fact that Gavi is mainly a vaccine, provides access to vaccines. So I think the instructions from our board have really been to continue to focus on that a particular part of what we do. That doesn't mean to say that the health system strengthening is not important, but I think there's recognition that our focus is on the other piece. So the health system strengthening will continue, but it will be on a smaller scale, given the constraints. So yes, I hear you and I understand that these are the challenges, but I think given everything that we have, we are protecting the vaccine component for now, more than the health system strengthening.
Okay, I don't see any questions. Oh, there's one last question, and then we are going to say thank you to Michelle for this really thought-provoking presentation. Please.
Thank you for the good presentation. My question is about how Gavi interact with other institutions in the field. I think of CP or are there CP or CPI? I don't know. Yeah, thank you. That was my question.
Thank you.
You mean CEPI? Yes. So given that we provide support for global stockpiles and vaccines for outbreak response. We do have a close collaboration with the ICG, for example, as well as CEPI. We keep a track through what we call living assessments. So a framework of data that we collect on all the priority pathogens of epidemic and a pandemic potential. And we update those regularly in collaboration with partners like CEPI, WHO, and others to ensure that we have the right information to hand. If there is an outbreak, we can review those living assessments in a really quick way and be able to provide a recommendation on whether Gavi needs to respond or not, or whether there's other partners that are better placed to respond to a particular outbreak. So that is one of the things that we do to ensure a timely response for outbreaks.
Thank you very much for this time. Can we give Michelle a round of applause for this time? Michelle, thank you very much for always making the time for supporting not just what Gavi does, but also giving information all the time and being a really, really great partner in terms of the communication strategy about vaccine and vaccine investment on the continent. So greatly appreciated. And thank you very much. And we hope to see you again in another installment of these conferences.
Happy to do so. Thank you very much for having me. Have a nice day, everyone.
Thank you. So the next talk is by me. And I will try not to stand there. As I always say that I leave the pulpit for preachers, and I'm just like a mere mortal. So so what I'm going to reflect on is what people have touched on, the issue of vaccine equity, kind of addressing some gaps and challenges as far as vaccine equity is concerned. Please, because I'm chairing again, please just stop me if I'm talking too long, and I don't want to eat into lunch because lunch is very important. So those are my disclosures. I think I left a couple of ones that I was told were not that important, and those are about safety boards and study advisory. Apparently that's quite okay and it's not a conflict of interest. And I'll tell you, the reason I do this is because I've come to this realization that to speak a bit about disclosures all the time, especially when it comes to the area of immunization and vaccination, the real theory behind all these disclosures is that you talk about it so that people can see what is biasing your thinking and your talk. And there are many people in the States. If you talk up to Stanley Plotkin, who have been hauled before groups for apparently being biased because of potential conflict. And that assumes in many ways that because I may have given a talk for an organization, I will kind of be biased towards a product. But I've really realized that the truth, I don't know how, oh, what do I press again? The middle one? No, this one here. It's supposed to work, right? Ultimate does in the end. But I realized towards the last 2 years, and I took this picture when I was in one of our rural areas in the Eastern Cape, where I'm involved in the work of an organization, that my true conflicts lie elsewhere. And I've always declared this because I found them important to do. This is going to be a lot of work. That my real conflicts are those, if you really want to know why I'm biased, I value people, and I'm committed to the poor, and I'm responsive. And that means that I will always value people before money and even before my job. And the reason I do my job is primarily because I value people. So I'm going to be biased, and it's probably less so to you because you're all from this poor continent. I have to remember this when I'm speaking before what people call a global audience that include high-income countries, that I am blatantly biased because I'm biased the most marginalized and the poor, which are our constituency, the people of our continent. And so that for me is important to note. That is my true conflict, and I don't apologize for it. So when you talk about vaccines, I think the important thing is to realize the context in which they play. There's this picture here, which the other end was, that was from Time magazine in 2015, where the assumption was the child born around that time has got a potential to live up until 142 years. It's not very surprising. Right now, as we speak, the so-called baby boomers, born, just after the Second World War, a third of them are expected to live to 100. And that may look very, very normal in our context. For the majority of the existence of human beings, the life expectancy barely touched 30 until something happened largely over the last 300 years, in which suddenly we gained like 55 years, with 2/3 of that time having been gained for the last 100 years. It's really remarkable overall when you think about it, because we usually assume that most of you would be like old people according to the longitudinal history of humankind. And the reason why this has changed, you'll be surprised, has a lot to do with what we managed to gain as far as infectious diseases are concerned. And I'm going to come to that. I mean, if you use the example of the United States, for example, here, that over the last, am I still going to get the click out where we've given up? We've given up. Sorry, it's now clicking. So if you look at this is the US, like we don't realize that in the beginning of the 1900s, the life expectancy was like sort of mid to high 40s. But over the last 100 years or so, they've gone up to like almost double of that life expectancy. And a lot of that has got to do with this. If you look at the yellow bars in this graph, it's the proportion of people who died of infectious agents and infectious diseases. And you can see how much that has declined. It's really remarkable to think about thousands of Germans dying of diphtheria in the beginning of the 1900s. And that's literally what happened. But now, most of you, if you ask if anyone has seen diphtheria here, you'll say no. And you'll have to come from very specific areas on the world where they've had this, what is seen as an aberration and an abnormal occurrence of disease. And that is the impact that vaccines have made. And probably the person that statement puts this into context from Stanley Plotkin, that if you look at all public health interventions of the last century, only the supply of clean water probably has had a much bigger impact. Not even antibiotics, which was a really phenomenal discovery, have had the same equivalent impact as what vaccines have done to public health. That is how important that is. What does this matter? Well, if you look just at that curve and the life expectancy we're talking about, you realize that these have not accrued equally all over the world. And in fact, if you're African, you are still likely to die sooner than anyone else on earth. Okay? And I just didn't put another slide, not to have too many slides. A lot of those Africans are going to die of infectious diseases, and a lot of those will die of infectious diseases for which a preventative intervention is already available. Okay? That means there's something wrong. So what you're seeing is that access to vaccine is access to life, and lack of equity to vaccines is lack of equity to life. And the greatest losers are Africans. Okay. The interesting part here is that the ones who are poor are probably most likely to benefit from vaccines than anybody else. So if you look at that, it shows the so-called return on investment. This comes from the project, the DAF project that was done at Johns Hopkins, the decade of vaccine econometrics. if you look on top there, you realize an interesting thing, that the poorer you are, like Afro, the more likely you are to benefit from an intervention. And that's for very good reasons. Poor people derive the greatest benefit from vaccines than the rich. And in part, even within countries, if you're rich, you'll probably, even if you die, you'll probably die in a really nice bed in a private hospital. You'll probably also get ICU in the middle before doing the dying part, right? But if you're poor, you just die. You don't have intervention. You may even not even be able to see a healthcare worker. We know even in this city, which has got both so-called first world and third world communities within 5 kilometers of each other, that a lot of children of the poor, they die at home. They don't even die in hospital, even in this city. And that's what happens. So if you're poor, the best thing that you can do is not getting sick. And as a result, it means that vaccines are worth much more to them, because vaccines stop you from getting sick in the first place, because once you get sick, you're going to die. Right. This is just a simple. So, and interestingly, as we've shown, the ones who will benefit the most from these are the ones least likely to get those vaccines. Okay, I'll have to go back. Look at this, for example. This is a story of vaccine coverage, different vaccines. The red part shows poor vaccine coverage, right? You can see the antigens and all and whatever it is. And if I highlight Sub-Saharan Africa, which is in the bottom, we really live on the red, most likely, right? I couldn't separate Mediterranean and North Africa. So, but you can see even then that it's not really encouraging. And we know even from the previous slide, which showed those return on investment, that Eastern Mediterranean and the Afro region are some of the ones that have got the least coverage, but with probably the highest rate of return. And again, this is what you're seeing from here. Okay. So I'm gonna just probably I've always used this example because it probably demonstrates this best, the rollout of pneumococcal conjugate vaccines. And they're probably easy to use because one of the largest killers of children, in particular even in Africa today, is pneumonia than anything else. Lower respiratory tract infection. Okay. There were a number of editorials, including here in the Lancet, that was talking about the disgraceful neglect of childhood pneumonia, but it was being spoken about in the context where there are available and highly effective interventions for doing so, but still the carnage continues in this context. Look at this. I mean, this is a no-brainer. It's just the inverse relationship between availability of conjugate pneumococcal vaccines and death. If you don't get the vaccine, you die. The deaths are higher where children not immunized are also higher. So, and that it's a no-brainer. You all know this. This is not like, this is just obviously what happens. It's this, and this is a slide that shows you what inequity looks like. You don't get the vaccines, you die. You get the vaccines, you are less likely to die. It's just as simple as that. Okay. The coverage looks like this in terms of in the world, but you can see that even the global coverage at that time, I think this was 2022, of pneumococcal vaccine, it's not really been great, even if it has improved. And it's also the time it takes, and we'll come back to that again, right? This commentary used some of this kind of review data to make sense of this phenomenon. And this is just what we've partly spoken about not long ago, with Gavi and so forth. If you look at this slide here, you can see low-income countries that receive Gavi. Suddenly, they start exceeding the lower middle income. So the lower middle income countries are too rich to receive Gavi support, but too poor to afford the vaccine. So they've got poor. What does this tell us? It tells you a very simple fact, that the cost of the vaccine dose matters. The lower middle income cannot afford the dose. The low income can afford it because someone assisted in doing so. This is just boils down to money. Again, to put it simply, if you don't have money, you'll die. It's yes. Okay. Look at this story. The first country to start vaccinating was the US when the conjugate pneumococcal vaccines became available. And you can see how long it took, 10 years. We're lucky in South Africa in 2009, we managed to introduce that, but the rest of the world was still not getting those vaccines, and specifically the African continent. And then 2015, this way we were, and here we are in 2021. I think there's been maybe more coverage now looking at other, but this is much more complex than that, and I'll come back to that, because the fact that a country is now looking blue does not necessarily mean that most of the children in that country are actually receiving the vaccine. Okay, so this is just what countries have managed to do. But what is interesting is that if you reverse and go backwards, those who needed the vaccine the least because they have a safety net are the ones who got it first. The ones who needed it the most got it 15, 20 years down the line. And in the intervening years, they were just doing all the dying that is needed when you don't have money and vaccines. And most of those were African children. They were your children. And why it's important to think about it this way is that we've just been talking about RSV, and we know that the vaccine is effective, it's available, and it works. I just haven't put a slide again of which countries right now have got RSV interventions, whether it's long-acting monoclonal immunoglobulins or the vaccine. But the vaccine right now is being rolled out to the ones that least need it, because 97% of all children that die of RSV, they die in low and middle income country contexts. The vaccines are being rolled out with the monoclonals in high income countries that account for 3% of mortality due to RSV. That is what inequity looks like. And this is in the context where we know right now, for example, in Africa, that pneumonia is the greatest killer of children. So you need to just kind of think about this, okay, about these things as we go. Like put the idea of Sabin and Salk, right? Because given how vaccines are developed and what costs currently and who does investment into vaccines, they gave the patents for their vaccines. If that happened, most of Africa may still not be covered because we may not have that vaccine. Even the Qatar instance that Mike spoke about was out of Salk making its patent available. At the time the vaccine was actually approved by the FDA, a number of other companies had already produced stocks waiting for the rollout impact that vaccine would have. But it meant that that brought the price way down and made it available and affordable for those that needed the vaccine. This had been a really great investment into humanity, with someone saying, how can you have a patent for such an intervention? But the question is, how do you produce the vaccines in such a way that there's a partnership? And maybe we need to have other models. We've had models during COVID where partners could come together, countries, philanthropic entities, and so forth, and industry, and work together to find common grounds where you reduce the risk from one partner in such a way that it becomes affordable for everyone. Otherwise, without doing so, the pattern will be repeated over and over and over again. And what we saw with pneumococcal conjugate vaccines is going to happen with RSV. But the world is much more complex than that. It's why I was asking Michelle the question that I was asking. Even during COVID-19, when the vaccines were actually made available, donated, free, didn't really mean that everyone accessed them. And vaccines, you soon or later realize, I'm sure you know this already, they actually don't work very well unless you give them. You know, it's like toothbrush, you know, like you could have a pile of toothbrushes and toothpaste under your bed, and if you don't brush your teeth, you still have rotten teeth, right? And vaccines work pretty much the same way. So a number of stockpiles of these COVID vaccines expired because a number of countries did not have the infrastructure to be able to deliver them. And that is because there are other variables that you need to worry about, whether it's the healthcare workers to worry about to deliver them. And we know that Africa, for example, has got this major problem where it's got a very high burden of disease, but you can see that small bubble that shows the number of healthcare workers to actually intervene and to actually, whether it's preventive level or even when it comes to intervention. That's why it's not even surprising why our children die if they get sick, because there's no one to actually look after them. So you'll think that what I was saying now is talking about inequity with rich countries getting everything and poor countries all being in trouble, which is probably the case. But you'll be surprised to know that in many ways, most of the divide, the inequity, happens within our countries. And the reason I'm making you aware of this is that I want you to not feel that you're off the hook because someone else is screwing you and your country. I want you to realize how much we are screwing our own countries with inequity that we create within our own context, where we can actually do something about it. Okay. This I just used, Indonesia and South Africa, just showing the inequity across the road within one country. These two groups, they receive different healthcare. They receive different access to vaccines, even if the vaccines are free, because it's much more than what you think about. And that's why I've loved this cartoon before that reminds us that even if you can get the dose in, the dose is not the whole cost of immunization. The cost of immunizing a child is much more than the vaccine dose. During COVID, I think Greg was like in the forefront of thinking very hard about how we're going to do these things in the Ministerial Advisory Committee. We realized later that the cost of infrastructure that didn't exist exceeded the cost of the vaccine doses, even though we were screwed in terms of being made to buy the vaccine doses at a higher cost than our high-income country colleagues. But still, the cost hidden below the surface is probably higher. Infrastructure, cold chain. I mean, I remember when we had the measles outbreak in South Africa during the COVID period and wanted to roll out this campaign. The rate limit at that stage was not even the vaccine doses. We just didn't have enough syringes at that stage because we needed to procure them. And we had to pause that because so it had nothing to do with the vaccine dose. There were other elements that make immunization possible. And that may very well be the basis for having inequity than just a vaccine dose. And some of those things are within our control to change within a country. And the reason, again, why this matters at country level is that you have to really think very carefully about who needs the vaccine the most and what you need to do. And again, I'll reference Greg's NITEC group here when they made the decision about conjugate pneumococcal vaccines. You may think that this is HIV-infected children on the right and uninfected on one side, but look at what the case per 100,000 looks like. Invasive pneumococcal disease was much higher in the children that had HIV. So one needed to think carefully about how to prioritize them in crafting a schedule. And that's why South Africa ended up with a schedule that it has. I mean, at that stage, the choice would have been, let's say, 3+0, because the government could only afford 3 doses per child. That was what was affordable. Greg tells me that they did a back of an envelope calculation. You were allowed to do that. You didn't need health economics that time. Yeah. You just needed Greg with a pen and an envelope. And what the government could only afford 3 doses. How do you give them? At that time, a famous schedule was 3+0. That probably works for high-income countries. But if you gave 3+0, it meant in our context, you stopped vaccinating before 4 months of age. And then the immunity will wane, as you heard Eliza speaking this morning, right? Which meant that those kids, who, especially with HIV, will get pneumococcal disease in the 2nd year of life because the immunity had waned. So you have to worry about them. You can't afford a 3+0 without excluding them from that. So what do? You then have to worry about going experimental at that time and going 2+1. Why? Because the booster dose would allow them to have meaningful immunity in the second year of life. But we also had another problem, and I'll talk about this as we go to another slide. Vaccine coverage fell precipitously in the second year of life. If you look at the vaccine coverage was barely 60% at 18 months, which is the other vaccine point that we knew. So you needed to make sure that you're going to have as high vaccine coverage with the third dose. And so what the plan was then to move the schedule earlier than what high-income countries will do, which would be at a 1 year of age or so forth for the booster dose, and you bring it forward to 9 months where we still had meaningful coverage. But it was geared towards protecting the most vulnerable in our context. That is what equity should look like. So you change the whole schedule in order to accommodate a population that may seem not a majority, which it wasn't, but still one that had the most vulnerable. And you can see what the impact has been. That impact shows that for invasive pneumococcal disease, children with HIV had 60 times the benefit of the vaccine than their counterparts. That is basically what that meant. For every outcome, 10 times more for radiological confirmed pneumonia, 40 times more for bacteremic pneumonia. They actually had much greater benefit. But unless you deliberately say, we are going to make the most vulnerable, who are the ones who are most likely to benefit, a affected constituency, and make sure that the schedule will serve them. Otherwise, they're going to lose out. And that's basically what I mean by in your own context. But here is the rub, and this comes up a number of times, and someone talk about manufacturing and so forth. A lot of vaccine and vaccine trials are not done in our context. And they are not even necessarily the priority. That's why we talk about neglected tropical diseases and so forth, right? Because, and as someone jokingly once said a long time ago, it was talking about someone from Europe, and they said this young guy is like an expert on tropical diseases, which we call diseases. For us, that's basically what they are. And they are neglected tropical diseases because they are ours. And so there's not enough investment. If you look, for example, at Africa, and you look at the number of trials that actually take place, that is the blue line in the bottom. It means the results that we work with have to do with whatever is someone else's priority than our priority. And this is not very safe. I'm just giving an example. This was a RSV vaccine that we had a lot of hope on. This was Novavax vaccine. It was felt not to be very sufficiently efficacious, and so it was dropped. But it was when you break down the data and you realize that in low and middle income countries, it would have had a meaningful impact if you disaggregate the data. But because it was not going to make an impact in those that matter, the vaccine was abandoned. And so it never saw the light of day. That is what happens when you don't have the powers of making those decisions, which is the reason why we need to worry about manufacturing, where you can, as a continent, think about things that will equalize things by making sure that the things that matter for populations are actually addressed as a priority, and the agenda is not set elsewhere. And these are the things we're going to have to grapple with if we're going to have access and close the gaps with respect to our own population. We did this study some time ago. It was Ndambi was our MPH student, did this to realize that equity inside countries can be due to other things. So let's take, for example, pertussis. That's my area of interest. If you're going to die of pertussis, you're probably going to die. You're going to die in the first 6 months of life. Probably the highest in the first 2 to 3 months of life. That means, and this is also what worries me all the time. It's when we look at DPT3, for example, at 1 year of age, because you managed to catch up at 8 months, 9 months. That is when you're already dead. Okay. So equity means not just receiving the vaccine, but receiving it in time to actually make a difference in your life. And in particular, for the most poorest, vaccine coverage happens much later because they're not receiving them when they are at the highest of risk in their lives. And someone has to worry and think about this. And this happens to populations that are marginalized, farm workers, seasonal laborers, and so forth. People who very often have to think and say, do I take this child for immunization and either get fired or lose my wages and not be able to feed the other two? And as a result, they will delay it until the harvesting season is over and then bring the child in if the child has not yet died of an acute infection that could have been prevented because they couldn't bring them in earlier on. And these are the kind of things we have to grapple with. And this doesn't happen just between the Netherlands and Guinea-Bissau. This happens within South Africa, within Zambia, within Kenya. This is basically what we need to address ourselves if we have to deal with the real issue of equity. It's not what other people do to you, it's what we do to our own children. And this is not surprising. That's why we talk about all these zero-dose children. And we know that a large proportion of these are actually African children. You know, I was just talking to colleagues during break time here that even when you talk about zero-dose children, for me, the term worries me, and I've protested sometimes vigorously against it being seen as a problem to be solved. And someone will talk about it later in the week, because zero-dose children, and you know, you can actually even you can find a way of fixing zero-doseism, so you can die later of measles, because remember that clearing zero-dose children does not fix your measles problem. It only fixes your DPT problem. And if you make it a problem, then seeing that this is a symptom of the inequity we have within our community and our society. Zero-dose children don't occur in isolation. They occur within a socio-economic context that we have created within our different countries. And so we need to think more about it as probably a barometer, a marker of what we do, but to fix it, we're going to have to deal with structural issues that impact on the children of the poorest and the marginalized of our community. So just keep that in mind, because that is part of your responsibility. Do I still have a few minutes? Okay. Remember when I talked about about pneumococcal conjugate vaccine and how we're covering this? If you look at all of these countries that are shown there, you'll realize that even though the country has got a conjugate pneumococcal vaccine program, the vaccine coverage is not as good as you would like it to. So the vaccine is supposedly available, but it's not being received. And the outcome is the same. Toothbrush without brushing teeth still leads to dental caries. And that is basically what we're dealing with all the time. And we need to really think about what that means in our context and what we need to shift to make this change. So what it simply means is that this child will probably live 142 years. This one, which is yours, will probably not. This is the reality of what inequity means. Because this one here is your responsibility and your constituency. That graph will continue to digress. You saw Greg showing the gap between the US and Africa with vaccine coverage as more antigens came in. As long as this persists, especially even when vaccines are available, but they are not delivered, this will remain the case. And this will remain impossible for us, the idea that we want to leave no one behind, which is what equity is all about. So for me, and I'm about to conclude here, it's when you start really thinking, I mean, this again in a rural village where a community worker was going from house to house, making sure that this child was going through the road to health card, the child receive all the vaccines and so forth. We're going to have to do something like this to make sure that the children of the most vulnerable, marginalized of our community are actually reached if we have to close the gap. And it's important to remember that equity is not the same as equality. Equity means going an extra mile to reach out to the ones that need it the most. That means you have to think about deploying unequal resources in order to achieve the same outcome. That's what equity actually means. So why am I telling you this? This is my grandfather and I. I'm just putting it there so I can justify using H-Venda proverb. Wamba livindu really well. It means simply, let evil be outed, that it may move and create a place for the good. We have to confront these difficult topics for ourselves when we go back within our community, within our constituency, because it's only in confronting those that we can create a new good out of those conversations, because hopefully they will lead to different actions to deal with it. This is what equity means. Crossing those rivers in order to go and reach the one who has not received a polio vaccine because they live on the other side of the river with no bridge. Okay. So as I conclude, just remember, immunization may have been a really amazing intervention, as I shared earlier on. But we need to worry about equity. We need the courage to address this issue and confront the reality of what we're actually doing. And that disproportionate accrual of vaccine benefits to marginalized must be realized by equity in terms of how resources are deployed. And finally, just remember that we're probably going to need a much more societal and including political will to engage with these things, which is why I was talking about the showing you that the HIPPO and what is required to actually achieve an outcome. So thank you very much for that. That, by the way, in the middle is Ben floating in the Dead Sea. It was when we were in Jordan. Really? It's two of our colleagues, it's Ben and Julie floating. Thank you very much. And I don't know if you've got any questions. It's quite amazing. You can't actually sink in the Dead Sea, even if you try. Even if like you can't swim like me. I tried to sink, I couldn't. Are there any questions or comments? If there are none, that would be really great. You can. Oh gosh.
Thank you. That was a very good presentation, and I think one of my favorites so far. I am glad that you somehow tapped into the things that the research field tends to overlook, especially when it comes to the poor people that are supposed to be our target population. Sometimes we look at immunization success as something that is developing the product and ensuring that it's there. But actually the main thing is, does it reach the target population? Because like you said, if we have products, but they do not receive or they do not reach the people that they are intended to help, then there is no benefit in having them. And I think sometimes it just comes to the point that we have to rate, what is immunization success to us? Is it having the product, but letting it go to waste instead of actually helping people who need it? And I think that's just a comment that I wanted to make, that we need to put certain things into perspective when we develop this immunization programs and everything. Will we be able to have people get vaccinated? What are the measures that we have to put into place to make sure that our target population is actually helped with our research and everything? Thank you.
Thank you. Thank you for that comment. Interesting, I had a conversation last week with one of our really strong partners in immunization through the Wellcome Trust. And they were talking about antimicrobial resistance and the use of vaccines in that space. And they're talking about developing new vaccines for like, you know, your enteric pathogens and stuff. So I was saying, wait, why don't we pause and think, why do people prescribe antibiotics? It's for syndromes, cough, diarrhea, fever. And the second thing is, okay, what pathogens cause that? Are the vaccines for that not yet developed? And the answer is probably that they are, but they're not accessible. So we're thinking about developing new tools where the ones that actually work are not being deployed. RSV is one of those things that we're talking about now. RSV is probably the one virus that's the most pneumonia death in Africa right now. And that, if you access it, there's no point talking about new products because we know what works. We just don't have access to it.
I also just have a comment, and I was thinking about the challenge of seasonal workers. And I grew up in a place where there were lots of farms around, right? And then people obviously move in and out. And I was wondering whether one of the solutions, and maybe someone can speak to this if they've implemented it somewhere else, is speaking to the employers of those that and have them make a commitment to whoever they're employing, that they would support, you know, that those women would be able to immunize their children. So when they move into onto the farm, or they come in for the season, that practice is done, and you know that those children are protected, because there is a real power dynamic and an equal power dynamic there, where people aren't empowered to be able to say, you know, I have a priority towards my child because the financial, the financial situation is such that they have the employer, and sometimes the employers are really not sympathetic or empathetic to you know the kind of the family dynamic and health of the kids that comes with it.
So I mean, this is an important issue. I think the first thing that for me as a pediatrician in particular, I have to always try to remember, it happens with immunization, child who's missing a lot of vaccines, or even coming to hospital late in the course of an illness when it's much harder to intervene. And people would shout at the mother, and I'm sure some of you may have done that sometimes. Why didn't you bring the child earlier? Those parents love their children, and they are making real trade-offs given what other competing interests are there. So taking a child in for immunization who seems well today, but stopping to feed the other two is a real consideration in someone else's life. And the question is, one of the ways what you're talking about now, but could we find other mechanisms of making the vaccines go to the people, you know, go and how? And these are the kind of things that we really have to think about if we have to close that equity gap. So you're absolutely right.
So thank you for the presentation. Yeah. So a lot of the country coverages will mask inequities in marginalized societies, for example, urban informal settlements. How best do we ensure that we tease this out and actually put interventions for that?
I probably will end up giving a very long answer, and I just didn't show other slides. For the last 3 years or so, 2.5 years, I've been involved in a Lancet Commission project of the impact of racisms on child health. And by racisms, we don't just talking about issue of colorism, it's caste system, it's how you treat children of of immigrant populations, and so forth, right? What is really clear when you model any of these variables is that this is a matter of very many complex variables that coalesce to make that inequity possible. And we really need to think about what variables need to be changed in order to address that. And what you'll notice is that even immunization just become one factor within a complexity of things. That is why I was mentioning the issue about not liking dealing with zero-dose season as a problem, because it isolates it from a much more complex societal problem. And so I think what we need to do, which has surprised me a lot over the last number of years, since I started working on systems approach to issues, is that the area of public health, which is where complexity should be a dominant theme and tools for complexity should be deployed, is the one area where we do these narrow silos. You get some epidemiologists who count things. It's like, for example, if you get a measles outbreak, even in high-income countries like North London, you can go and count the number of children that have got measles, and you can count how many of them didn't get all the vaccine doses. But if you don't go delve deeper in a complex and qualitative type of research to ask why that happened and how you address it, you can't fix the problem. You can only count it. And it's easier to count things. And if you're a politician, it's better sometimes to undercount things so you look good. So this is the issue about it. And almost all complex analysis of problems shows that political will is in the center of most of the problems that you have. And political will doesn't even have to be positive. It can even be passive. People just stopping you from doing what you should do because it doesn't suit some outcome. So I don't have a good answer, except that we probably need to think very carefully about not doing the same things we've been doing in the past. And in particular, when how we do things is imposed from outside without listening to those people who are deep in the context. And what we learned with grounded theory type approaches is that those who are within a system and who experience the problem usually have got the seeds of the answer to respond to that question only if you listen to them.
Okay, thank you very much. Thank you very much. Mine is more of trying to secure advice or technical expertise. We've been seeing most of the countries will embark on routine immunization as a program, and also SIS, supplementary immunization activities, which my country is one of the typical examples. We just concluded MR campaign nationwide. In fact, as I'm speaking to you, there's what they call rapid convenience monitoring going on to support to regions in terms of the big campaigns. Now, my issue is like areas that I think now we should now think of shifting from mass vaccination during SIS. We already have a system functional that's routine. Now, why don't we start mapping of areas that we know that we can have this under immunized or zero doses, and we tailor our SIS. You know, vaccines are very expensive, as some of you have alluded. And now we're looking at our infrastructures at government level, we are overwhelmed. Some areas do not have much skilled people to even deliver these immunization services. Looking at all those local contests as challenges, the funders, the amount of, like, we just did the MR campaign, the whole country, right? And our coverage mostly is outstanding compared to other countries. Maybe you know better. But why not, we said, okay, why not by regions, we do a mapping. Which of the areas do we think we are having either under immunized or zero doses? Now we tell the funders, instead of spending billions for nationwide, paying human resources and logistics, why not we tailor this? That part of the resources could have been used for something else, right? Than frequently embarking on that. So that's my thinking. I don't know what would be the implication in looking at in the section between routine and SIS. That's why I want to seek for how will I call it, your opinion. Thank you.
So I've got an inherent bias, again, that comes from my pediatrics, maybe background. I like the EPI program, the routine EPI program. And the reason I like it is not because of the vaccines. I think I say this over and over and over again. The EPI program and EPI schedule is probably more valuable to the child than the vaccine it contains. For most of your countries, it is the only healthcare infrastructure for children to access the healthcare facility, even without the vaccines. If you're a pediatrician, they say there are periods in which you're going to get referrals for patients, for children, either poor development, not sitting, failing to thrive. They are only picked up when they go for their vaccinations, that there's something wrong. If you're in the UK, there's going to be a well baby clinic run by a GP, or I'm just giving an example, right? So one thing which is important, which is why the strengthening of the EPI program, and we need to figure out where, I mean, the SIA is supposed to be a way of gap filler where the program had failed to reach that, right? And I think that's just going to become so important to think about this in a very broad terms. I even asked them, you know, the slide that Greg showed of what the 50 years of EPI had achieved. I even asked, did you model the impact of the infrastructure itself on child health outcomes other than just vaccine? Because remember, although we all care about vaccines, we believe in vaccines, we love vaccines, vaccines are not everything. Vaccines are only important as much as the health they promise to deliver. And part of that, and I know this from a South African perspective, for example, you refuse a vaccine because bringing it in may have other unintended consequences that may undermine the health outcomes. Is there one of the reasons for NAGI, for example, why we did not support the introduction of the hepatitis B birth dose? Because we thought that the impact of that will go beyond just getting another extra vaccine that looks like a good thing. So we're going to have to think about this. Even when it comes to donors, you're going to have in a national context. It's the reason why Michelle was talking about the NITEX and how important they are. The decisions have to be context specific. And sometimes the right thing for your context is to refuse funding from a donor to make you do something that will undermine your health of your children. Going and doing a polio eradication campaign and then having a measles outbreak that actually kills children 18 months later is folly. You need to think about how to manage that when someone says, you have to do this now. And if you sit there thinking polio is not killing any children, but in 18 months, we're going to bury children who have died from measles because there's funding for the polio SIA now. You're going to have to really think about what your responsibility is to your context so that they can derive the greatest benefit of health, not for the donor, but for the children.
I think you touched a little bit of this in terms of doing qualitative research and finding out about the communities that we're going to. But sometimes in such health systems, I don't think we also focus on health information for the people that we're going to, especially the mothers and the families that we're going to. As part of the RSV study at Red Cross, sometimes there is a lot of hesitancy from the pregnant mothers to take RSV vaccines because they don't necessarily understand what it's for and what it is. And I think there is a part that is lacking when we talk about vaccines or immunizations, especially the newer ones, without explaining what exactly we're giving to the communities. And now we did touch on it yesterday, but I don't think as much because we do have a plague now of misinformation. Everybody, yes, does have access to phones, but it's telling them very different information. Not everyone is going to have the wealth of information of going to university or postgraduate or reading research papers. So there is a lot of fear with everyone because online it's just a race for attention. And I do think when we're talking about immunization, we should also, and like being cutting edge, we should also be cutting edge even in disseminating accurate information, particularly in African context. That picture was very striking that you had about poor communities, but information does get there. Even bad information does get into those communities. So sometimes it is very much easier for them to see us coming with like trucks and being like, oh, this is the people that are coming to kill us or give us something that is whatever like misinformation that they've gotten. So sometimes I fail to see the advancement also in health promotion, health communication, and educating people, just like we did when there was the HIV epidemic.
That's what I'm doing my PhD on with Prof. That's what I'm working on. So
I think this will be the last question, right? So that you guys can have.
no.
Okay. But do I have to respond to that? Okay. For me, it's what I was talking about when talking about complexity. The hesitancy or lack of confidence or however you define it issue, and you can sit with Iman and she'll have multiple ways of doing that. It's one of those issues that shows you what complexity looks like. It's just a quick one. When you talk about a simple problem, it's a problem that can be solved by algorithms. Algorithms can be solved, like resuscitating someone who drops dead. It's very simple. You do ABC, you look for airway, breathing, then you give them so many breaths and so many compressions. You can teach anyone. That's basically what a simple problem looks like. A complicated problem needs usually people who are specialists. You tell them this variable and whatever it is, and they will kind of find a way of dealing with that. A complex problem. It's not a complicated problem. Complexity is defined by how difficult it is to determine what the next outcome is going to be based on where you are. And you usually have got very little antecedents that will tell you about what that future is going to look like. And how you engage with complexity is where we've been failing things. And the hesitancy issue has been brought about by multiple variables, including the COVID pandemic and how we managed it, right? One of the things that we learned a lot is what you're talking about. It's not just giving information, it's not listening, right? And you see this all the time, that we already have answers to questions that we have not actually had. And then you start having to solve problems that are created. And that's basically what has happened. And we are in an unprecedented space. And the WHO highlights the hesitancy issue as one of the greatest threats to public health. That is likely to reverse that pattern that I've been showing of gains over the last 300 years because of how we've managed it. So we're probably going to have to think out of the box in terms of how to meaningfully engage with it. The giving of information will need to occur in a thought through context, because it's not just giving information. In fact, giving information sometimes changes nothing because those that you're giving information to have already made up their mind. So let's just, so I just don't have enough time to talk about it now, but this will come up when we talk about the issue of hesitancy all the time.
Yeah,
Sorry, let me stop.
Yeah, we know that over time, but just very quick question, Rodzani, for you. I know that you used to touch on this in your talk on equity, but what is the impact of issues like related to equity and to vaccine access in middle-income countries, low and middle-income countries, on high-income countries?
There are many issues. Okay. Others, I mean, if when I'm talking to different audience, like let's say not global north, I make them I remind them of this. I even quote John Donne, his sonnet, where he says, no man is an island entire of itself. Towards the end of that poem, he says, every man's death diminishes me, for I'm involved in mankind. Therefore, send not to ask for whom the bell tolls. It tolls for thee. And the reason why that matters is that we are living in a global village. Inequity that affects poor people, ultimately, apart from undermining the humanity of those who may think they are more human than those who are undermined, it actually develops their humanity. But secondly, as we are seeing with polio, until the last Afghan child is immunized and safe, the high-income countries will vaccinate forever because they're afraid. The only reason you stop vaccinating against smallpox is that the distribution of vaccinia was so equitable to reach the most unreachable people, so that even the rich now become safe, even though they had stopped vaccinating some years back, but they couldn't really be safe until the last. So we are so much connected that you have to worry and think about what that means in a global context. I'll stop.
Thank you. That's amazing. Thank you. We'll break for lunch. Thank you. We'll break for lunch and we be back at 2:00 here. No, 00.
Afternoon. Guys, I see we in our groups. I hope it's the think tank group. Yes? Good. Can I ask we not do that now and go to our seats? Let's start the sessions just so that we're not too far behind the program, please. So I know most of us are now very familiar with WooClap, and it's going to be our friend for the rest of the week. So now to get us all energized, we are auctioning away another gift set. So the fastest and most correct from today can receive it. Yeah, no, this time I made sure that authentication is off. You should not need to sign in. I'm just going to give maybe 2-3 minutes for people to join, and then we can start. Are we good? Ready? Okay, we'll give it 30 more seconds. Okay, same as yesterday. There's going to be 10 questions. Fastest whilst being correct wins the prize. We're going to start. We're not going to start. The Wi-Fi is a bit slow. As the Wi-Fi loaded, we good? Majority? Okay, let's go. First question. True or false? The COVID-19 vaccine changes your DNA to RNA. Five seconds left. Okay, time is up. And the correct answer? True. I'm very glad no one said true. Imagine what would happen. Okay, we ready for the next one? Coming up. The following vaccines are recommended for pregnant women except. So which one is not recommended for pregnant women? You have 20 seconds left. Ten seconds. 5, 4, 3, 2, 1. Time's up. Do people know what the Do people know what the correct answer is? What do people say? Correct. Okay, most of us are correct. Cool. Next question. We ready? Yeah. Which of the following vaccines are recommended at birth? Guys, I see a lot of deliberation with each other going on. Do you not want to win? You can't win two at once. Okay, time's up for this question. What do we think the answer is? All of the above? Correct. Okay, we're going to move on to the next one. What is the most successful and widely used travel vaccine? Are people giving away the answer? Okay, someone said yellow fever over here. If you haven't yet Picked one. 10 more seconds. Okay, time is up. The correct answer? Yellow fever, yes. Yeah. Okay, next question, number 5. True or false? You only need to get the HPV vaccine if you are sexually active. Five seconds. Okay, time is up. And the correct answer? False. Okay, no one said true. That's a good sign. Okay, let's go. Next question. We have 4 more questions left, or 5. Which of the following confers passive immunity? "15 seconds. "10 seconds left. Okay, 5. Okay, time is up. And the correct answer? Immunoglobulin. Why did a lot of people say natural infection with measles virus? Yeah, can I explain it? Oh, no, I can't actually. No, I actually can't. Although someone, if they do have an answer, can explain. Who chose number 2? Do you want to explain why? Oh, you can't. It's just number 2. Okay, well, for this occasion, number 3 is correct. Let's go. Next question. Development of a safe and effective HIV vaccine has been challenging because "10 seconds left. Four, 3, 2, 1. Time's up. Correct answer? 4. Correct. Okay, let's move on to question number 8. True or false? The HPV vaccine only protects females. False. True. Time's running out. 5 seconds. Okay, time's up now. And the answer, true or false? False. Except for 3 people who said true. Guys? Okay, two more questions left. The next one. Why do we need boosters for some vaccines? "15 seconds left. "5 seconds. Okay, time's up. Correct answer? To maintain immunity over time. I feel like this is a bit of a contentious one. What do we think? Sorry? One is wrong. Okay. Is one right or wrong? Because if it's right, number 2 is correct. Because one can't be. Is it correct? It is correct. Different stereotypes. Do you know what the beauty of this is? I didn't make the quiz. So you're telling me I don't know if it's right or not? We're gonna go to the last question. We can discuss it. We can discuss it afterwards. That's why we're here. Okay, next question. This is the last question. Quick and correct. True or false? Vaccines cause autism. True or false? Okay, time is up. True or false? Who was that one? Who was that one? Guys? Okay, so that's been the 10 questions. Now we need to see who won. Drum roll. Food science. Come on. Well done. Please come up, accept this amazing prize. Congratulations. Yes. So the time and the correct, yes. Who got the correct answer, the fastest? Yeah. Yes. Is there cheating happening here? Okay, that's it for me. Give me like 30 seconds. Can introduce her or yeah, just 30 seconds. I need to download a presentation to this laptop. Yeah.
All right. Thank you for that, Salim. I was having so much fun, I forgot I was chairing. But yeah, so welcome back from lunch. I hope you had something delicious to eat. I did too. And so the plan is to stay awake because we have a very action-packed next couple of talks this afternoon. So it's my pleasure to call up Eloise Dutoit, and she will be talking to us about antimicrobial resistance and its role or relevance for immunization programs in Africa in terms of combating antimicrobial resistance. This is very often a highly topical issue. And just to say that this year we've done something a little different because we get a lot of questions around antimicrobial resistance and immunization and what's the link between the two in terms of controlling disease and managing conditions. So after Eloise's talk, we're going to have a standing panel. So I'll quickly call up the panel and then we'll have a nice, rich discussion. So please prepare your questions and to the online group as well. Please have your questions ready as Eloise speaks. And Eloise, I will hand over the stage to you. All right, can we please give her a warm welcome?
Thank you, Adina, and thank you to the organizers for inviting me to share some information on a topic that I'm very passionate about, which is antimicrobial resistance. My name is Eloise, and I am a microbiologist focusing on bacteria specifically, and I'm here on behalf of the One Health Trust. My talk is going to quickly outline the health burden of antimicrobial resistance. I'm going to talk about how we can leverage vaccines to reduce it. I'm then going to pick out some selected evidence, which we've put together using mathematical modeling on how vaccines, and those are the available vaccines as well as vaccines under development, can contribute towards mitigating AMR. I will then give a brief overview as to who the One Health Trust is, and more specifically the South African group, the Global Antibiotic Resistance Partnership. So in 1928, Alexander Fleming discovered the first antibiotic. And since then, over the next couple of decades, various new antibiotics have been coming into the market to be used and registered. And we find ourselves now, with a problem of no new antibiotics. We now find ourselves in a time where this is what the literature is showing: the overlooked pandemic, which is sometimes also called the silent pandemic, increasing resistance to antibiotics and bacterial infections in humans, and the need for better data. So it turns out that antibiotic-resistant infections kill 1.27 million people each year, and that equates to 5 people every 2 minutes. So that, as you can see, this is no silent pandemic. So globally, bacterial infections themselves kill 7.7 million people a year. Those deaths that are associated with antimicrobial resistance, amounts to 5 million deaths. When we look more locally in South Africa, we have about 78,000 deaths per year from bacterial infections, and those that are associated with AMR is about 35,000. So antimicrobial resistance is stalling the progress towards achieving the one sustainable development goal, which is one of them is child survival. And as you can see from this graph on the right, the horizontal line at the bottom is the target. And you can see that when there is no AMR compared to when the actual numbers, we are far slower in achieving our goals than we should be. So who's affected by AMR? Well, it turns out that absolutely everyone is affected, even if you've never taken an antibiotic in your life. However, it disproportionately affects the most vulnerable, and those include the very young, the very old, the severely ill, and those in resource-constrained settings. So what exactly is antimicrobial resistance? It is a natural process. However, it is speeded up by exposure to antibiotics, as well as several other factors. And what it is what you can see in this diagram in the top left is that what you would see cartoon-wise when you look down a microscope. You have an infection, a bacterial infection, and when that bacterial is susceptible to an antibiotic, as you go across, you can see that the infection is cleared. When you look at the bottom row, that's the resistant bacterial infections. You can see that the little bug colored in yellow, that is one bacteria that is resistant. So you treat with your first line antibiotic, and you can see then that the susceptible blue bugs decrease, but the resistant bugs increase. And so it goes. You treat with the second and the third line antibiotics, and then you are left with your resistant bacteria. And how is it that bacteria do this? They have several mechanisms of adapting and mutating. And as you can see, sorry, is this a point? As you can see in the top, bacterial cell walls can change themselves to not actually allow that little red dot is the antibiotic. So it doesn't actually allow the antibiotic into the bacterial cell to do its work. It can also the bacterial cell can also inactivate the drug, making it useless. The cell can also use funnels through its cell wall to actively pump out the antibiotic. So it is getting through into the cell, but it's actively being pumped out. So again, it's useless. And then it can also do this by modifying the target. So it doesn't actually fit into the little niche spot where it needs to fit to do its work. So some of the factors that drive antimicrobial resistance, that is, they include the overprescribing of antibiotics, patients not finishing their treatments. This is a hot one, the overuse of antibiotics in livestock and farming. This is a really, really, really big, deep black hole of concern. Poor infection control in hospitals and clinics, the lack of hygiene and poor sanitation, and then the lack of new antibiotics being developed. So how can we leverage these vaccines to reduce antimicrobial use and therefore resistance? The WHO has just endorsed this action framework where they outline how vaccines can be used to mitigate AMR. So once an individual is vaccinated, that's the little figure at the top, they are protected from disease. That in turn then leads to preventing complications such as secondary infections, where you would need antibiotics again. It also safeguards communities by providing herd immunity. And all of this together results in a decreased number of infections. If you have fewer infections, you're going to have less antibiotic use and therefore less pressure for the bacteria to develop resistance. I always love this example, although it is a little bit old. It just shows it so well. So this is the introduction of the PCV13 vaccine in 2010. So this is just showing the number of resistant cases. And you can see that at the introduction of PCV13, there was a sharp decrease in the number of cases. Then the little the arrows on the right shows the decrease in resistance to macrolides, 63%, and penicillins, a decrease in 83% resistance. So this is just a great example of showing the power of vaccines. This is from a recent series from The Lancet, where they have shown that really well understood interventions that we currently have can significantly reduce AMR in low to middle income countries. Examples are improving infection prevention and control in healthcare settings can avert 337,000 deaths per year. Access to wash services, which is water, sanitation, and hygiene, can reduce 247,800 deaths per year. And then high priority pediatric vaccines. So by achieving universal coverage of high priority pediatric vaccines, we can avert 181,500 deaths a year. This is a study showing, just highlighting the problem, showing that Africa and Southeast Asia have the highest vaccine avertable AMR burdens in the world. This study analyzed 4,500 surveillance records from 32 million isolates across 101 countries. The main point that I want to, they looked at different factors affecting antimicrobial resistance. And what they found was increasing coverage of vaccines to 100% came out to be the second most effective intervention to reduce AMR. This is a busy slide. The image on the data on the left is just showing you different WHO areas in the world and their AMR associated deaths. You can see that sub-Saharan Africa is way more than the other areas. the image on the right is showing the number of no-dose vaccines as well as incomplete vaccines. The point of this that I want to highlight is here in the Africa region. Can you see my pointer? No. On the left of the graph, that has got the highest number of zero-dose and incomplete dosed vaccinated children, and how that corresponds and is the mirror image of the highest burden of AMR. So now we move into the mathematical modeling evidence. And first of all, I'm going to talk about the vaccines that are available and then those that are under development. So vaccines can reduce antibiotic consumption. If we look at the rotavirus vaccination, you can see in the graph on the left, that is the number of cases of rotavirus attributable diarrhea when there's no vaccination. The next little section is under the number of deaths, number of cases under the 2018 coverage. And then on the right is the universal coverage. And with universal coverage, 32 million cases can be averted. How about the maternal RSV vaccine? It also reduces antibiotic consumption. This slide here is showing the reduction in the number of prescriptions of antibiotics. So against all antibiotics, it reduced the number of prescriptions by 10.9%. And then against lower respiratory tract infection prescriptions, it lowered it by 12.8%. The influenza vaccine, it also reduces antibiotic prescriptions. 11,000 antibiotic prescriptions averted by vaccinating 30% of the population over 65 in South Africa. Almost 8,500 averted prescriptions by vaccinating 30% of population under the age of 5 in Ghana. And almost 14,000 of those prescriptions can be averted by vaccinating 30% of the population aged 2 to 5 in Senegal. So the vaccines can also reduce the transmission of drug-resistant pathogens. So here I want to talk about the malaria vaccine, which may reduce the drug-resistant cases and deaths. And the numbers that they calculated is 236 malaria cases per 1,000 children cumulatively. 0.6 resistant malaria cases can be averted per 1,000 children. And 0.6 malaria deaths can be averted per 1,000 children. However, with an effective vaccine and with the predicted increase in drug resistance to 80% by 2030, the number of averted resistant cases goes up to 10.4. How about the typhoid vaccine? So in 73 of the countries eligible for Gavi support, the routine use of the TCV was able to avert 46 to 74% of the number of cases of typhoid. So moving on to some of the evidence with vaccines in development, the maternal Klebsiella pneumonia vaccine, when looking at reducing neonatal sepsis cases and deaths, it has been calculated to reduce almost 15% of neonatal sepsis deaths averted. Almost 400,000 neonatal sepsis cases can be averted, and 3.4% of all neonatal deaths can be averted. And of course, which is often the case, the greatest benefit is expected in Africa, and particularly in sub-Saharan Africa. This is a very small picture of a post that a colleague of mine did. She looked at the Klebsiella pneumonia vaccine, and through mathematical modeling, they predicted it could avert $6.9 billion across 107 LMICs each year. So globally, we have about 1.3 to 3.9 million neonatal sepsis cases, very often caused by Klebsiella pneumonia. By vaccinating mothers against Klebsiella pneumonia, it could avert $1.17 per capita in Delhi's. Of course, African countries would benefit the most. And this vaccine could reduce the societal economic burden and catastrophic health expenditure. And the problem is, with the increasing resistance to third-line antibiotics globally, this vaccine should be a global priority. How about a TB vaccine? It can also reduce the resistant cases and deaths. In 30 countries, which accounted for 90% of all TB incidents in 2018, an M72-like vaccine with 50% efficacy could avert 10% of RIF-resistant TB cases and 7.3% RIF-resistant TB deaths, and that's between 2020 and 2035. So the take-home message that I want to pass on to you from this section is that AMR is a massive problem that is responsible for more deaths than HIV and TB and malaria together. So now just a quick introduction to the Global Antibiotic Resistance Partnership, GARP, which is a South African group working with the One Health Trust, and it's actually why I'm here today. It was a group, an interdisciplinary group of experts, including Ben, and the idea was to increase visibility and elevate the AMR in the political agenda. So we developed snapshots of AMR burden in the country, and we informed the development of national action plans on AMR. And the meeting and the group resulted in, number one, a perspectives paper that's been published in South Africa, and a policy brief that has recently been launched in South Africa. And the main message of the policy brief is really highlighting the importance of collaboration between these groups, so between immunization and AMR groups. So to conclude, I hope that I've shown you that vaccines are a very important tool to decrease infection and therefore decrease prescriptions and use of antimicrobials. So the modeling data is a really great way of and a good metric to use when considering the value of vaccines and their added benefits. And it's often, this aspect is often an overlooked aspect. Because the costs of AMR are so enormous, the value of vaccines specifically for this case are high. And again, just to iterate that a strong collaborative AMR immunization partnership would help to inform these policies to expand coverage and introduce new vaccines. And with that, I thank you.
Oh, give me a second, Latif. Louise, thank you so much for that. I think that was really brilliant, setting the stage for us. I see Latif has a question. You're welcome to ask just as I bring up the panelists for our session. So Ben, can I please have you? And Tony, please. And Latif, you can pose your question before we start the panel discussion.
Thank you, Louise, for the interesting presentation. Really good stuff. I just wanted to make a comment that we have data coming out of Malawi, which was one of the 3 countries that piloted the malaria vaccine. And the RTSS malaria vaccine, remember we implemented it in clusters, vaccinating and non-vaccinating clusters. And what is coming out now from a cohort study is that it may contribute to the increase in prescribing.
Oh. Yeah,
it's paradoxical. So let me explain how. When a child is brought to a primary healthcare facility, at least in our setting, if the child has a fever, first thing they do is do an MRGT. If it comes back negative, then the clinician at a primary health facility level will prescribe antibiotic because it's not malaria. Now, in the clusters that we're vaccinating, the transmission rates of malaria and the incidence has gone down. That means there are more children presenting with fever, which is not malaria, and then they're prescribing more antibiotics. Yeah, so that's just something to watch out for.
Yeah, thank you. That's a really interesting point. And I think, I was talking to Rudzani about something something similar. I think that also highlights the importance of a kind of antimicrobial stewardship program, very strong stewardship program at the primary healthcare level. Yeah, so thank you.
All right. So Ben has the mic. As I mentioned, we are going to basically have a standing panel discussion. We're honored to have Prof. Ben, Dr. Tony, Eloise as well. And basically they'll start off the discussion and then we will have, we'll field a few questions. I'm sure there are a lot of questions either in the room or online. So Ben, I believe you have a question for Eloise as we get started.
Yeah, first, thank you so much. It's excellent. I think, is this the third time you've given this talk? Fifth.
Yeah.
It gets better every year. So fantastic. So I think just for the context, we've got microbiologists, correct? Clinicians, public health clinician. I call myself a vaccinologist, but Greg disagrees. He says I'm an immunologist. But I think just to start with, the three fields represented here by the three panelists. Starting from my side, when I've been, for the 15 years I've been working in the field of vaccines and immunization, I've looked at the impact of vaccines in the context of reducing morbidity, mortality, hospitalization, making sure that our children don't miss school because they are not sick. And when we look at how we make decisions in terms of should we introduce a vaccine, should we introduce a booster, there is a very clear framework and a lot of concept, we've tackled them. Is it a public health burden? All right. What are the benefits and harms of the vaccine? So when we talk about the benefit and harms for vaccine in the context of let's use this vaccine or let's not use it, we talk about efficacy and effectiveness, all right? And for many decision makers, I've not seen the issue of MR being considered as part of the benefit. And this has been the case because from the immunization perspective, when you design a vaccine, there is what you're expecting the vaccine to do. It is to teach the immune system to do a very specific role, all right? And that is to prevent you from getting infected. So we're not thinking about antibiotics use when you're designing a vaccine and when you're testing a vaccine. But now we've reached a point where we realize, hey, hang on a minute. Besides the direct benefit that we are thinking about vaccines, besides the indirect benefit of community immunity or indirect protection, there are other benefits, which is the reduction of MR. So this is a new field, and looking at it alone, we're not going to get it. We're not going to be able to advance the field of addressing antimicrobial resistance if we just look at it from a vaccinologist point of view. So that is my side of the story. And that is when I met actually Eloise from this partnership, the GAP. And I said, hey, hang on a minute, we need to work together. Because I'm looking at it from the lens of immune system, purely. So Eloise, tell me whether from a microbiologist perspective, have you been traditionally thinking on the immune system or you've been thinking more on the bio perspective?
Definitely not the immune system. And I'm very glad that you're up on this panel because I'm imagining there will be quite a few immunology related questions. So I look more at the definitely at the pathogen side of things, and working in a diagnostic laboratory, it's pretty terrifying to see the number of the increased number of resistant infections that are coming in, and at the rate that resistance is increasing, and the fact that we're struggling to find new antibiotics, we're never going to win the race, because we'll never beat antimicrobial resistance. So this is where the preventative side of things come in, which has resulted in this great partnership. And as Mark Mendelsohn always says, AMR is terrifying, because you can go into hospital now for a standard operation, and that used to be really risk-free and easy, and die from a resistant infection. So,
Thank you, Eloise. Public health clinician.
There we go, public health physician. All right, so I'm supposed to wear a doctor's hat. How many of you are clinicians? Yeah, okay, so some of you know about stethoscopes and mic there. All right, so clinicians usually get blamed for the problem here, with good reason, okay? Over-prescribing, inappropriate prescribing, the wrong antibiotic for the wrong indication and so on. And I think over the years, it's become more closely policed. I don't know what it's like in your countries, but if you practice in one of our academic institutions here, Red Cross, and even the district hospitals, you probably have Professor Mendelsohn or one of his staff coming around once a week and doing something called an antibiotic ward round. And then they go to each patient and they say, "Doctor, why did you put this patient on amoxicillin?" And then you must please explain. And that's great, because it kind of raises the bar and it keeps people on their toes, and it makes them think about what they're writing down there. I used to work with a doctor many years ago. I won't mention his name, but he had a standard prescription for any child under 5 who came through the door. They all got amoxil, they all got Panado, and they all got Demazine, 5 mils of each 3 times a day. It was very quick for him. That's bad medicine. Okay. But there's another side to this story. And so if you're not in one of those academic ward rounds and you're sitting in a clinic somewhere and there's 50 patients outside the door, then sometimes the brain doesn't quite go through the same process. So last Tuesday, I think, is Ruth still there? Yeah, Ruth is still there. So I was at Professor Maloya's research day at Red Cross, and there was a very interesting presentation on rheumatic fever and rheumatic heart disease. Do some of you still see that in your countries? Yeah, you still see a bit. So you know the link between group A streptococcus, pharyngitis, and rheumatic fever and rheumatic heart disease. And what they found in this, and Ruth, you can correct me if I get the details wrong, but it was done in 2 of our primary health facilities here in Cape Town. And the stat that I took home with me was that 20% of the kids, these were under 5s, with symptomatic pharyngitis turned out to have group A strep in their throats in this study, okay? So I said to them in the question time, so now, what are you advising the clinicians? Because the way to prevent that from progressing to rheumatic fever and rheumatic heart disease is to treat them appropriately with an antibiotic. But they can't tell clinically which ones are viral and which ones are bacterial. So my response as a clinician would be treat them all. Give them all amoxil, give them all penicillin, give them all whatever is on your code. And then the microbiologist would come down on my head like a ton of bricks. So how do you resolve that? I mean, you cannot have a microbiologist advising at every single. We have 17 million patient contacts a year in the Western Cape alone. You cannot have a microbiologists are like hen's teeth. They're very rare. You cannot have them advising on everything. We have guidelines, but sometimes the guidelines are put on the shelf and never read. So how do you get around this?
Yeah, Anthony, that's a massive question. First of all, to just point, I'm not a clinical microbiologist. But I think it really highlights the massive extent of the problem. So initially, I wanted to say, well, we need to focus on diagnostics. We need to have point of care diagnostics that can quickly determine if it's viral or bacterial. But you know, the likelihood of that happening in the next coming years, and the affordability, especially for low and middle income countries where the problem is the biggest. Then I want to say strict antimicrobial stewardship programs in hospitals. Yeah, again, who's going to police it? Yeah, so yeah, I can't answer it. It's a massive, massive problem. Yeah.
All right, so we're now gonna open it up. And just to contextualize, the reason we have this panel is in the past, we've struggled to show people the link between immunization and MR. So we hope this has helped, but we're very happy to have engagement with you because it is very important as a tool for advocacy. I think when we talk about the return on investment for vaccines, we still do not have robust methodologies to actually quantify the benefit of MR reduction in that return on investment, which means we are hugely under quantifying the benefits of vaccination. And this is an area that is rapidly evolving from economics. You've heard about modeling side. And the more we can be able to quantify those benefits, the better we are able to engage with policymakers, with our politicians, showing how much return they are going to get by investing on immunization programs. All right. Now, the chair, over to you to.
Right. So thank you all for all of that. Can I check with the room if there are questions? I see 1, 2, 3, 4, 5. Sorry, and you also have a question. Oh, and you also have 6.
First, I want to thank you for this wonderful presentation. Actually, it's my first time. Okay. Actually, it's first time to hear. I am microbiologist, by the way. Okay. Actually, it's my first time to hear about the link between AMR and vaccine. And I want to ask about, you say maternal Klebsiella pneumonia vaccine. I didn't hear about it before. I need to know more about the current situation of this vaccine. It is in the preclinical trial or clinical trial. And the second thing I want to say, can we link the antibiotic usage in livestock do you know that there are also a burden and heavy burden of antibiotic misuse in livestock and effluent under the umbrella of One Health approach? Can we link the vaccine usage to reduce the AMR under the One Health approach?
Yes, in relation to livestock.
There are a big issue and big misuse of antibiotic. Sorry. Also, there are a big misuse of antibiotic in the livestock, in the farming, in veterinary medicine, and you know that's the trend today to treat the AMR not only from human perspective, but from the environment perspective, from also from animals, from the environment. All of this have affect the AMR.
Yeah, absolutely. So currently South Africa is busy updating their national action plan on antimicrobial resistance, and that will definitely take a one health approach, because you can't do one without the other. If you think about the antibiotics pouring into the environment, or this absolute black hole of antibiotic use in livestock, yeah, they have to go together. So I know that national action plans around the world are really focusing on a One Health approach. Yeah, I mean, currently in South Africa, I don't know what the situation is in your country, but you know, a farmer can just walk into a local shop called a co-op and buy kgs of antibiotics, no prescription needed, and just kgs of it, and you know, go and yeah. So very strict legislations have to come into place. Yeah.
How do you link this to vaccine? How do you link that to success of vaccine? You said in your last presentation that if we that the vaccine can reduce the AMR. How is that linked to the One Health approach? I didn't find.
Okay. So I hope I'm understanding correctly. So I mean, the same situation goes for in animals. So they also have to focus on important vaccines for animals in order to reduce their usage of antibiotics and decrease the levels of resistance.
We need more studies. We need more studies in this area.
I think that
to find the association.
Yeah, I think they need to get together and put together their priority pathogen lists. You know, so South Africa, the agriculture department may have a specific list that might be different from, and those need to be the vaccines that they need to target.
And I suppose, Eloise, what the point you're driving at is also that if we can strengthen veterinary vaccination, you reduce the disease burden among animals, and then you don't need kgs and kgs of antibiotics.
Exactly. Yeah. Yeah.
Drug-resistant brucellose.
Yeah.
And then passing on the milk to the human by consumption.
Yeah.
Getting drug resistant.
Yeah. Yes. Yeah.
So we'll take another question here, and then there's a third here. Can we take two questions and then we?
I have a question with regards to, I often see in district hospitals, before Prof. Mendelsohn comes to do their rounds, while patients are admitted, before patients are admitted and they've gone through the emergency room, they often get doses of antibiotics then, and then they get admitted by medicine, and then only then the consultant make the decision on whether this antibiotic is continued or not. And I just wanted to know what the contribution of that 1 or 2 doses of antibiotics, whether that contributes to AMR. And then the other question that I had was, oh, it's just left my mind, so I'll stick to that one.
So I can just quickly say, this is the conversation I was having with Rudzani earlier, is that these patients being treated syndromically, so may or may not be correct, maybe viral, maybe the right target might not be. And then on top of that, it is the duration of the antibiotic also matters. So yeah, the 1 or 2 doses definitely has an impact on the microbiology side of things.
So my response would be, the most important thing is whether there was a bug there in the first place and whether we actually eradicate that bug. So if you start with a broad spectrum antibiotic and the prof comes in and says, no, you need something different, or you need something higher dose or lower dose or whatever, and the bug eventually gets wiped out, then that's fine. You're not contributing to antibiotic resistance. But if you give a stat dose and it turns out it was a bacteria, and then for some reason an incorrect antibiotic is given or it's stopped or whatever, then yeah, we've got a problem. So I think it's very contextual.
From an immunology perspective, vaccinology perspective, think about if the patient is suffering from a vaccine preventable diseases and you vaccinated, they did not need to end up in the hospital. This is the link, because you have prevented infection. And this is how directly actually the vaccine helped prevent the use of antibiotics, because you're preventing people ending up in hospital. So when you look at our coverage of 40%, 50%, it's terrible, because we're missing opportunities to prevent people getting to the hospitals and getting to take those antibiotics. And I think this is the link we want all of you to see, how actually from vaccination, you prevent people ending up in hospital and subsequently getting antibiotics.
So if I may just, if you don't mind, Chair, I mean, what Ben is saying now, I mean, we currently have an outbreak of typhoid fever in Johannesburg. Okay, so there's an alert typhoid fever, and many patients will come to see a doctor with diarrhea, all right, and be given an antibiotic and maybe given the wrong antibiotic, and then start generating antibiotic-resistant typhoid fever. But if they had the typhoid vaccine before, they wouldn't have landed up there. Okay, we have a question here. Sorry.
I think this also kind of just follows on the same topic, just from like personal experience. So I got COVID, I think 2 weeks before the vaccine was rolled out. And following that, then I had a secondary infection that was bacterial and ended up needing antibiotics. And then subsequently, after being vaccinated, I did get COVID again, but I didn't get a secondary infection. So even though I wasn't being incorrectly treated per se for COVID, I think it allows you to prevent secondary infections. I don't know what your thoughts are on that, whether that's, or how much of a role that would also play rather than just incorrect treatment. Yeah.
Yeah. So basically, I may have skipped over it here as well, but I was also, obviously I spoke about some viral vaccinations as well. And what does that have to do with AMR? It's the secondary infections. And I suppose the syndromic treatment that comes into play.
But what could have also happened is that when you got COVID, you got mild COVID, and therefore you didn't get very sick, and therefore you didn't get an antibiotic unnecessarily.
Okay, I think mine is two. One will go to Benjamin. I've heard you saying vaccine prevents infections. I just need clarification whether it prevents you from getting sick, disease, because my understanding of infection is like the virus getting in or whatever microorganism gets into you. Like example, HIV. I may be HIV infected, but not necessarily having AIDS. So my understanding is like if I'm vaccinated with HIV virus, I can still be exposed and have the virus, but I will never get AIDS out of it. That's my understanding. I want clarification from that part, whether vaccine prevent infection or prevent you getting sick or diseased. The other one is about the role of vaccines in preventing or minimizing the use of antibiotics, which is the ultimate goal, as you highlighted. What my understanding about vaccine, they are specific. Each vaccine is specific for a particular disease. Now, and we don't have vaccine for all the diseases, especially that one caused by bacteria. Now, and we all know, I don't know for this area, in my country, like we have broad antibiotics. Somebody come with pneumonia, probably may give amoxicillin. Somebody come with another bacteria infection, they may give amoxicillin. So if we have vaccine for pneumococcal, it can prevent some of the use of antibiotics in pneumococcal condition. But we still have other diseases that we still use antibiotics, all right, which are not, that have no link to any vaccines. So the essence now of reducing the use of antibiotics, there should be a gap. That's my understanding. But if you can elaborate more on that, I'll be very grateful. Thank you.
I answered it before. Oh, sorry. Excellent questions, and I can see you were awake when it was an immunology lecture, because a lot of those things that you've said makes a lot of sense. So the first one, in terms of prevention of infection, or getting sick, or getting hospitalized, or dying. So it depends on the type of immune system you generate. So you are correct that each vaccine is designed for a specific disease, right? And if you look at how the vaccine are designed, you identify a part of the organism that need to train your immune system to generate an immunity against that specific pathogen. So if you are able to generate a very strong immune responses, which we call neutralizing antibody, you're not going to get infected, especially if those antibodies work, all right? So you get the infection, antibodies respond, neutralize, no infection. But very few vaccines are like that. Otherwise, you'll get an infection, but the immunity takes time. manage to fight the infection established, but eventually it resides. So it prevents you from getting hospitalized. And then there are other cases that, there is partial immunity. You possibly get sick. Influenza vaccine are actually some of them that give you sort of some partial immunity. So in that way, you don't, or even if you end up hospitalized, you're not going to die. And COVID-19 vaccines were actually very good ones that had all those sort of 3 or 4 outcomes: infection, severe COVID, and hospitalization and death. All right, and you could see the efficacy of all of them was very different. Like for death, it was almost 95%. For hospitalization, it would go to 70%. All right, the other question you asked was about, you've got broad spectrum antibiotics, but vaccine is for specific. That's correct. That's why we have our routine immunization program saying vaccinate against all these number of vaccines. And that's why we have the pipeline for other other pathogens that have been identified as actually big contributors to antibiotics use. So Klebsiella pneumonia is one of them. And now the Gates Foundation specifically, because they invest a lot on immunization, they are looking at it even in a way that when they are making the vaccines, the antigens being selected are for pathogens which have shown some drug resistance. So there is a lot of contribution to immunization, immunology towards reduction of antibiotics use.
So and one thing, so this whole question about a very good point that we don't have vaccines against every pathogen. And we say, well, yeah, that's what we accept. But why do we accept that? If you read the vaccine journals, and there's like 10 of them, just about every bug known to man, somebody's trying to make a vaccine against it. And what is the reason why we don't have a vaccine yet? And one of the questions I was going to ask, if you look at the R&D spend globally, how much of it is going into antibiotics and how much of it's going into vaccines? And antibiotics is where the profit is, because people get repeated infections, and every time they get an infection, they get an antibiotic. Whereas if we give a vaccine, they don't get the infection. So you stop making money after that. So I think there's a huge problem in terms of our priorities when it comes to R&D. And I don't think we should just accept this adage that I get where you're coming from. I mean, if a kiddie's got an inflamed ear, and they've had the pneumococcal vaccine, they've had the Hib vaccine, but it could be some other bacteria, so we're still going to give an antibiotic. But why is it that we can't get to a place where just about all of the pathogens, which particularly the ones that affect kids, are covered? And it's a question of investment. We've got to get people to invest more in vaccines. Thanks. Yes, okay. Keep your question short and don't keep your answers so short. So we can start here 1, 2, 3, 4, 5, 6.
Yeah, I think most of the things have been covered. So my colleague, thank you so much. But we need to also consider the aspect of economical burden as well, because you talked about how somebody can go to the pharmacy and take just over the counter. It works because going to the hospital is expensive, taking the drugs is easier. I'm coming from a country where this is a practice for everyone. To go to the hospital is very expensive, and a mother has to save money for weeks. That's why some of the children comes when they're already beyond even saving. This is the economical thing. Also in my country, we depend on donation drugs. So beggars cannot be choosers. So you cannot choose which drugs reach you. And whatever you get, this is what you use vastly, especially if it's for free, because most of the regions use drugs that are donated, and they give you the list of drugs that goes. So amoxicillin is the biggest. And then for injectable, we have ceftriaxone. So whatever you have, this is the 2 drugs that you're going to receive. So we have a lot of drug resistance, and this is why, and pneumonia is the second killer in my country. We have malaria number 1, pneumonia, and then diarrhea. That's why we introduced PCV, and then we have rotavirus and malaria vaccine. So it's the part of it is drug resistant. I know we did not do any studies, because we don't have research as well in my country, but there is drug resistance kind of thing happening because any child's come with any cough, regardless whether viral or what, ceftriaxone injectable immediately. So it becomes a problem, and drug management training has been advised everywhere, even for clinicians, because we don't have doctors, we have doctors, clinical officers and these things. So you have some facilities run by clinical officers or nurses, not just doctors alone. So you have many factors that are affecting many players that are playing in the field that is not controlled because of the situation. So we have a long way to go from just what you said. And I think, and I agree, vaccine is will be beneficial for something, for countries like ours to reduce the burden of drug resistance. Thank you so much.
Keep it short, Adele.
Okay. I just wanted to say thanks to Eloise. I thought it was excellent, really, really excellent presentation, because I too have not really looked at the link, okay, between AMR and vaccination. And I think that we're sitting on a golden opportunity here. So when Ben says something around advocacy, I actually start to visualize the fact that in our messages, and now I'm coming from a communications aspect and I'm coming from an advocacy aspect, and I think that's what we should be doing. I think when we're going to speak to the minister, right, where you can affect change, that you should be linking these two together, because what you're doing is you're doing a favor to the vaccination programs as well as tackling antimicrobial resistance. So it's a no-brainer. Okay.
Okay, thank you so much for the great presentation. I think my comment is a bit linked to the previous speaker. So basically, we're talking about vaccination so that we don't get to a point where people get infected and there's no need to use antimicrobials. So then I'm talking from a perspective of, let's think about people in the villages, the deep rural villages, like what Prof. Mureiwa was talking about in terms of the poor people. So how do we communicate such messages? We are all here, we are healthcare workers, we are researchers, but the people that need to hear it are out there in the community. I understand that people do go to the clinics, to the hospitals. They probably get a bit of health talk. But then what about those that can't even afford to go to the clinics? Because I know of people, of clinics that are very far away from people, like what she was also saying. So how do you communicate such a message to the people out there? And also, if we are communicating such messages in terms of, do we focus more on vaccination, or do we focus on both vaccination and also AMR on the other hand? One last thing is that in terms of AMR, a concern can be in the villages. You find that there are people who are educated, yes, but there are also those that are not educated. So those ones that are not educated, how do you explain to them what antimicrobial resistance is? Because you find that there's no direct word that explains what antimicrobial resistance is. I'm saying so because I've heard some researchers do research in my village or villages around Limpopo, where I come from. You find that it's very difficult to actually, talk to the people what AMR is to the people in the village. Thank you.
Yeah. I don't know if maybe you can address that, because I mean, I hope that my presentation again highlighted the inequality of healthcare services in LMICs. So, a lot of the work that's going into, for example, the National Action Plan here in South Africa, I'm hoping that they're going to put vaccination in there. And then also there's going to be a whole section on education where it's awareness and communication. But you do raise a good topic in that, how do you communicate something like this? So I don't know if you can.
Well, we can talk more, but just to. Yeah, and tomorrow, no, when is it? On Friday. I'll talk about it more in my presentation on Friday. But just to say that it is complicated and it requires a number of people right from the top all the way through to the bottom. So, but we can address it then, if that's okay.
Just one little add-on. it is a new field, and as Adele nicely put it, it's a goldmine. And let's not forget, how many of you knew about messenger RNA before COVID-19 vaccines came? Not just to know that there is messenger RNA, but in the context of the platform for vaccine manufacturing. She's an immunologist. And the reason I'm bringing in is that we need to be open to learn. And I can tell you, after these 5 days, you just scratch the surface of vaccinology. In 2019, we had to do a refresher because many of those who have trained for the last 18 years, they needed more information on vaccinology. It's a field that continues to change. So I think we need to look at ourselves as part of the solution, and we need to ask ourselves, what can we do? And the good thing is you've got the support. Immunization is a community, and we can learn how others have done it, because it's not the first time. I mean, there are some countries where they've done a brilliant job in communicating the link and getting the community to buy in that. So you are right, we don't know, but we need to work towards that and feel we are part of the solution.
Can I add something quickly? I think that communications, if you just do a Google search for antibiotic resistance on, whatever, most of what you'll get is about superbugs, huh? Yeah, man-eating superbugs. And actually, that's not that relevant. What we're much more interested in is like the relative resistance, the stuff that's just complicating and we've got to change that. We've got to get that basic message through. Yeah.
Okay, thank you, Alois, and you've done a great job really breaking down, AMR, what is AMR? And because I work around AMR communication as well and One Health. But my question is now on the other side. Recently, I'm seeing a launch of use of azithromycin in newborns, mass drug administration, and recently even combined with our outreaches for immunization campaign. And yes, we know AMR affects mostly newborns. Again, they are most susceptible. But I'm just throwing it out here. What's our thought on mass drug administration of azithromycin in children 1 month to 12 months? It's a new thing launched by, again, some major funders. And currently in Nigeria, some pilots are going on in selected areas. And I'm just wondering, why are we exposing these children to antibiotics at 1 month? And just also putting it out there, maybe perhaps it's the maternal vaccination, should be the acceleration and not exposing newborns to antibiotics. Just throwing it out there if anybody has thoughts on or has read much about this it's called reaching every newborn with azithromycin. Mass drug distribution, and then combining it with vaccination services. It's giving me a bit of concern, and just if anyone has thoughts or anything, I'm not trying, or just the facilitators the speakers on ground. Anyone aware or other countries doing this?
It kind of went totally against the grain because it's not how we were taught, but they did show some benefit to it. So maybe, yeah, I'll reserve judgment, but I'll go and find the paper.
Okay. Just from advocacy perspective, do you think especially after you hear Adele's talk on communication, maybe you can raise a voice that, there are ways that we can use vaccines better to prevent these kind of activities? I don't know. It's just a thought, because the whole idea of this session is to help you understand that link between immunization and MR.
Okay, we've got 2 questions or comments here.
Thank you. Okay. My question is, how can we practically integrate the AMR control measures into our immunization programs?
Well, I think that that's part of Ben's point of having this panel here, is to show that the importance of collaboration. So we can't work in silos anymore. The AMR people can't work on their own, trying to fight AMR, and the immunization people, fighting on their side. It has to be done together. So Ben, do you have anything else to add to that?
So one of the things practically we are trying to do when we train NITAGs to advise the Ministry of Health, so some of the criteria of evidence they need to look at, besides the efficacy and effectiveness, is the contribution to MR. And at any given time, unless if your country has endless resources, is there any country that has, you will realize you need to prioritize, all right? You heard Jason talk about prioritization and optimization. So we might need to start asking ourselves, out of 10 vaccines that we need to introduce, where do we get the most return on investment? Where do we address inequity? And we bring in MR into that. So I think one of the reasons that we start having Eloise here is to see how do we incorporate this into the decision-making framework.
Okay, thank you. My question is, which EPI data can support the fight against MRI? Which?
I think my answer to you is the slides that I saw from Eloise, showing that where coverage is high, there is substantial reduction of antibiotics use. So if you know that if you give the vaccine and you achieve high coverage, then you are addressing MR. I think that is helpful. That is good evidence, isn't it?
So we've got some questions on that side.
Can I just answer that as well? Sorry, quick. So a link to that and a suggestion for the previous question. So who of you work at district health level? A couple of you. All right. So you're supposed to have something called a district pharmacy and therapeutics committee, and you're supposed to have a district antimicrobial stewardship committee. This WHO stuff. They meet separately. They don't talk to each other. Get them in the same room. So when you get the pharmacists talking about drugs, they should also be talking about vaccines. Easy.
Before we go to the right, while I've got the mic, and Eloise, thanks for a great talk, and you can see how it has generated a lot of discussion in the room. But I didn't hear you talk about or mention the issue of rapid diagnostics and point of care, because I mean those are things that can play a role, even in the remote villages of Limpopo, et cetera, et cetera. So do you want to make a comment on that quickly?
Definitely. So I think that rapid diagnostics is the one of the main missing links in solving this problem. As I said, if you had a point of care when you went to the doctor and he took a swab of your skin and was able to immediately identify if it's a virus or a bacteria, that would be absolutely wonderful. So there are absolutely really fabulous diagnostics available. However, they're not affordable for LMICs. I know that the MSF has started a project in West Africa where they're rolling out an app. It's a cell phone app that is going to be used by microbiologists that really kind of guides them through the kind of decision making. It sends alerts, it adds to surveillance, and these are the things that we really need. So rapid diagnostics is the key, I think, to helping this situation, and it's lacking. So, and also then in the remote areas, there are tests that do not require electricity, or they don't need a specialized microbiologist to run. You can get little lateral flow assays that can tell you what kind of carbapenemase enzyme the bacteria is producing, so that you can then guide your decision making on the antibiotic to use. These things are they are available. I know that in South Africa they are used in the NHLS, for example, as well in the private labs as well. But still, they're not affordable for the rest of of Africa.
Thank you.
Okay, a long wait for this one. So thank you. I just want to make some comment. So some people have already now stolen my comments, but I want to emphasize the fact that we really need to bring in the social sciences. So I'm a pharmacist by training, and this is one of my focus areas as well. And we are battling with low vaccine coverage, so we need to get coverage up. We're battling with vaccine hesitancy. So antimicrobial, the use of antimicrobials and vaccination should be paired at all interactions. So we spoke about innovation yesterday. We need, when you go to a patient in the hospital, we've been doing studies. Why can't you see whether this person lying there with XYZ infection has had a vaccine or not? That information is not available. So in community pharmacies, we've just completed some studies now where 42% of people, so we waited outside pharmacies, they came out of the pharmacies with antibiotics bought over the counter. So that's an opportunity. Why do you want this antibiotic? Do you know that, if you had a vaccine, it can prevent XYZ, together with a point of care testing. Another study has shown where people would buy antibiotics for urinary tract infections, which is also unnecessary. Okay, so vaccines come not in there. But the other thing is that I want to mention is vaccine hesitancy. So people are scared of vaccines. They are seen as dangerous and hazardous. But antibiotics are seen as effective and safe. You can just buy them. You can just use them. So we need to really make an effort to pair those two and use each and every opportunity. And because people don't know. Many people said here today, I never thought about it. But people don't know that if you use a vaccine, you are not going to need an antibiotic. And then lastly, people don't understand basically anything about antimicrobial resistance. They don't know the dangers. They don't know they don't know what it means. It's not in their language, especially where we have various languages spoken in one country. So I think there are many opportunities, together with prescribing at primary healthcare level according to the AWARE book of the WHO. Thanks.
Thank you. Thank you. It was a very, very insightful presentation. And I think we have here something for us who are on the ground with many problems regarding the financing. We can take this opportunity to use your research and your all these new things that you are bringing. The dreams that I have now is to see how we can resolve the problem of co-financing. And I think here you are giving us something new. But just the thing that I would like to share here is about, and I think someone said it, but maybe it was not in my, the way that I would like to put it on the table, is about the warehouse. The national warehouse used to be vertical. We have warehouse for medicines, and you are a warehouse for vaccines. And when we look at the budgeting, the Ministry of Health used to prioritize the medicines and leave the vaccines to partners, Gavi and others. So I think this insight could help us. And this, for me, if our colleague of Gavi Alliance and other colleagues here, members of Gavi Alliance, we have there the solution for the co-financing. We can try to see how the increase of the coverage for the routine vaccines can decrease the purchase of antibiotics. Because in many countries, UNICEF, for example, we are the one who provide support to purchase the antibiotics and the vaccines. Thank you.
So thank you so much. So we have two more comments, one here and one online. I'm not sure, Eloise, why the gentlemen have left you.
I know.
Because the panel discussion is not done. But over to you, Jason.
Yeah, thank you very much. I think it's a very interesting discussion. So I just want to make a quick comment. One is that WHO has got a very comprehensive strategy on AMR and framework for implementation. And using these documents, most countries have developed their national action plan on AMR. However, the last time I checked, and I think most countries represented here have those action plans. I don't know how many of you have checked. I would encourage you to check. But what you'll find is that in most of these national action plan on AMR, the section on vaccination or the potential contribution of vaccines to combating AMR is either missing or is not there. And I think this could be an entry point for us to be able to advocate. And actually, although the data is scanty, but there is some data that we can use to sort of advocate for the national action plan to have a section on vaccines and the role of vaccines. Thank you.
So we have a comment from Abdul. I think part of it has already been discussed in the past hour. So Abdul is just saying we also need to realize that aside from the direct benefits gained through disease reduction, there are also indirect benefits. For example, viral vaccines like influenza would reduce secondary bacterial infections, which will ultimately reduce the need for antibiotics, hence resistance. And then he's also mentioning an immunology term here that through herd immunity and vaccinated individuals are also protected, therefore also diminishing the demand and use of antibiotics.
Yeah, thank you. Thanks, Abdul. And also just to add to that, that it also prevents hospital healthcare costs. Also devastating economic expenses for families when the breadwinner is ill, for example. So yeah, there's a whole variety of benefits, yeah.
Yeah, thank you very much for this nice presentation. I think my question is sort of it touches on microbiology and the immunology. I mean, considering that this is a new space, I think from a microbiology perspective, you probably have identified genes that are associated with resistance. And then coming back to the immunology and vaccinology component, is there any appetite from vaccine developers to incorporate some of the genes and antigens from the resistance mutations that have been identified to sort of like develop a pan vaccine that would also encapsulate genes that are associated with resistance? Or is that something that would probably kill the product before it passes phase 1?
I can have a stab at that. So my background's more in TB vaccines, and the message that we used to preach was that vaccines were in fact the only answer to multi-drug resistant TB, because a vaccine will prevent drug-sensitive TB and drug-resistant TB and MDR and XDR and every other type of DR that you want to come up with. So I don't think there was any question about incorporating resistance genes into the vaccine. The vaccine worked against all types of TB, regardless of susceptibility or resistance. That's my initial stab, but I'll give you over to my boss now.
Can I quickly, sorry, Ben? Yeah. So I'm going to skip the initial vaccine. Well, actually to say that what Anthony said is great. To have a vaccine that actually prevents the disease at all, if it's susceptible or resistant, is ideal. And then to think of a bacteria, let's think Klebsiella pneumonia, and say, for example, a new antibiotic that's coming into the country. Being able to identify gene in the bacteria before you treat with that antibiotic is very important. So antibiotics can be effective against Klebsiellas, but not against the Klebsiella that expresses a certain gene. So yeah, Wanda, I'm not sure what you think about the genes in a vaccine, but for me, it's more important a little bit later.
Yeah. I agree with both of you. The interesting thing is that Anela is an immunologist and he has an answer to his question. But there are some disease model already that especially because, I mean, in terms of vaccine design, you want to make sure that you target an antigen that is immunogenic, all right? And if that antigen is related to a gene that could be linked to resistance to antibiotics, it becomes a very key interest area for vaccine design. And I think for enteric vaccines, there's a lot of interest on that. And I think what we're learning from COVID, you know, it's a viral sort of model, which is very different to bacteria. But, you know, you select this protein and it gives you a very good efficacious vaccine, but that protein changes. And then you've got escape from immune recognition and you keep on chasing that. So if you can be able to target those genes that are involved in mutation, generate a protein that can sort of target immune system and generate that type of immunity, then in a way you address that. But this is something that is very important, I'm going to say. Out of all what we've said, this is the most important thing. Africa is the one that is likely to benefit the most by using vaccines to fight MR, right? Most of the data that we have on that link between immunization and MR is modeling. Is that correct? We do not have data. How are you going to convince the policymaker in absence of data? Right, when NITAGs go to make recommendations and they want local data, they go and find it's a black hole. So as a politician or a decision maker, and you presented only with modeling data, and we know modeling is as good as not gonna finish. But I think we need data locally available where we know what resistance levels we have, what kind of contribution immunization is doing that, because that gives us ammunition to be able to use to engage with the stakeholders.
Phew, okay. I think we've outdone ourselves. I also think that we've done something really remarkable, Eloise, Tony, and Ben, because online I'm seeing comments about, oh, I never knew there was a link between immunization and AMR. So I think you're going away. Please don't go away. I think that we have done something remarkable, but just before you all leave the stage, seeing as people, most of us have just been introduced to this link, your final take-home message. I think, Ben, you were raising the issue around we need data. Any final message regarding the link between AMR and vaccines that you would like the participants, both online and in person, to take home with? Quick 30 seconds, sorry.
No, just to say, look, when I talk about data, it can be done. In Israel, when they introduced PCV vaccine, I mean, you also showed data from the US. They were able to tell you to the micro details, the reduction in prescription for antibiotics. I'm sure most of us in our countries, it's difficult to find that data. All right? And I think if we wait for somebody to generate that data, it's not going to come. So when we go back to our country, let's see, how do we work together with other stakeholders to generate data? Because in absence of data, we're now in an era of data-driven decision-making process. In absence of data, it's very difficult for us to have a strong argument and advocacy message that this resort to this. Thank you.
30 seconds. So my take-home message is that we've got it the wrong way around. if you think about something like diphtheria, we spend a lot of time vaccinating, and then you get breakthrough infections, and those are the ones that we have to handle with antibiotics and whatever else. If you take something like TB, you spend a little bit of time vaccinating with a bad vaccine, and then we spend a huge amount of time treating the cases. And there's a couple of diseases like that. HIV is another one, malaria was like that, and there's a couple of others. We need to get our priorities right. Thanks.
Thank you very much. Eloise, the last word.
Thank you. So I think the take-home message would be to stop working in silos and to collaborate, to start communicating between groups.
All right. Colleagues, for their stamina alone, a round of applause. All right. And another round of applause to you for engaging. All right. So I am looking at Salim to confirm that our next speaker is online. Can we check if we can hear?
Can you hear me?
Yes, we can. Could you kindly share your slides?
Yes, let me do that. Please tell me if you start seeing my slides.
Yes, they are up and also in presenter mode. So, ladies and gentlemen, it is my pleasure to introduce Dr. Melvin Sinakis. He's a physician scientist with expertise in global health, immunology, and preventive medicine, and currently the Global Medical Affairs vaccine lead at Bavarian Nordic. And so I'm going to hand over to you, Melvin, to take us through the last talk of the day. You have a big job on your hands to get us through to the end of the day. Over to you.
So thank you so much. And yes, it's always a challenge to be the last speaker of the day, but I will try my best to make this as engaging as I can. The topic is evolving rapidly. We have new data, new recommendations, and really even conferences held just to discuss specifically mpox and smallpox vaccination. So in the next hour, we won't cover everything, but I will try my best to highlight the most important things I want you to remember, and we'll go through some history, biology, vaccination strategies, and I'll also show you what the latest evidence says about vaccine performance and safety. And please feel free to list down your questions as we go along, and we will have some time for discussions after this talk. So, like the speakers introduced, my name is Melvin Sanikas, and I did my medical degree in the Philippines. I studied vaccinology in the University of Siena in Italy. Studied infectious diseases at the London School of Hygiene and Tropical Medicine, and studied other things from the University of Leicester, Leeds, and the Harvard Kennedy School. I currently work at Bavarian Nordic, based here in Switzerland. But in the past, I've worked for the Singapore Health Sciences Authority. I've also worked for Sanofi Pasteur, Takeda, and the Gates Foundation in Seattle, in the US. So I've been working in vaccines for almost 20 years now in different capacities. And today I'm really excited to talk about mpox because I think it's something that is getting a lot of attention, especially in here in Europe and in the US, because of what's happening of the spread of the clade 1b mpox strain. So, before we talk about mpox, I think it's important to revisit smallpox, the original orthopox threat. So, smallpox was one of the deadliest diseases known to humanity, but it was also the first infectious disease to be eradicated through vaccination. And the legacy of smallpox vaccination really forms the basis for how we approach mpox today. And this image from the US CDC reminds us just how devastating smallpox was, with high fever, rash, and a mortality of about 30%. And for centuries, smallpox shaped human history, leading to major scientific breakthroughs, including the development of vaccines. And understanding the biology and control gives us insights into the orthopox family where mpox also belongs. And so in this slide, I just wanted to highlight why smallpox still remains a global threat. Although smallpox has been eradicated, it still poses a potential global threat. And the concern comes from two things: the existence of official variola virus stocks in laboratories in the US and in Russia, and the theoretical risk of recreation using modern synthetic biology techniques. On top of that, related orthopox viruses like mpox are emerging more frequently in humans, and this is why the WHO and many governments maintain smallpox vaccine stockpiles as a precautionary measure. Now let's move on to mpox. So mpox is a zoonotic disease caused by the monkeypox virus, or MPXV, which belongs to the same genus as smallpox and vaccinia viruses. Because of these genetic similarities, the smallpox vaccinia provides about 85% protection against mpox. However, since routine smallpox vaccination stopped in 1980, population immunity has steadily declined, leaving more people susceptible to both mpox and smallpox. So the mpox virus exists as two main clades: clade 1, found mainly in Central Africa, and clade 2, found mainly in West Africa. Clade 2b is the one responsible for the 2022 to 2024 global outbreak. There's also a recently identified clade 1b that emerged in South Kivu in 2023, and the severity and fatality rates vary by clade. Clade 1 infections tend to be more severe with higher case fatality rates than clade 2 infections. So, traditionally, mpox was transmitted from animals to humans, mainly from rodents, which are thought to be the natural reservoir. But in the 2022 outbreak, the pattern shifted dramatically, and transmission occurred predominantly from human to human, often through direct contact during sexual activity. And this change in transmission dynamics has major implications for how we approach prevention and vaccination strategies. So, mpox can enter the body either through broken skin or the respiratory route, but it does not mean it is airborne in the same way as flu or COVID or measles or TB. Transmission via respiratory droplets is possible with prolonged close face to face contact, but true airborne long range aerosol transmission has not been demonstrated in real world settings. Understanding this helps explain why vaccination and barrier precautions are both critical to breaking the chain of transmission. The virus spreads to large organs then, such as the spleen and liver, where it further replicates. So this is the secondary viremia, before spreading to more distant organs such as the skin, lungs, and others. So this is just showing us mpox infection progression. The infection follows a fairly classic progression from incubation to a prodromal phase to an eruptive phase with visible lesions. Typically, the incubation lasts between 7 and 21 days, followed by non-specific symptoms like fever, fatigue, lymphadenopathy, and lesions then appear and progress through different stages. You have macules, papules, vesicles, those with fluid inside, and pustules. In most people, mpox is mild and self-limiting, but complications do occur, especially in those with a weakened immune system. Um, so this slide shows how mpox lesions evolve over time. You can see the different stages of the rash, from flat spots to raised papules. Then you see the fluid-filled vesicles and pustules before you see the crusting. Patients can have lesions in different stages at the same time, and these may appear on various parts of the body. Clinically, recognizing these stages is important for diagnosis and for monitoring the recovery of the patient. So in this slide, we compare earlier Mpox outbreaks with what we saw in 2022-2023. In the past, most patients had widespread lesions across the face, trunk, and limbs, often with hundreds of them, with significant systemic symptoms like fever, chills, sore throat. But during the 2022-2023 global outbreak, the pattern was different. Patients typically had fewer lesions, often confined to the genital or perianal areas, and many had milder systemic symptoms. And this shift shows how the virus can adapt to new transmission networks and behaviors. So, although Mpox is often described as mild, it can cause serious complications, particularly in vulnerable individuals. Historically, we've seen bacterial skin infections, pneumonia, sepsis, and even encephalitis. And in the 2022-2023 outbreak, more local complications were noted. Things like rectal pain, proctitis, or genital swelling were noted, reflecting the new routes of transmission. And what's encouraging is that people previously vaccinated against smallpox tend to experience milder illness and fewer complications. So, even though Mpox is rarely fatal, certain groups remain at higher risk for severe outcomes. These include younger children, pregnant women, people with weakened immune systems, especially those living with advanced or uncontrolled HIV infection. We also saw that individuals with comorbidities or severe skin conditions like eczema may experience more severe or prolonged diseases. So, really, targeted vaccination and early medical intervention are critical for these populations. So, a little bit of history. Mpox was first detected in humans in 1970 in what is now the Democratic Republic of Congo. The virus itself had been discovered earlier in 1958 in laboratory monkeys, which is how it got the name monkeypox. And during the 1970s, cases were limited to several African countries, mostly in Central and West Africa. It was considered a rare zoonotic disease back then, but as we will see, its incidence has increased dramatically over time. And this slide shows how Mpox cases have increased more than tenfold in the last 5 decades. Initially, cases were sporadic and localized in rural African areas, but over time, outbreak became more frequent, and by the 2000s, we began seeing imported cases outside Africa, like the US outbreak in 2003. This was linked to pet prayer dogs. And this steady rise reflects both ecological changes and declining smallpox immunity worldwide. So, globally, from January 2022 to July, middle of this year, more than 158,000 laboratory confirmed Mpox cases were reported across 127 countries. The WHO even declared a global health emergency in July 2022. But as we know, this was later lifted in September 2025, though the WHO still emphasized that the threat remains. Um, and while global numbers have gone down, new clusters, including Clade 1b cases in places like in California, in Japan, Italy, Netherlands, Spain, and several other countries, show that Mpox is not gone. It's likely to remain an ongoing public health concern. So, one of the key features of the 2022-2023 outbreak was how concentrated it was in certain populations, particularly among men who have sex with men. And this really reflects close contact transmission through sexual networks rather than airborne spread. Importantly, this does not mean Mpox is a sexually transmitted infection in the traditional sense, but sexual contact was an efficient transmission route. And compared with older outbreaks that mostly affected children and families, this represents a major epidemiologic shift. And even though cases have declined, Mpox is unlikely to disappear soon. The US CDC has modeled several scenarios from low-level ongoing transmission to possible re-emergence if the virus spreads beyond the current networks. And factors like animal reservoir, international travel, and underdiagnosis make elimination um, pretty challenging. So we need to maintain vigilance, surveillance, and importantly, continued vaccination efforts to prevent resurgence. This is from a CDC modeling. According to this model, vaccination coverage remains low, and the probability of new mpox outbreaks increases over time. In regions with limited immunity, resurgent outbreaks could be as large or larger than what we saw in 2022. But when population immunity increases through vaccination or natural infection, the likelihood and size of future outbreaks drop dramatically. So vaccination isn't just about individual protection; it's really a key tool for long-term outbreak control. Now I will be showing some publications that I think are quite important in terms of this current mpox outbreak. And this article argues that waning population immunity after smallpox eradication, combined with zoonotic spillover and efficient sexual transmission, has driven mpox rise and ongoing global risk. It basically warns us that sustained human to human spread, now involving both clade 2b and more virulent clade 1, this creates space for genomic changes that can affect diagnosis, virulence, or other countermeasures. In mpox and other orthopox viruses, ApoBec enzymes introduce a distinctive pattern of mutations. And ApoBec enzymes are human antiviral proteins that mutate viral DNA. In mpox, they leave a clear genetic fingerprint showing that the virus is adapting under human immune pressure, potentially accelerating its evolution and underscoring the need for ongoing genomic surveillance. ApoBec-driven mutations are not expected to make mpox vaccines stop working anytime soon, but we study ApoBec because it tells us how the virus is evolving inside humans, and it helps identify where future changes might occur, and alerts us if any mutations could eventually, if at all, affect human immune recognition, diagnostic accuracy, or antiviral efficacy. So this study really talks about the ApoBec deaminase editing in mpox viruses. The distinctive ApoBec mutation signature shows mpox has been transmitting human to human since at least 2016. So preparedness must assume ongoing circulation, not a rare animal spillover alone. And if mpox is endemically circulating in humans, control strategies should move from one-off outbreak responses to long-term programs. For example, better surveillance, targeted vaccination, and integration of vaccination to sexual health care. This study is actually quite interesting. The authors did a community-based surveillance method using discarded condoms to detect mpox viruses and other sexually transmitted infection in the global south, specifically in Asia, Southeast Asia. And this is a low-cost, non-invasive way to capture transmission signals, where clinic-based reporting can miss cases due to stigma, due to cost and access barriers. And in the study, from over 20,000, almost 21,000 condoms across 16 countries in Africa, Southeast Asia, and in South Asia, 1.3% contained mpox DNA. Positives were found in multiple countries, and all sequence positives were clade 2b, confirming ongoing regional transmission networks. In this study, the authors support targeted pre-exposure vaccination for men who have sex with men at higher risk for mpox infection, and ideally within clinical trials within 12 months follow-up to document efficacy and safety. And this has been done in some parts of the world. So we have lots of studies that did this, as you will see later. The authors in this publication conclude that the scale of pre-exposure prophylaxis should match local risk and vaccine availability. Of course, with production ramp up to ensure access equity beyond Europe and North America. This DRC modeling study estimated that without vaccination, a 1 year Mpox wave like what we've seen in 2023 would cause uh, around 15,000 cases and over 700 deaths, and shows the greatest impact per dose comes from prioritizing children in endemic provinces. And the study showed that vaccinating those less than 15 years of age in endemic countries, like around 80% coverage, could cut cases by 54% and deaths by 71%, whereas under fives only yield smaller case reductions. So the implication of this study is really in resource limited settings, targeted pediatric vaccination in endemic provinces first is important, and using ring vaccination and surge strategies elsewhere can be efficient and cost effective. So this reports the first observational cohort of clade 1b Mpox in South Kivu in the DRC, and using standardized protocol to characterize transmission, clinical severity, and follow-up up to 12 months. It finds differential initial rash sites by likely exposure route, so genital onset in sexually acquired cases versus hands and face in community-acquired infections. And this paper notes more severe whole body rashes in younger patients. And the author also infer that clade 1 transmission in this setting includes animal to humans and limited household spread, with sexual transmission progressing into broader community spread. Now, moving on to vaccination guidelines and recommendations. So, the WHO recommends targeted Mpox vaccination, not mass immunization. Vaccines should be offered for post-exposure prophylaxis to close contacts of confirmed or probable cases, ideally within 4 days of exposure, but up to 14 days to reduce severity. Pre-exposure prophylaxis is advised only for people at ongoing high risk, such as healthcare workers, laboratory staff handling orthopox viruses, and individuals in high incidence sexual networks. The preferred vaccine of the WHO is the non-replicating MVA-BN vaccine, but replicating vaccines like ACAM 2000 are not recommended for routine or pregnant use. The WHO stresses that vaccination should, of course, complement and not replace surveillance, isolation, and behavioral prevention measures. For people living with HIV, Mpox can be more severe because of the fact that many of people living with HIV may have immunocompromising conditions, or especially those with uncontrolled viral load. The European AIDS Clinical Society recommends both pre and post-exposure vaccination for people living with HIV who are at risk. And the standard schedule is 2 doses, 28 days apart, given subcutaneously, although the intradermal route is also effective. And if vaccine supply is limited, priority should be given to those with lower CD4 counts or higher detectable viral loads. And these recommendations highlight how vaccination is integrated into broader HIV and sexual health strategies. In the US, the CDC's Advisory Committee on Immunization Practice, or ACIP, recommends vaccination for anyone at increased risk. So this includes men who are having sex with men, transgender, or those who had multiple sexual partners, those who have been diagnosed with a sexually transmitted infection, or those who have participated in events where Mpox transmission has occurred. And The approved vaccine is the MVABN vaccine, given 2 doses 28 days apart. And the recommendations, of course, as in other countries as well, they're being revisited as new data comes in. But this really shows how public health bodies are adapting quickly on evolving epidemiology of the Mpox outbreaks. Recommendations in Africa by the Africa CDC and WHO Afro region recommend targeted Mpox vaccination as well, not mass vaccination. Post-exposure prophylaxis is advised for close contacts of confirmed or probable cases. Again, ideally within 4 days of exposure, but up to 14 days if asymptomatic. Pre-exposure prophylaxis is reserved for people at ongoing high risk. So these are healthcare workers, lab workers, immunocompromised, and key populations in hotspots. This is often done via ring vaccination and phased local campaigns. And this is something, of course, that is happening at the moment in several countries in Africa. So there are 3 smallpox vaccines that are being used for Mpox prevention. And these are ACAM 2000, LC16, and MVABN. ACAM 2000 is a second generation live replicating vaccine developed in the US. LC16 is a third generation minimally replicating vaccine developed in Japan. And MVABN is produced by Bavarian Nordic. It's a third generation non replicating vaccine with a good safety profile. And among these, MVABN is the most widely used globally because it doesn't replicate in human cells and can be safely administered to people with immune compromising conditions. There's also a fourth vaccine called Orthopoxvac, but it's only registered and used in Russia. So looking ahead, there are also next generation vaccine candidates in early development. We have the BioNTech who's developing the BNT166. It's an mRNA-based Mpox vaccine. It's supported by CEPI's 100-day mission program. We also have Moderna testing the mRNA 1769, which targets multiple Mpox surface proteins. And early animal studies show strong neutralizing antibody and T cell responses. And these innovations could offer faster and more flexible responses to future orthopox threats. So traditionally, the smallpox vaccines that were used, especially Dryvax, which is now not being used, but even including ACAM 2000, which is still used, these are highly effective vaccines, but they can cause some serious adverse events, such as generalized vaccinia. It can exacerbate eczema and even myocarditis or encephalitis, especially in people with weakened immunity or people who have some skin conditions like eczema and related conditions. And about 16% of people would be considered contraindicated for these vaccines due to risk factors. Of course, the numbers are different in different countries, but around 16 to even 20% of population in specific countries would be contraindicated for these vaccines. And this is why newer non-replicating vaccines were developed to maintain protection while minimizing safety concerns. MVABN, the third generation non-replicating vaccine, is different because it's non-replicating in human cells, and it dramatically reduces the risk of severe side effects. And in clinical trials, around 8,000 participants in all these different trials, there were no cases of myocarditis or uncontrolled viral replication. In contrast, for replicating vaccines like ACAM 2000, it still carries risk for myocarditis. LC16, the Japanese vaccine, has a better safety profile than ACAM 2000, although there's minimal replication can occur. Overall, MVABN offers the best balance of immunogenicity and safety for both healthy and immunocompromised individuals. So according to the label, MVABN is administered subcutaneously, 2 doses of 0.5 ml each, 28 days apart. However, during the outbreaks, intradermal administration has also been used under emergency authorization in different countries. And this method, of course, uses smaller doses, allowing more people to be vaccinated from the same supply without compromising protection. Subcutaneous injection is still recommended for children, people with keloids, or those with weakened immune system. And understanding these routes is important for outbreak logistics and maximizing coverage of vaccines. Um, Unlike MVABN, both LC16 and ACAM 2000 are administered via scarification, using a bifurcated needle to prick the skin multiple times. And this technique leaves a scar and requires trained personnel to perform safely. While effective, it carries a higher risk of auto-inoculation and transmission from vaccination sites to others. And the procedure also demands careful hygiene and follow-up. So while these vaccines remain in strategic stockpiles, they are not ideal for mass civilian immunizations. So this slide is just showing the different approvals of MVABN. So it has now received full approval in several major markets, major countries and regions under different brand names. So it's called Jynneos in the US, it's called Imvamune in Canada, and it's called Imvanex in Europe and in the UK. More recently, it has been approved in Switzerland, Singapore, Mexico, and several other countries. Um, um, Really, each approval covered smallpox prevention initially, but indications have since expanded to include mpox and other vaccinia-related infections. And this is because the animal studies for MVABN actually included other orthopox viruses. So in the non-human primate macaque studies and studies in prairie dogs and rabbits and mice, the efficacy was studied in not only mpox and smallpox, but also in mousepox, camelpox, rabbitpox, and other orthopox viruses. So now we go to the safety of mpox vaccines. So the reason why we have this data for MVABN is because it was the vaccine that was used extensively during the 2022 outbreak. And during the US rollout between May and October 2022, over 1 million MVABN doses were administered. And the CDC monitored safety through their systems. So they have this VAERS, or the Vaccine Adverse Event Reporting System. They also have the Vaccine Safety Data Link and other systems. And about 2/3 of these doses were only one shot, and over half were given intradermally. And the findings match what we saw in clinical trials. Most of the adverse events were mild local reactions, and there were no unexpected safety signals. And this large scale real world data gave public health authorities strong reassurance about the safety of the MVA-BN vaccine. And again, after the rollout, post-marketing safety monitoring became an essential part of our global pharmacovigilance strategy. It's a requirement, especially in other countries where this vaccine is approved. So, among over 1 million doses, serious adverse events were extremely rare, and there were no confirmed cases of myocarditis. There were no confirmed cases of pericarditis, and most reports involved mild injection site reactions, fatigue, headache. These were the most common ones. And again, this is consistent with what we saw in pre-licensure trials and confirms the safety profile of the vaccine in large scale use. Now, when we compare MVABN to older replicating vaccines like ACAM 2000, the difference is quite significant. While ACAM 2000, while with ACAM 2000, myocarditis rates were about 1 in 175 recipients, for MVABN, no cases were confirmed in either trials or in the real world setting, even among people with underlying heart conditions. And this underscores the progress we've made in vaccine technology, where we are able to maintain high immunogenicity and protection while minimizing risks like myocarditis. And it's also why MVABN is now the preferred global vaccine for both smallpox and mpox preparedness. So, because this vaccine was used extensively during the outbreak in 2022, 23, 24, we have all these different studies, real world data from multiple countries, from Spain, from the US, UK, Canada, Israel, and really in these real world studies, some were just one dose, some received two doses, some were given subcutaneously, some were given intradermally, and really a real world setting, right, where all these things were not controlled. And as you can see here in this table, the protection is around 36 to 89% after a single dose, but the protection increases substantially after the second dose, reaching up to 66 to 90% against symptomatic disease. And these studies also showed that vaccinated individuals who do become infected tend to have milder illness and shorter duration of symptoms as compared to those who are not vaccinated. And these findings support completing the two-dose series for optimal and longer-lasting protection, especially in high-risk individuals. At this point, it's important to note that the correlate of protection for mpox remains unknown. In other words, we don't really know yet. We don't really have defined neutralizing antibody levels or immune markers that predict protection against clinical disease. Most studies have measured antibodies against vaccinia or MVA itself, not necessarily against the monkeypox virus, which limits what we can, of course, interpret. And it's still unclear which specific monkeypox virus immune responses, is it humoral, is it cellular, that drive the protection observed in vaccine effectiveness studies. Most probably it's both, but we can't really tell which one is more important. That's why at Bavarian Nordic, we are supporting studies and we are conducting comprehensive literature reviews to better understand MVABN induced immunity and potentially identify the correlates of protection for mpox in the future. So beyond antibodies, MVABN vaccination also triggers a strong T cell response, which is key to viral clearance and long-term protection. Studies show that activation of both CD4 and CD8 T cells and this response becomes stronger after the second dose. In one study, mpox-specific T cell responses were detected in most participants after a single dose, and these responses persisted after two doses. There's also evidence of increased interferon gamma producing T cells, highlighting a robust cellular immune response. Interestingly, in people living with HIV, both MVABN vaccination and mpox infection were able to generate lasting orthopox-specific T cell responses, which is a very promising finding. Also, some studies have shown that while neutralizing antibodies are of course waning as we expect from other diseases, we still see that the people who have been vaccinated 2 years ago, they are still protected until now.
Melvin, you have just 1 minute remaining.
Yes, and I'm ending. So these are my two last slides.
Perfect.
So, thank you. I think as a conclusion, mpox is a zoonotic orthopox virus infection endemic to West Africa, Central Africa, but it's now, as we've seen, in many places all over the world. While deaths are uncommon, immunocompromised people face a significant higher risk of severe disease. The WHO advises both pre and post exposure vaccination for those at high risk. We have 3 smallpox vaccines that can prevent mpox, but MVABN is the preferred vaccine for immunocompromised individuals because of the safety profile. And I think this is something that I really want to highlight. Mpox is likely here to stay. Outside Africa, we will see low level network focused transmission with periodic flare ups rather than community spread. The right approach is targeted vaccination, pre exposure for people with ongoing risk, and post exposure for close contact. Um, also, we should hold off on general population vaccination at the moment until we better quantify the morbidity and program impact of vaccinating more vaccinating the general population. Although it's often framed as a self-limited rash, growing evidence shows that mpox has long-lasting sequelae, a higher risk in the immunocompromised and pregnant women, and also some meaningful sexual health complications. So I think we should know more about mpox. We already know a lot about mpox since 2022, but I think we still don't know more, especially with the morbidity that it can cause for those who have been infected. And with that, I thank you all for your attention. I know we covered a lot in a short time, and this is truly an evolving field with new findings coming out every few months or even every week. Today's session is just a snapshot of where we are. I hope you learned something new, and with that, I am happy to take your questions.
Melvin, thank you so much for that. I must say, I think you caught everyone's attention with that condom study. That's the one that got us. But
Yes, that
really,
got me too.
yeah, a great presentation. And perhaps I could start with the questions, just asking from my side, in terms of, I guess, a comprehensive approach to controlling mpox, especially in countries that are experiencing, a significant burden. What are the additional public health measures in addition to the targeted vaccination approach that we should be looking at?
Yes, a very important question. So there was this study that came out in was it viruses? ID reports, infectious disease reports recently showing that in the U.S. at least, um, the vaccination in behavioral change, of course, helped with the outbreak of mpox, right? But they estimated, this was a modeling study using CDC numbers, that vaccination averted around 8,000 cases of mpox. Behavioral change averted around 5,000 cases of mpox. But if you combine them, these two averted over 53,000 cases of infections. So I think it shows that if you combine strategies, they're stronger. So vaccination plus behavioral change, these two, if you combine them, they are able to avert more cases of mpox. So I think it's similar to other diseases, right? Like COVID, for example, or flu. If you have vaccination plus masking or distancing, you increase the effect of these public health measures.
Okay. I don't know if I missed that, but my question will be, given the availability of the mpox vaccine, what are the main considerations for choosing targeted vaccination strategy over mass vaccination? Is it like because of the side effect, or is it just not cost effective?
Well, so I think this is something that specific countries would be able to evaluate depending on the situation, because the vaccine. So one, I think we know that the vaccines are not available everywhere, right? So, for example, we have some, at least for MVABN, we have some vaccines um, in Africa CDC. And I think they're trying to allocate these doses in the different countries that need it. But of course, we are not able to vaccinate everyone who's at risk, right? So we really need to vaccinate only those at risk or do the targeted ring vaccination. That's one. The second thing, I think, what is being asked of us, which I think you guys can help us, we of course we need to show data, right? We need to show data that the vaccine can be used with other vaccines in the EPI. So, for example, if you want to give this to kids, is this vaccine okay to be given co-administered with the vaccines in the EPI, right? So we don't have that at the moment. There are some groups that would like to study that, and hopefully we will see these data in the next few years.
Thank you for the presentation. I'm just wondering, a follow-up question to the previous one. Is the vaccination of the high-risk individuals or populations, is the recommendation to vaccinate even in countries where there've been no cases or very few cases?
So you're asking if vaccination of high risk in countries with very few cases, right? So I think it really depends on the epidemiology of the country. But if you ask me, this is my personal answer. I think if the country has 0 cases of mpox at the moment, maybe that would not be really recommended. But if there is some transmission, I think it would be better to have those high risk vaccinated.
Melvin, Michael here. Thanks for the excellent presentation. Can you talk to us a bit about the post-marketing surveillance? We've had a lecture on vaccines safety this morning. In particular, the mRNA constructs and myocarditis.
Oh, so you're asking about the safety risk of myocarditis and mRNA? Is that your question?
Yeah, and what measures are in place for post-licensure surveillance?
Okay, so as far as I know, for the studies that are being conducted for mRNA mpox vaccines, of course, they have the standard safety. Measures in place, right, for the studies. Of course, the adverse events are being monitored. I think it depends on the vaccination schedule. For example, for the MVABN, because it's a 2-dose schedule, the first 14 days after the second dose, you have a more targeted safety assessment. And then after that would be less, I would say, not as strict, but of course then in the next 6 months we are still following up for severe adverse events and those adverse events of special interest like myocarditis, right? Now, having said that, I actually don't know what's really happening at the moment for the mRNA trials because I think they just started, but I would imagine that the safety assessments would be similar to the MVABN vaccines as well.
Hello, thank you so much for the wonderful presentation. So my question is regarding the fact that mpox is not considered an STI/STD, despite the routes of transmission being mostly sexual. And maybe I understand there might be a stigma component attached to that, and it doesn't change the epidemiological reality. But is there some merit to classifying it as such in terms of public health? Because, for example, for people living with HIV may get treatment in reference centers and the staff is more adept to in terms of offering counseling and messaging and risk communication. Do you think there's merit to that?
Yes, it's a very good question, and I agree with you. It's because of one of the reasons is because of that, right? The stigma, if you call it a sexually transmitted disease. Having said that, I can share with you that in countries like the U.S. and Germany, it's actually being integrated now. They're trying to do this in the sexual health clinics. So in clinics where they are really treating or diagnosing people with STIs, mpox vaccination is being a recommended vaccine. So it's a sensitive discussion, but I think that is one of the directions of mpox, especially now that we are seeing it in sexual networks. So it will be like that moving forwards.
Hi, Melvin. Thank you so much for the presentation. I have a very quick question. I'm not sure if I may have missed this, but is there any consideration to run some of these vaccine trials in the in the continent, Africa? I'm asking because oftentimes you get these technologies and products being investigated in the global north, and then they just come here to be implemented
Yes.
without
Yeah.
any consideration for the differences in climate, in
Yeah.
genetics, and all of that.
Yes.
So
So
just, I was just
very
curious.
good question, and thank you for asking that. There are actually studies happening in Africa. Some of them are supported by us. Some of them are not supported by us. But so, for example, there is this study in 2 to 12 years of age. It is not yet published, but it was done in the DRC in Uganda. Also, there is a study ongoing, also in the DRC in Uganda. It's looking at pregnant women, vaccinating pregnant women, maternal vaccination, seeing if the antibodies are also transferred to the newborn when they drink their mom's milk. So there are several studies happening, at least in the DRC in Uganda.
All right. Thanks, Melvin. I think you're speaking to an audience from 40 African countries. And I know Bavarian Nordic is relatively new compared to other vaccine manufacturers that are quite well established on the continent. And I hope it would be okay for your details to be shared with our participant in case they want to speak to you and establish connection, because that will be great.
Of course. Thank you. Yes, I would like that. In fact, I was there in Cape Town last week for the ISV Congress, and I would really love to be there with you guys face to face. But I had a meeting today, so I had to be back. So, but yeah, please, yes.
Yeah, I'm sure most people would want to know, how did you select those countries for clinical trials? Because, during the first session,
Mm,
we had how Africa is underrepresented in
mm,
clinical trials, and we are the continent now has the highest challenge with mpox.
mm,
But I wanted to ask two questions. One is on the immunology, because you say there are no correlate of protection.
mm.
And, this is the vaccine both for smallpox and for
And
mpox.
inbox, yeah.
Are you telling me we eradicated smallpox without knowing actually how the vaccine works immunologically in terms of the kind of immune response that it induces and how it confers protection? So that one question. The other question is on the programmatic aspect of targeted vaccination. So in Africa, and I think this is something that has been discussed at a regional level, anytime you start talking about, sexually transmitted disease, men who have sex with men, these are some of very culturally sensitive issues in some settings.
Yeah.
And actually, even in absence of cultural sensitivity, we've seen targeted vaccination doesn't work very well.
Mm.
So what are some of the lessons learned, best practices in making sure that targeted vaccination actually work?
Mm.
And maybe the very last one, when you talked about the efficacy, I think you mentioned about pre-exposure and post-exposure. So
Mm.
what's the efficacy for both pre and posts? Thank you.
Yes. So the first question is, um, am I saying that we eradicated smallpox without knowing the correlate of protection? I think so. So those vaccines were discovered and were used in an era where we are not yet that advanced in studying immunology. And even now, right? If you look at the studies that are being done by BioNTech or Moderna for their mpox vaccines, it always focuses on neutralizing antibodies, right? But we know that that's just not the antibodies that are in our immune system, right? We have the complement fixing antibodies, we have the non-neutralizing antibodies, and not just the neutralizing antibodies. But we focus on the neutralizing antibodies because it's easier to study, right? And the other thing is, of course, the T cells as well. Is now we are studying it better because of COVID and because of what we learned from from COVID in terms of T cells. But I think we I think we need to look at all these components to be able to really look at what is the correlate of protection, because there are individuals with very low neutralizing antibodies, but they are still protected from mpox. And why is that? So I think there are other parts of the immune system that are working there, right? That's one. Second question is what again? What was your second question again? Sorry.
The programmatic aspect
Yeah.
of targeted vaccination.
Yes, and also
Like I say, pre and post
pre and
exposure.
post. Okay, yeah. So for all these studies that I've shown, the real world studies, it's difficult to show which one is pre-exposure, post-exposure. But I would say that most of these in the US and the UK were pre-exposure, and some of them were post-exposure within the 4 days period. And so, yeah, that's the short answer. We can't tell which is pre or post, but it's both pre and post-exposure, because these are basically real world evidence, right? We were not controlling the study like in clinical trials. And I think the third question you're saying is you ask about the stigma, right? Especially in population, men having sex with men, and vaccination. So I can share that in countries like, for example, in Thailand, right, where this was also a like a big stigma in the past. What has worked is they had um, health centers or health units where people can actually go without registering their names, without giving personal information. And I think this has helped make vaccination more accessible without people being scared that they're recorded, they're going to be branded as men having sex with men, and something like that.
Thank you. Back to the chair.
Thank you, Melvin, for a wonderful presentation. So I speak for the online participants, and they're very happy with the presentation. So I'll just highlight a few comments that we have online, and we do have one question as well. So there's just a comment from Cornelius, who just mentions that the limited vaccination in Cameroon, or very limited stock in Cameroon, has led to issues with outbreaks before. And he just mentions that they've seen cases once in a while, but vaccination has never been an option. And he just mentioned that the last case that Cameroon had was in 2023. And then we also have Dr. Sambo, who just mentioned that in Sierra Leone, there is strong technical and financial support from multiple partners. But he just mentions that they're still having issues with persistent outbreaks of mpox. So I think Dr. or Abdul, you had raised your hand. Please go ahead and ask your question.
Thank you very much, Dr. Melvin, for the very exciting lecture. I am Abdul Jalal. It's very inspiring to meet an alumni of the Siena program, which I am currently part of, still much a student under Professor Su and Ralph Clemens. I have a question regarding global preparedness and access to, you've highlighted a lot of points, a lot of data showing that the MVABN remains the preferred candidate for a lot of whole reasons. Now, my question is, in terms of future or potential outbreaks, how do we avoid bottleneck of a single manufacturer? Now, given that Bavarian is the sole manufacturer at the moment, are there maybe stockpiles, or are there active discussions on transferring technology, regional licensing, or pool procurement? Thank you.
Yeah, thank you, Abdul. Very important question. So yes, so there are stockpiles of the vaccines, and at least for MVABN, I know that it's really coordinated with the Africa CDC, and with in Europe, for example, it's coordinated with the ECDC European Council group, In the US, of course, they have their own. From the Congress I was in Cape Town last week, I learned that companies like BioNTech work with the WHO to have this mRNA facilities in Africa. So I think this is one of those mechanism. And BioNTech, as I've mentioned, they have their own mRNA mpox vaccine in development, and they have some sites in Africa and also in Asia. So I think this is trying to address that fact that no, we have 3 vaccines at the moment, but of course it's not enough for the whole world. So we have BioNTech and Moderna who are also trying to do this, right? Increase the production capacity. And for us in Bavaria Nordic, we're trying to change the manufacturing, the way we do the vaccine, because at the moment we're doing it with a chick embryo fibroblast (CEF). We're trying to do a quail cell production, which is faster and produces more yield than the CEF. So I think we are also trying to improve the process so we can make more doses in the future.
Thank you. What's the technology again? I've forgotten the platform. No. The platform for the vaccine.
mRNA for BioNTech and
No,
BioNTech
not. I mean the current the platform for the MVABN used. I think
it's called MVABN basically. Yeah. So MVA means modified Vaccinia ankara. So it's a Vaccinia virus that's modified, and it's from Ankara, Turkey.
Thank you.
All right. Melvin, just thank you so much for taking the time to speak to us. I've been keeping an eye on the discussions in the room based on the questions. I've been keeping an eye on our online participants on Zoom and some external stakeholders who are simply listening in via YouTube. And I can say to you that everybody really, really appreciates this particular talk. So thank you so much for breaking it down for us and your time. Thank you.
Thank you. Thank you so much, everyone, and enjoy the rest of the course. Thank you.
Thank you. Okay, so we will be saying goodbye to our live streaming participants on YouTube. And basically that brings us to the end of the talks for today. So that's the end of day 2, but not just yet. We are going to move